Institute in charge, CCRAS, Regional Ayurveda Research Institute, Gwalior. Our keynote speaker, Dr. Mukesh Dubey, Associate Professor, Swedish University of Agriculture Scientist, Sweden. Dr. S.I. Alam, Scientist F, DRDO, Gwalior. Honorable Pro-Chancellor, Lieutenant General V.K. Sharma, Ati Vishish Seva Medal. Officiating Vice Chancellor, Professor Dr. Anil Vashist. Pro-Vice Chancellor Research, Professor Dr. M.P. Kaushik. Director AIB, Professor Dr. Vinay Dwivedi. Dean Academic, HOI, HODs, faculty member, participants, and the students of MET University, Madhya Pradesh. I, Dr. Asha Singh,
I, Dr. Prachi Dikshit,
AIB, feel privileged to welcome you all on behalf of MET Institute of Biotechnology, MET University, Madhya Pradesh, to the third International Conference, Recent Advances in Biotechnology and Nanobiotechnology 2014. Today, we all are gathered here as a wisdom comes not from age but from education and learning. As the genius said, the aim of education is the knowledge not of fact but of values. It is an occasion in which we get opportunity to get our knowledge and understanding. We look forward to get an exposure about what the best of brain thinks about the emerging techniques. To make this event a blessed one, we invoke Goddess Saraswati by Saraswati Vandana for seeking the choicest blessing. For the same, I invite all the dignitaries to lighten the lamp and garland to Ma Saraswati. I request
शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्नमोऽस्तु ते शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः
thank you very much, sir. I would like to welcome our dignitaries on the dais. For the same, I request Honorable Pro-Chancellor Lieutenant General V.K. Sharma, Ati Vishisht Seva Medal, to kindly welcome our Chief Guest, Professor Dr. P.S. Bisain, former Vice Chancellor, Jivaji University, Gwalior, with a sapling. Thank you, sir. Now, I request officiating Vice Chancellor, Professor Dr. Anil Vashish to kindly welcome guest of honor Dr. A.K. Meena, Institute in charge CCRAS Regional Ayurveda Research Institute, Gwalior, with a sapling. Thank you, sir. I request Professor Dr. Vinay Dwivedi, Director AIB, to kindly welcome Honorable Pro-Chancellor AUMP Lieutenant General V.K. Sharma, Ati Vishik Seva Medal with sapling. Thank you, sir. I request Professor Dr. Vikas Srivastav to kindly welcome officiating Vice Chancellor Professor Dr. Anil Vashisht with a sapling. Thank you, sir. Now, I request to Professor Dr. Raghavendra Kumar Mishra, AIB, kindly welcome Professor Pro Vice Chancellor Research, Professor Dr. M.P. Kaushik with a sapling.
Hello.
Thank you, sir.
Thank you, all the dignitaries. The guiding force behind our constant effort is Honorable Pro-Chancellor Lieutenant General V.K. Sharma. I feel immense pleasure to introduce you Honorable Pro-Chancellor Lieutenant General V.K. Sharma Ati Vishesh Seva Medal. Dr. Sharma is an alumnus of erstwhile King George School, Shimla. He did B.Tech from College of Military Engineering, Pune, M.Tech degree from Pune University, and MMS from Osmania University, all in first class with honors. He joined the National Defense Academy and passed out as a permanent commissioned army officer from Indian Military Academy in 1972. He was granted a President's Commission in the Corps of Signal in the Indian Army. He served the Indian Army for almost 40 years and retired from the second highest rank of Lieutenant General, which is the 3-star and the flag rank of the Army. He participated in Operation Rakshak, Operation Vijaya during Kargil War. He also headed military delegation to France, Germany, USA. For his meritorious services, he was awarded Ati Vishish Seva Medal by Honorable Presidents of India on January 26, 2011. General Sharma is an excellent academician too. Under his able guidance, MIT University Madhya Pradesh has grown by leaps and bound towards excellence and has been awarded as the best private university of Madhya Pradesh. May I now request Honorable Pro-Chancellor Lieutenant General V.K. Sharma sir for his opening remark. Sir, please.
Good morning to all of you. Professor Dr. P.S. Besant, the former Vice Chancellor of Jivaji University and an expert in research on cancer. He has written several books on cancer and he is a known person for total cancer research in India. Dr. A.K. Meena, the officer in charge of CCRAS Regional Ayurvedic Research Institute, who is our revered guest of honor. Dr. Anil Vishisht, officiating Vice Chancellor. Dr. M.P. Kaushik, the Pro Vice Chancellor Research and Dean Research. Dr. Vinay Dwivedi, the convener of the workshop. All HOIs, HODs were present here, and my dear students and the participants who are also listening to me online on this conference. The topic for today, advances in bio nanotechnology, been made in the recent years. Biotechnology is involved for all of us. All of us are getting affected by biotechnology, and all animals also, plants also. And the research which is going on in biotechnology certainly affects our well-being, our health, and our physical part of the body. And physical part of the body also affects us mentally also. Recently, we have just overcome the COVID wave in which all of us were affected, and the universities, schools, colleges, the whole of the country, whole of the world was shut down for nearly about a year and a half, which it continued. And ultimately, the viruses which were found were thanks to biotechnology and the pharmacists who worked hard to find a treatment for that. Nanotechnology is very important aspect, which is the sunrise technology today, and in which the nano subparticles, nano means 10 to the power of minus 9. That is the subparticle, that means 1 divided by 10 to the power of minus 9. 1 followed by 9 zeros. So such small particles, they are so small that the characteristic of the metal from which they are made changes. Like gold is yellow in color, but the nanoparticles of gold, they are red in color. Similarly, the nanoparticles of a metal which is a conductor, they become, they conduct less or they start conducting more. The properties change. Carbon nanoparticles, they become so strong in strength, their textile strength and compressive strength increases suddenly at that level. So nanoparticles behave in a different manner, and the relationship of nanoparticles to technology as far as biochemistry and biotechnology is concerned is a field in itself in which not many changes have been made. Nanoparticles today are being used for drug delivery. All treatment of cancer today, it is done by chemotherapy, which is the normal mode of treatment for cancer. But the chemotherapy severely affects the good cells also. Chemotherapy is like trying to kill a mosquito with a sledgehammer. So the treatment is so heavy that the good cells or the healthy cells are also getting destroyed along with the cancer cells. Cancer, after all, is a multiplication of cells at much higher rate, their production. So it is they start expanding in such a way, the cancerous cells, that they block the normal growth of normal cells, and the affected area starts growing in a tremendous manner. Right? Madam, please, third row. Madam, red, आप खड़े हो जाइए, please get up. Yeah, you know it. You know I am whom I am referring. Just get up. Get up, please. Leave the room. Students, please pay attention to somebody who is speaking from here. Don't start. Can you not be concentrating for about 10-15 minutes or for some time when a speaker is speaking? Do not start cracking jokes in between. It is insulting to the person who is talking, and it becomes so visible and obvious. Not that we can't see. You can see. After all, I am making eye contact with all of you while speaking. So please learn to pay attention. Okay? Okay. So now I was talking about nanotechnology in medicine. The effect of nanoparticles are today being used to carry the medicine to the point where it is required. Even the lymph nodes to for which reaching of the medicines is very difficult. The nanoparticles are being used to carry the medicine to the correct portion where it is required. And it is the research is still on, it's at nascent stages. But it has been told that it is making substantial progress. Now, what is happening is that the nanoparticles, once they enter the body, the drug delivery has been done, but the question is how the nanoparticles will get extracted from the body. Because they are so small, they stay inside. We are not very sure how whether the body will be able to take them out or they become part of the body and keep circulating inside only. That is one aspect on which very little research has been done. And that is one aspect which needs to be really thought over in times to come. How exactly the nanoparticles are affecting and what is their adverse effect on the body is not known. Some of these cosmetics which are being used, some of the creams which are being used, they contain nanoparticles. And through the skin, they can enter the body. And it is so far believed that they are doing good to the body. They are tightening the skin. They are making the skin uh, more uh, 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 more soft the way it is required. But the ill effects of this are not known. Any technology which comes up, the ill effects get known after some time. But all researchers should start working on those also. Like when they initially, I remember in the early uh, 60s, the plastics came. And at that time, I remember everybody used to say the plastic bags, plastic in even in clothing, the plastics even in shoes, everywhere so strong. It has got long life. But later on, you started finding this plastics now have now become a headache. Now we are trying to make universities, campuses, cities plastic free because the plastics don't get destroyed. And even after they have worn out, lived their life, they continue to stay. We are not sure similar things about the nanotechnology. Nanotechnology is also finding its way into the construction, into the cleaning of the water. Like hyacinth, if you would have heard this, hyacinth is a growth which takes place in stagnant water or sometimes even in flowing water in rivers. And that plants which grow inside, they are of no value, they are of no use, they cannot be eaten, even the animals don't eat them, but the plants are there inside and they obstruct the flow of water. They obstruct the living beings. And those living beings, fish, etc., who eat them, they die because sometimes the hyacinth is poisonous. So, to how to do away with that? What to do? I remember in 2002 when I was posted in army in Jalandhar. So there's a river called Satluj River, which is flowing in Punjab. So in Satluj River, we were so much bothered about this hyacinth that we had to deploy the army units and manpower to go inside the water and clean and cut this hyacinth because the flow of the water is being obstructed, and it is resulting in flooding of the areas because the flow has been obstructed by this. So much growth. And even in ponds and lakes, the cities today which are dependent on water, like we are dependent on water from Tigra Dam, from where we are getting water. Similarly, Bhopal, they have got a lakes in between which are providing water. But the growth of these things in plants, hyacinth, and some other plants which are not desirable in water is a great challenge. Studies are being done, and I have seen some research projects being done here, also some PhDs being done, how to use nano technology or nanoparticles to be pushed in water and to clean the water. So nanotechnology is something which is of a great importance and great use in day-to-day life, and it can give a fillip to the quality of life of all living beings, animals. It can also transform the uh, plants to produce hybrid materials from the plants to increase the growth of food. Also, nanotechnology is being used to have fertilizers which are more potent in giving uh, the boost to the plants for increase of food production. Even that is on. All these technologies, positive aspects are there, but when we study my recommendation to all students who will do research and go into higher studies is whenever a technology comes, please try and study the negative effects from the very beginning, because if you later on it becomes too late. Once the damage is done already, it becomes too late. So that is one aspect. Today we got excellent speakers who are lined up, who will be talking to you from their expertise, from their practical knowledge, and who have spent their lifetime in doing research on these subjects. So please pay attention, listen to them. You will gain some knowledge. And I'm very happy that this conference, which has been organized on such a topic, which is of great relevance to all of us, and which is important in our lives because we all are getting affected by all this. Whatever you may say, nanoparticles are going inside us and they are there to stay. This technology is such that it's not going to stop. And it has got positive effects. So far, about 80% of the effects are positive. But the other black side or the gray areas which we do not know. That is one aspect on which more research needs to be done. So thank you very much. Jai Hind.
Thank you so much, sir. Your thoughtful, your thought-provoking address set a perfect platform for our speakers to deliver their presentation in their areas. I feel immense pleasure to introduce our chief guest, Professor Dr. P.S. Bisain. Professor Bisain received doctorate in 1972 and DSc in 1981 from Jabalpur University, Jabalpur, India. Professor Bisain guided 65 doctoral theses having 350 research publications and reviews to his credit in indexed international journal of repute, 10 patents published in India, US, Europe, and Japan, 15 international books in biological sciences. Professor Bisain served as director of the Institute of Microbiology and Biotechnology, Barkatullah University, Bhopal, Vice Chancellor, Jivaji University, Gwalior, Director, Madhav Institute of Technology and Science, Gwalior, as well as Director and Dean, J.C. Bose Institute of Life Sciences, Bundelkhand University, Jhansi, and Principal Director and Advisor, Jaipur National University, Jaipur, Rajasthan. Sir was a fellow of the UNESCO, UNDP, ICRO, and Hungarian Academy of Sciences at the Institute of Plant Physiology, Biological Research Center, Hungary, US. National Science Foundation Fellow in the Department of Biological Sciences at the University of Illinois at Chicago. European Commission's Marie Curie Fellow at the Institute of Environmental and Biological Sciences, University of Lancaster, UK, and Dard Fellow at the University of Bonn, Germany, awardees of First Indira Gandhi Fellowship for his outstanding work on environment conservation and management. National Overseas Associateship of the Department of Biotechnology, Ministry of Science and Technology, Government of India, UGC Career Award, State Level Kailash Nath Karju Award of Government of Madhya Pradesh for contributions in life science. He was awarded the Lifetime Professorship, the highest honor of Bundelkhand University, Jhansi. He has also worked as Emeritus Scientist, CSIR at Defense Research Development Establishment, DRDO, Gwalior. Ministry of Defense, Government of India, Gwalior. Professor Bisain is the elected fellow of the National Academy of Sciences India for his enormous contributions to the field of biology. The diagnostic technology developed by Professor Bisain is unique in several respects and has received recognition worldwide. The technology has been patented in India, United States of America, Europe, and Japan. He has served as the external scientific advisor to a Barcelona based Spanish multinational company. Currently, Dr. Bisain is Professor Emeritus at School of Studies in Biotechnology, Jivaji University, Gwalior. Sir, please.
नहीं ठीक है. Good morning, everyone. General Sharma, the Pro-Chancellor of the MIT University, Professor M.P. Kaushik, Dr. Alam, Dr. Meena, Dr. Dikshit, learned faculty from MIT University, Gwalior, scholars, and my loving students. It's always a privilege and honor to address the students of biotechnology and to let them know that how the biotechnology is advancing day by day. But before I say about the development, let me tell you how the biotechnology teaching has initiated in the country. Fortunately, I was a part of that as a member or as a chairman of the committee which was constituted by Government of India way back in 1992. In 1992, the life sciences subjects were only zoology and botany. Let me tell you that I did my MSc in 1969 in botany, in plant sciences. When I obtained my MSc in botany, there were no subject like microbiology, biochemistry, and biotechnology even did not initiate at all. So I was trained in the classical subject like botany, where you were taught about the classification and ecology and physiology and some portion of genetics. So that was the total life science syllabus, which was based even in BSc bachelor's program or in MSc program. In 1980 or maybe 1979, the first microbiology department was initiated at MS University, Baroda, and Professor V.V. Modi was the professor of microbiology there. Then the people realized that if the bioscience students, they have to get job, then we have to start some vocational programs in life sciences. Because for the students who are coming out with the life science streams, zoology and botany, the only way to get a job was for the medical college. The number of medical colleges were limited, seats were limited, and the competition was very tough to get admission for MBBS. Then Government of India realized that these students, they should be trained in a way that they could get a job at BSc level. And Dr. M.D. Tiwari, he was the Joint Secretary in University Grants Commission, UGC, and I was appointed as a chairman to constitute syllabus at undergraduate program for vocational programs in life sciences. We visited all national labs, almost all, and most of the labs were situated at Lucknow, met the directors of different national labs, and identified the subjects like seed technology, biotechnology, industrial microbiology, so on and so forth. And Ministry of Education, Government of India allocated some funds, and the colleges were asked that what type of program they want to run, and then every college was given some grants, and that is how the vocational programs started at undergraduate level. So that is how the biotech in a different way has started, initiated at undergraduate level. Realizing the importance of biotechnology, it was emerging globally. In 1992, UGC appointed a committee under the chairmanship of Professor Ashish Datta, who was the Vice Chancellor, JNU, New Delhi. I was also a member of that committee. Professor Padmanabhan from IISc Bangalore, Balram from IISc Bangalore. We all formulated that what programs we should start with MSc Biotech programs. The first biotech teaching at PG level was started at JNU. And Dr. Das Gupta, he was given the charge to start MSc Biotech. The admission was very tough. It was a national competition to get seats in MSc Biotech program at JNU. Now looking to the interest of the students for going to biotech program, then 5 different centers were identified in the country like Pune, Chandigarh, and some I don't remember the name. 5 centers were identified: JNU, Pune, and BHU, and 2 more centers. So that is how the MSc program in biotech started, and every center were allocated 15 or 20 seats. But the number of students who were applying for MSc Biotech program was enormous. Maybe out of 10,000 students, only 1 student were getting admission. It was more tougher than the PET, PMT, or NET examination. Then after realizing that, then under my chairmanship, another committee was constituted to identify the universities to start MSc Biotech program. That is how the different university, almost in every state, 2 or 3 centers were identified to start biotech, MSc Biotech. But of course, to start MSc Biotech program was only limited to the university teaching department. We did not identify any of the colleges to start MSc Biotech program. The idea was that the biotech teaching will be associated with research. If you don't have the research programs, or if you don't have the research lab, the biotech teaching will not be very effective. And the UGC allocated 50 lakhs rupees that time in 1994-95, 50 lakhs rupees with 3 faculty, 1 professor, 2 lecturer, or 1 professor, 2 reader. So that was the strength of the teaching faculty, which was sanctioned by UGC for all the departments. And that is how they again, the Jivaji University started biotech program. Then I was there at Bhopal. We started at Bhopal. And what one at Devi Ahilya Vishwavidyalaya Indore that was started. So that is how the biotech program that was initiated at different places. So that was in 1995 or 1996. Today we are in 2024. Biotech has changed. Syllabus has to be modified, but still we are running almost the same type of syllabus with very minor modifications. Now the technology has changed, and because of the importance of the subject and because of the interest of the students, various colleges, I mean, the mushrooming of the program has started. Substandard institutions have started biotechnology, and because of that, what happened? The students are unemployed. The biotech industries, when they conduct interviews, they say that the students, they even do not know how to isolate a bacteria, how to isolate a fungi. So these are the basic experiments which one must know when one goes for biotech programs, for practicals or for theoretical portion. Now, when I look back, while I was a research scholar, as a botany student, I was working in mycology. And we used to work first for the campus, within the campus. We used to isolate the microorganism from the soil. Soil is the biggest factory of microorganisms. You can get different types of bacteria, different types of fungi performing different types of activities. And that time when we used to isolate any fungal member from soil, water, or air, it was difficult to identify. And the instrumentation was very poor. At the most, we were getting only a microscope. We prepare a slide, and there was a book. Still, I remember of Lillian Burnett, the illustrated fungal genera, something like that. And with that, we used to identify the different type of fungi, and then those isolated strains we used to send to CMIQ, Commonwealth Mycological Institute at Kew in England. And there they identify your organism and they send back to you. And we used to publish our papers like, if you see the old papers, you will find the fungal flora of Sagar University, the fungal flora of BHU. Now you have a campus, MAT, you can easily do. Many a time, some of the teachers, some of the students, they come to me, sir, get me some internship in some industry. Instead of going to industry, you can identify your own fungal flora. You can prepare the fungal flora, identify them, and now it has become more easier with different apps. Now, if you isolate the organism, see under the microscope, use the app, then what will happen? You will start to know how to use the app, how to identify the organism. Instead of going for that, you would be able to know about the various technologies which are emerging, and these emerging technologies are being applied in different fields. Now, whatever you are learning, whatever you are studying the old syllabus, the classical way. Now, everywhere there is a role of artificial intelligence and machine learning. Now, you can identify any type of bacteria or organism with the help of different apps. And for using and for preparing your own app, you have to know the technology like Internet of Things, like blockchain technology, like cloud computing, like big data. So unless or until you know all about these, you will have a paper degree in the form of a paper, but will not know about much about the biotechnology. Now, if you read the latest publications in the area of biotechnology, whether it is medical biotechnology, agricultural biotechnology, or chemical biology, you have to know about the different aspects of machine learning. And whenever you go for the machine learning or whenever you go for the app, you have to know about the languages. Languages means Python. Python you must know before you go for the different technologies of machine learning or artificial intelligence. Now, artificial intelligence and machine learning, they are the future technology. They are going to come in every field virtually, including medical sciences, agriculture sciences. So you are all young, and you must know about the technology, how to use it. Now, if you think that after doing BSc or MSc or BTech or MTech, Biotech, without knowing all these about the technological development, your degree will be useless. If you know about that, even if you are not having any degree, but if you are trained in different type of new emerging technologies, probably you will be more valuable for the industries for any teaching program than with the degree, because you must know about these developments which is emerging, which is coming up. And I will suggest for that in most of our universities, if you go for the teaching faculty, by and large, our faculties are not trained with the modern development in technologies like we. I only read with the paper, but when I go in detail, I do not understand ABC about the technology, but I know about the application. Your role will be how to prepare the application. Your role will be how to use these technology in the area of biotechnology for medical biotechnology, for immunology, for agriculture sciences, for identifying the phytopathogens, the fungal or bacteria which are responsible for causing plant diseases. So these are the things which you must know. Your role, bookish knowledge, you will get all bookish knowledge in book or in Google. But you have to know that how to prepare the app. You identify the thing that I want to work in food technology. In food technology, I want to work in functional food. I want to study about the different functions of the different constituents of food. How do they adversely affect our body? How do they expedite our digestion? So that all you can do by reading research paper, by reading books and applying with the applied aspects of the technology. Now you can ask me, sir, how to know about the technology. Then I will suggest you that you do your degree program here, but with that, you do some value addition in your program. How will you do the value addition? You enroll yourself in the appropriate program of technological development from Cambridge University, Oxford University online program, Boston University, and anywhere. In the evening, you can sit on your computer online. You can learn about the Python language. You will know that these are the languages which are essential to know before I go for the development of app, before I go for the cancer drugs. Do you know that you are all a student of biotechnology? You must be knowing that for releasing each drug, it takes nearly 2 to 3 years when it is released for the public use. Now, for 2 to 3 years, that means a chemical is identified. After identifying a chemical, then various trials of different age, of different geographical zone. So all this, it takes nearly 2.5 to 3 years. Now, an Indian scientist, Indian doctor, he has developed a cancer drug against liver cancer, and which was completed within 20 days or 30 days. In 30 days, he has released a drug that this is the chemical compound and that act against these and these sites. When he identified the sites, he prepared a model. After preparing a model, then theoretically, and with the use of the technology, he developed a chemical compound. And after developing the chemical compound, whatever data he was expecting, he got the same data when he performed the experiment. And that chemical compound has been released lately. That paper you can find, you just go to the internet and say the cancer drug against liver cancer identified in 20 days or 30 days, you will get the details about that. That has been released. And Dr. Singh is a doctor, MBBS doctor. He is MS MD, but he is an expert in developing the app for medical sciences using AI and machine learning. So there, you need not to go anywhere for your job. If you start developing your own app and sell your own app in the area of biotechnology, whether it is crop protection or on your meteorological problems about the atmospheric sciences, you can develop these things, and that is the future. Not that you are running here and there for a small job of medical representative and all that. So I will expect from you that go for value addition and abreast yourself with the latest technology which is emerging, embed with the latest technology with your teaching program, and that will be much more valuable. And with these words, I wish you all the best. Update yourself with the technology, and you have a bright future. Thank you very much.
Thank you very much, sir, for sharing your experiences and knowledge with us. My gratitude to you for all you have done in your professional journey. I sincerely appreciate to Professor Vishen. Thank you so much.
May I now request Pro Vice Chancellor Research Professor Dr. M.P. Kaushik, AIB, to kindly welcome our keynote speaker, Dr. S.I. Alam, Scientist F, Defense Research Development Establishment, DRDO, Gwalior.
Welcome, sir. Now I proceed forward. I feel immense pleasure to introduce our guest of honor, Dr. Ajay Kumar Meena, Assistant Director and Institute in Charge, Regional Ayurveda Research Institute, Gwalior, Madhya Pradesh. Dr. Veena did his master in 1997 and the PhD degree in chemistry in the year of 2006 from University of Rajasthan, Jaipur. His professional journey started as a technical officer in 1997 at DRDO, Ministry of Defense, Delhi, Research Officer, Institute of Ayurveda, CCRAS, Chennai, 2006, Research Officer in 2008 at National Institute of Ayurvedic Pharmaceutical Research, Patiala, Scientist 2 in the year 2012, National Research Institute of Ayurveda, Siddha, Human Resources Development, Gwalior, Madhya Pradesh. Assistant Director 2014, Ministry of Ayush, New Delhi. And currently, he is a Assistant Director and Institute in charge since 2020. His interest research areas are development of quality, stability parameters for various classicals and investigational new drugs to facilitate their registration and traditional medicine under the THMPD of European Union. Development of safe, effective, affordable Ayurvedic and Siddha drug based on strength of Ayurveda and Siddha, considering national and the global needs on scientific lines at the par globally accepted norms. He has development of quality control parameter and standardization of raw material, extractive development of analytical method for Ayurvedic and Siddha drug quality control by using a modern instrument equipments and updating the present data available. He has also developed the treatment and disposal technique for wastewater. He has worked as a technical manager for NABL ISO IEC drug testing laboratory at CSM RADDI Chennai. He carried out the work on explosive safety and audit keeping as per the current STEC regulation on various safety aspects. He has completed various research projects as a 20 projects as a principal investigator, 5 projects as a co-investigator and 40 projects as a co-coordinator. Sir has published more than 165 papers, 8 books, also conducted various guest lecture and invited talk. Sir has supervised PhD, M Pharma and MSc students. Now, I would like to call Dr. A.K. Meena to deliver the lecture. Sir, please.
Good morning to all of you, respected dignitaries on the dais, faculties of Amity University, research scholars and students. So I congratulate and appreciate the organizers if they select that nice topic for this international seminar because this is the requirement of today's society. Because biotechnology and nanobiotechnology is a major requirement of today's society for us, or nanobiotechnology is the major part in drug discovery, agriculture, and different industries. So already our guest speakers and Sarmaji already informed so many informations about the nanobiotechnology. Or Bisen sir inform ki how that biotechnology start in our country. But I say ki we are syllabus or that things we developing now in 70s or 80s. But actually that nanoparticles is about nanoparticles in we have so many literature in our classical literatures in Ayurveda and Siddha systems because these systems I think more than 5,000 years old. Now we discussing everywhere. If asking any researcher, they talking about nanoparticles. Everybody doing studies on nanoparticles. But these classical text already mentions how we prepare nanoparticles or what is the therapeutic usage of that particles. For example, basmas. Now everywhere Western countries publishing ki all Ayurvedic drugs, Siddha drugs are toxic drugs, but it is not true because we are preparing nanoparticles. That is what is that? That also prepared by herbomineral particles by we using metals there. So our basmas or that Siddha herbomineral formulation that also are the nanoparticles because they give different temperature, they have different procedures for preparations. So now we preparing silver particles, gold particles, nanoparticles, but that time they preparing gold basma, silver basma, copper basma, iron basma, that also in nanoparticles, or they have very nice or very good therapeutic usage in cancer like that disease. Or currently also so many studies going on, or since are informed ki we don't have earlier that much nice or good infrastructure for instrumentation or like that, but we are today we have in very good positions, or we are doing very good studies on these nanoparticles. So now for Ministry of Ayush means that our Gwalior institutes also have 5 or 6 collaborative projects with different CSI laboratories or IITs, or these basmas or nanoparticles. Now so many studies going on in IIT Delhi, IIT Indore, AIMS Bhopal, AIMS Delhi, that renowned institutes are doing now that work. So I request all faculties or students ki this is the vast field of nano biotechnology part is, or if interested persons doing that field work means they have very good chance, very golden chance also, because our Ayush system have so many information, so many things they have, but the problem is we don't have validated data, validated procedures, or nanoparticles or mineral herbomineral formulations are not toxic if we properly follow the procedures how we prepare that procedures, so that formulations don't have toxic effect. But if we don't prepare properly, we don't follow procedure, that time that formulations are toxic. So I want to say ki that nanobiotechnology have very good technology that have very vast area, or that is very useful for our society, or that is the requirement of today's our society or our nation. So interested persons can choose this field and they can do the studies for this purpose, because so many nanoparticles we are preparing, but simply lab scale we are preparing, we doing study, but after that we don't complete or we don't reach till targets. So that is the good, or that is if we prepare planting, we prepare nanoparticles, we do all analytical methods or all technologies. After that we reach for preclinical studies, after that we reach means or therapeutic usage or clinical studies means we get successful results for that purpose. Or nowadays so many formulations that nanoparticle formulation that are used is cancer or some different disease also. So that is the important role for nanoparticles. Everybody interested or they're doing studies nowadays for nanoparticles, that is good, but we reach our target, that is the requirement for us. So I wish again all the organizers, or I say all the participants ki now so many experience expertise they are sharing their knowledge with you, so you get something there, you learn something through that, or that feel use or implement that what you people's learning that things. So I say thanks to all of you and wish of you all the successful seminar.
Thank you very much, sir, for very informative talk with us about the Ayurveda and Siddha. Really, nanobiotechnology is a boom for the biotechnology world. Now, I would like to request all the dignitaries to kindly release the abstract book. Also, I would like to call Professor Dr. Vikas Srivastava and Raghvin Kumar Mishra to join the days to release the souvenir.
Thank you very much to all. Now, I would like to introduce our first keynote speaker, Dr. Mukesh Dubey, Associate Professor, Department of Forest, Mycology and Plant Pathology, Swedish University of Agricultural Sciences, Uppsala, Sweden. Dr. Dubey is already connected with us on Zoom. Dr. Dubey earned his PhD in botany in the year of 2009 from Central Institute of Medicinal and Aromatic Plant Sciences, Lucknow University, Lucknow, India. He has published more than 50 research publications in international and national review.
Yes.
Sir has supervised 6 postdoctoral
Hello?
researchers.
Okay. Okay.
Postdoctoral researcher, 7 PhD students, 7 visiting PhD students, and more than 20 masters and bachelor students. Dr. Dubey delivered more than 15 lectures as guest speaker in the national and international conferences. He is also served as senior editor and reviewer for many international and national journals. May I request to sir kindly enlighten us by deliver the lecture.
Hello?
Hello. Hello.
Can you hear me there? Or...
Yes, sir. You are audible.
Okay. And can you see my PowerPoint?
Yes, sir.
Okay. Very good. Okay. Thank you very much. I would like to thank MIT University and organizers for inviting me and letting me share my ongoing research on RNAi and its technology and how we can use RNAi technology for plant disease management. And I think in my slides, there would be a lot of fluorescence image. It's a GFP and some other imaging. So it would be nice if we have a little bit lower light on the stage so the audience can easily see the image.
Sir, please full screen your presentation.
I think it's on the full screen, but maybe I have to switch the mode probably. Let me see.
By switching presentation mode, sir, you can present your PPT on full screen. Press F5.
I think let me try one more time. Hope it works now.
Yes, sir.
Yes,
Answer.
excellent.
Yes, sir, please.
Okay, yeah, thank you. Thank you very much. It's always some problem with technology. And when it works, then it's excellent. Okay, then I will share my yes. So this is a title of my talk, RNAi technology for plant disease management. I think we have been listening some very good ideas and very good knowledge about our other panelists. So it was very thought-provoking. But I will take you to more research that I'm currently doing in RNAi technology. And I will start by providing you a brief background. What is RNAi technology and how it actually works? So probably we all know that we have central dogma. And then meaning that all the genetic information is stored in DNA. And this genetic information, flow from DNA to RNA and to protein by the process called transcription and translation. However, for proper functioning of the cell, this flow of information is tightly regulated. And this flow of information is regulated at both at transcription level and also at translation level by a different way. And one such way that this information is regulated by a small RNA, a small molecule that is around 20 to 40 nucleotide in length. And these small RNA, they mediate this post-transcriptional gene silencing here after the transcription level. Let me change to the pointer here. Yeah, so for this stage. And this small RNA mediated technology, it's like a very emerging field. Recently, what we saw that vaccine for COVID-19, it was also mRNA-based COVID vaccine. So in our research in agriculture, we are trying to use this technology for plant disease control. But before I show you my result, I would like to explain to you what is actually RNAi and how it works. So if you see this image, it looks quite complicated. Yes, this RNAi is a bit complicated, but you have to, I will try to explain to you how it works. So you can see here, a double-stranded RNA or single-stranded RNA with hair loop structures. They are produced in nucleus from DNA. Once they are produced, they are cleaved by endoribonuclease called a dicer into small pieces, and this small piece is called a small RNA. Depending on their origin, these small RNA can be classified into different groups like microRNA or siRNA, but I will not go into that. I will use a general term, small RNA, for both category of the RNA. Once these small RNA are produced, they are unwinded into two separate strands. That is called the guide strand and passenger strand. The passenger strands go for degradation. However, the guide strand is loaded into a complex that is called RNA-induced silencing complex. In this complex, we have another protein, an enzyme that is called AGO. This is argonaute protein. So this a piece of RNA guide strand guide this argonaute protein to the gene target. This is based on sequence complementarity. And then once they found the gene target, the argonaute protein cleaves the gene into small pieces and thereby, inhibiting the protein synthesis. As we all know that protein is the functional and structural unit of a cell. So once a particular protein is not produced, meaning that its function is inhibited. So this whole process basically is called RNAi silencing, meaning that this RNAi that is mediated by small RNA can silence the function of a particular protein. So I will talk more about this and how we can use this knowledge to apply in agriculture. So, but I will request you to remember this thing, that we have a small RNA and these small RNA, they need a gene target for their proper functioning or for gene silencing. And in last decade, we know quite a lot. Before that, we know that small RNA mediated RNAi is a conserved process within eukaryotes and also in prokaryotes. But recently, what we know that this small RNA, they not only regulate their gene expression at endogenous level, but also they can move from one species to another species, basically during the interaction. So what you can see in this image, small RNA can move from human to its parasite, and similarly between human and viruses, and between human and plant, and between human and fungi, and so on. So meaning that these small RNA, they moved from producing cell to recipient cell, and they also do RNA silencing in recipient cell. Meaning that there is a cross kingdom or cross species RNAi. So this is a groundbreaking discovery that small RNA, they can move between interacting organisms. Another groundbreaking discovery was that even you spray this small RNA, you can synthesize them in the laboratory because they are very small. And if you spray them on any fungal hyphae, what was found that these fungal hyphae, they can absorb the externally applied small RNA. So what you see in this case, this in the green, this is a small RNA tagged with GFP. GFP means green fluorescent protein. So the protein that is tagged with GFP, they will emit green light that you can see here. So this is different plant pathogenic fungi, and they all, if you spray them with a small RNA, the small RNA will go inside here. So this was even more important that there is a possibility that this fungal pathogens or any kind of pathogens, if they are pathogenic to human or pathogenic to plant, they can absorb externally applied small RNA. However, you can also see in this image, the uptake efficiency is not it's different in different species. So for example, Botrytis cinerea, it's a very common plant pathogen that can infect more than 200 crop species. It can absorb very high amount of small RNA, but if you see these ones, for example, verticillium and rhizoctonia and other pathogens, they have a low ability to absorb. And this might affect the application of RNAi. So it's very important to know which pathogen can absorb externally applied small RNA. And what you can see in this image, even certain pathogen, for example, Choledotrichum, they cannot absorb externally applied small RNA. So here you see they are accumulated outside of the cell, but they are not going inside the cell. So this is very important information to know before you apply any kind of RNAi technology or small RNA for plant or human disease control. But I ask a question, you think about this, why do we need RNAi-based technology? Why we want to use this? And it is mentioned very beginning by the first speaker that we need to know before we use any kind of technology. We need why, and we need to know how it works, and we need to know its side effect and so on. So we must ask ourselves a question, why do we need this kind of technology? So I think I'm in agreement that we have to adapt to emerging technology, but we need to know their pros and cons. So I will give you one example that why we should use RNAi-based technology in agriculture. We all know that human population is growing, and by the end of this century, we will be reaching 10 billion people. And the meaning that we need much more food than what we produce today. And we also know that we cannot expand our land under cultivations. They have already reached maximum. And then we can already have a lot of cropping intensity. So there is a very limited chance that we can increase our food production using these two methods. However, the one method that is, that has higher chance to supply the food demand, that we need to increase our yield. And this can be done either by developing new crop variety with higher yield, but this can also be done by just reducing the losses to pest and diseases. So we know that if we don't apply pesticide or fungicide, then our crop loss can go up to 40-50%, even in some cases 70 to 80%. So we need to control our crop loss. If we do that, then we can already increase the yield, and then that can be very good to feed the human. So currently, how this is done? So most of the diseases right now, they are controlled by the application of chemical pesticides. And we all know that chemical pesticides, they are not environmental friendly. They can cause many problems in any ecosystem. And and even recently, in last decade, there is a growing demand among consumers that they want a food that is free from the pesticide, meaning that it should be either grown organically or it should be grown in the situation where no pesticide is used, meaning that we need to find one alternative of chemical pesticide. However, the biggest problem is this. So what you see in this is one example that wheat. It's a most widely grown crop all over the world, and it is attacked by many diseases. And then to control these diseases, farmers use a lot of pesticides of different kind. But this is a paper published in Denmark. They show that over the many years, the efficiency of pesticide is reducing. So in the beginning, if you use pesticide, you had 80 to 100% control, but now the efficiency of this pesticide has reduced up to 20 to 40%, meaning that farmers has to use more and more pesticide for their yield. And that's as I already told you, this is toxic to the environment, to the human, and so on. Another problem with this one, why this is like this, because most of these pathogens, they are developing resistance to the fungicide. It is very similar. We all know probably that human, if you have an infection with any kind of bacteria, most commonly like tuberculosis, then many times many antibiotic will not work because bacteria has developed multi-drug resistance. Same applies to the pathogen to the crop plant. So this raised serious concern, and this demand for an alternative strategy to control plant diseases. So now I come to the question, maybe I can also explain that this is even more relevant because due to the climate change. And what you can see in this slide, that this is a recent discovery of that due to climate change, there is a new fungal diseases are emerging. So these are new diseases that has never been discovered before. And we don't know how they are going to behave. They can be very problematic. They can they can destroy whole crop. And this problem is not limited to a particular continent or in a particular country. It's a problem to all over the world. So this again, asking for an alternative strategy. Can we go beyond to pesticide application? And in my opinion, yes, we can do that, but we need to do a thorough research. Then come to the another question, can RNAi technology be used as an alternative to chemical pesticide for plant disease control? If you say yes, then another question, how we can do this? And in my opinion, the best way we can do this by generating more knowledge on RNAi technology and its application, and of course its side effect. So now I can give you some examples of our work that we are doing here in Uppsala, Sweden, where we are trying to understand how RNAi technology can be used. And then we have performed a series of experiments. I can briefly take to some of the result. So as I told you before that, if you want to RNAi to use, then we need to identify a suitable target in pathogen. And in this case, this target can be a gene that is essential for a pathogen, for its pathogenicity or its virulence. So once we identify that, then we can design a small RNA that can bind to this target, and then that can silence the gene. So this part is relatively easy, but however, the complex part of this is how to apply this small RNA to the pathogen. What is the delivery method? And before that, some speakers, they explained nanotechnology. That could be one option in this case. So what are options we have? We have basically two options to deliver the small RNA to the pathogen. One option is called host-induced gene silencing. So in this option, we have to generate a transgenic plant that can produce this small RNA that can target the essential gene in the pathogen. And then you have to, if you apply this transgenic plant, and then probably that would be resistant to many pathogens. And for example, I will come to the examples later on, but another option could be we synthesize small RNA in the laboratory and high throughput laboratory, and then we spray this small RNA to the crop, And then, as I told you, these pathogens, they will absorb this externally applied small RNA to their cell, and then they will silence the essential gene in pathogen that can cause the disease. So we have basically right now two options. That is one, host-induced gene silencing, and another option is spray-induced gene silencing. I will briefly tell you pros and cons of these two methods. But I can start with first with Higgs, like host-induced gene silencing. And for this, people used a model plant called Arabidopsis because this is easy to work with. So what they have done in this case, they generated a transgenic Arabidopsis, then can overexpress a double-stranded RNA that can target DCL gene. It's a dicer gene that is important for botrytis to germinate and infect a plant. And then you infect Arabidopsis with botrytis and Verticillium dahlia. This is another pathogen. And then you can see a plant is sick, basically very near to die. But then in the mutant line that produces small RNA that can, target virulence gene in botrytis and Verticillium dahlia, then you see the plant is very healthy. And you can measure the biomass of the plant. You can take some leaf and root and then do some laboratory analysis, and then you can measure the biomass, so it's significantly low in the transgenic plant that produces dicer small RNA. And similar in another example, this is a fusarium head blight. This is also a very problematic disease. It can destroy the wheat, not only reduce the yield, but also this fusarium produces mycotoxin. If you eat food contaminated with mycotoxin, it can have a serious effect on human and animal health. So in this case, they identify a gene called chitin synthase in fusarium. And then we know that this is very important for fusarium pathogenicity. And then again, you can create a mutant line in wheat. And then if you see here, this is a Sumai 3 line that is resistant to fusarium. Then you inoculate fusarium here. There is no disease. But in the susceptible line, there is a lot of disease. But then this L1 and L3, this is a transgenic wheat that is producing small RNA that can target chitin synthase gene in fusarium. Then you have very low disease. So meaning that host-induced gene silencing is very successful and it works perfectly. However, the main problem with this is that you need to generate transgenic crop. And many times it's not possible to generate transgenic for all the crops, maybe only limited crops. But another problem with this method is that transgenic crop and food is not accepted, especially in European and in Europe. There is no way that it's not allowed to grow any kind of transgenic. Maybe in the future, but right now this is the problem with this technology. So now I'm coming to another method where we can use SIX, like spray-induced gene silencing. So in this method, we don't have to create transgenic crop. But what we do, we try to create a small RNA in the laboratory, and then we spray on the plant. So for this work, we used a model, a pathosystem, potato, and late blight pathogen Phytophthora infestans. I think we grow a lot of potato in India. Probably we also know that, like in this year, if you have a very humid condition during the winter and rain, then you have a lot of Phytophthora potato late blight. And within a week, it can destroy whole crop. And then to control this disease, farmers has to apply here, for example, in Europe, they has to apply 10 to 15 times pesticide to control this. So this is a lot of pesticides that they need to apply. So we use this model to test spray-induced gene silencing. As I told you that if you want to do spraying of small RNA, it's super important to know that if pathogen can uptake the small RNA that you spray. So what we did in this case, we generated a pathogen, Phytophthora infestans, tagged with GFP. So again, it gives green color under the microscope. So this is a sporangia of the pathogen. And then if you spray, this produce GFP in the green. So what we did, we generated a small RNA, double-stranded RNA in the laboratory and tagged with Psi3. It's a kind of a dye that gives a red color under the microscope. So what we did, if we spray the Psi3 on sporangia, you can see it's going inside the side 3 going inside of the sporangia, meaning that Phytophthora infestans sporangia can uptake externally applied a small RNA or double-stranded RNA. So again, what we did, we infected, this is potato leaf under the microscope. We infected the potato leaf with Phytophthora infestans. And you can see this is the GFP tagged Phytophthora growing on the potato leaf. And then if we spray the side 3 tagged double-stranded RNA, so what you see here, it seems potato leaf can also uptake this small RNA. This is stomata guard cell, they are in the red. So this is the zoom image you can see here. So the summary of these two slides is that based on this experiment, we can say that Phytophthora infestans, the lead blight pathogen, and also the potato leaf, they can uptake externally applied small RNA, meaning that we have very high chance that if we use small RNA to control Phytophthora, there is a high chance that it would be succeeded. Again, what we did, then we developed a theory to test this. What we did, we have again here potato leaf. Then we inoculated this potato leaf with Phytophthora infestans tagged with GFP, that is in the green. So under this situation, this was the control experiment. So what we did in order to confirm that small induced gene silencing is working, we created a small RNA in the laboratory, and then we spray the small RNA on the leaf. So meaning that small RNA will be taken up by the Phytophthora, and then small RNA will silence the GFP. So there would be no green color. So that's what you can see here. If you spray the small RNA, then the GFP is degraded. And we can also measure, the intensity and the biomass of the fungus. It was significantly reduced. So meaning that the Phytophthora infestans can uptake externally applied small RNA, and the small RNA that was taken inside the Phytophthora can silence the gene in Phytophthora. So that was really good. Then we tested this knowledge by identifying these 4 genes. And we know from the literature that these four genes, they are very important in Phytophthora to infect the potato. And one of this we used as a positive control because this is also a target for fungicide. So what we did, then we identify the target gene, and then we created the small RNA that can target these genes, and then we perform our experiment. So what you see in this case, this is a control and this is potato leaf. If you infect this with Phytophthora, and then if you stain the leaf with a trypan blue stain, it will, because trypan blue stain only dead necrotic area. So you see this area is necrotic, meaning that this is a disease, and this is also kind of control. So in both control, we have a lot of disease here. But if you spray small RNA that target GPB1 gene, then you have no symptoms at all or very limited symptoms. And then a little bit more symptoms. So meaning that small RNA spraying is working very good in case of potato and leaf blight pathogen Phytophthora infestans. But again, another take-home message from this experiment was that selecting target gene is very important. So here we had very high effect, but here it was low effect. So what kind of gene we select to silence, it's very important. So this was one example where we showed that spraying of small RNA can be used as an alternate to pesticide. But people ask many questions that how long can small RNA be stable when you spray? Because we know that we know that a small RNA can be degraded. So to test this, what we did here, we spray this double-stranded RNA, and this is control on potato, they are died. But if you stay small RNA, and they were quite stable up to many days, and the plant looked very, very healthy. So with our experiment, we confirmed that small RNA can also be stable. Now this was another example where we used Fusarium graminearum, and we saw that it was also working fine with GFP double stranded RNA. I'm getting a phone call from Raghavendra. Should I stop here, or what do you say, Raghavendra? Can I just take this call? Excuse me. Yes. Okay. Yeah, I will do that. Okay, so it seems yeah, there is a lack of time, so I need to summarize it. But I think I can stop here, and in summary, I can say that using RNAi technology based on small RNA is a successful alternative to chemical pesticide. If you have any question, feel free to, I will be happy to answer, and you can also contact me later on. Thank you very much. I hope I make you understand. Was it very complicated? Feel free to ask me if you have any question.
Yeah, if you have any question, you can ask. Everyone, student? Sir, okay. Okay. Thank you so much, sir, for very informative lecture.
Thank you very much, Dr. Dubey, for the excellent address that you gave. Thank you so much.
Thank you, sir.
Now, I would like to call our next keynote speaker, Professor Dr. S.I. Alam. Dr. Alam is working as a scientist at Biotechnology Division of DRDO, Gwalior. He has completed his PhD in microbiology and has gained more than 30 years of research experience. His area of research include bioremediation of organic waste in high altitude region, bacterial systematics, disease proteomics, and detection of biothreats agent using the tandem mass spectrometry. He has described two new species of bacteria from Antarctica. He has more than 80 publications in various international journals. Sir has represented India on various international platforms pertaining to disarmament of scientific collaborations. He is currently engaged in developing bioverification strategies for unambiguous identification and characterization of known, unknown, and advanced pathogen of biowarfare significance. I would like to call Dr. Alam to deliver his lecture. Sir, please.
Thank you so much. A very good morning to all of you. There is a gathering of students I can see here. I'll be very brief on what I have been doing on using this technology, or our lab is doing using this technology.
Rather, I'll be giving an overview as to what is happening in the area of proteomics. So this all started with I think I can remove this. Can I remove this panel? This panel, hide this panel. Oh, that's. Oh, this was better here. Still down. It's hiding the presentation now. Oh. So it all started with the Nobel Prize in Chemistry, which was shared by these three gentlemen, and they developed a method to ionize and bring the large biological molecules in the gaseous phase, so-called soft ionization techniques. So before that, it was not possible to analyze the biomolecules, which are generally large, using the mass spectrometry tools. So what is proteomics? It's actually a large-scale systematic study of protein structure and function, usually with respect to a defined entity. We are looking for a pathway or an organelle or cell, tissue, or the whole organism. So, and the central core of the proteomics is actually mass spectrometry. So the current strength in proteomics is that you can uh, identify and quantify the entire complement of proteins in a cell or tissue. So that would mean that on a single run, you can identify about 10,000 proteins and about 40 to 50,000 peptides. Synonymously sequence the proteins at the level of the amino acids on a high throughput scale. So just think about 20 years back when you're going for Edmund sequencing to generate about 10 to 20 residues of amino acids and then trying to generate probes to fish out the gene of your interest and then study the structure function relationships on a very elaborate level. Now you have the sequences on your desktop from the whole proteome of the cell or the microbe in a matter of a week or in a few days. So proteomics evokes everything post-genomic, and I'll not go into the details. There is not a single area of life sciences where proteomics has not been applied. And there are different areas in which people are working. I'll be largely focusing on the disease proteomics. That is what has been done in the disease proteomics aspects. So there are about 20,000 genes in humans, and there are specific expressions of the subset of genome where that makes the backbone of the phenotype. And then you have higher order of complexities. So you have splicing variants, post-translational modifications, subcellular localizations, and so on and so forth. And then are also time-dependent tissue and organelle-specific proteome networks, and then they respond differently to the perturbations. So they respond to the disease, and you take a cup of tea and your proteome changes. So its proteome is that dynamic. So as against the genome that remains more or less stable for a given organism. Though there are reports of genetic mosaicism that the cells have a different genome even within the same organism. So what is the technology? The basic technology is the mass spectrometry. The mass spectrometer actually consists of an ion source, the mass analyzer, and the detector. The ionization techniques that is currently used for analyzing the large molecules is ESI, the electrospray ionization, and the matrix-assisted laser desorption ionization, that is MALDI. So here, basically, what you're doing is you're pushing, solvated analytes, peptides largely, if you're going for the bottom-up approach. And then there is desolvation, and there are charges that they burst out, and they impart the charges onto the peptide molecules. And these peptides in the gaseous phase are flown into the mass analyzer, where you determine the accurate masses of these peptides. So it's all about determining the accurate masses of the peptides and their fragment times, and at a very high resolution. So that all translates into the protein sequences and the modifications that you are looking for. There are different kinds of mass spectrometry available. Largely, you have two MALDI and ESI ion sources. There are different combinations of these quadrupole, time of flight, orbit trap, and ion trap. You will have one or more coupled in a given instrument. For example, what we are having is a top line, and then we are also trying to get one which is having an orbit trap with a quadrupole combination, and then you also have time of flight combined with different combinations to give you very high throughput, high resolution at the level of more than 2 lakhs these days is very common, and a sub ppm level of mass accuracy is also not very uncommon. So this is a very, highly simplified scheme. How do you identify a protein using mass spectrometer? So basically, what you do is you have a protein where you digest the protein into peptides, and these peptides are flying into your mass analyzer, and you're determining the accurate masses of the peptides. So once you get the accurate masses of the peptides, you select one or two or most abundant of them, and then fragment them into the fragment times, and then deduce the sequence of the peptides. So if you do it on a very simple analyzer, you generally sequence about 20 most abundant peptides. And on a very high throughput machines these days, you're sequencing about 50 to 60,000 peptides in one run. So then you're going for the database searches. There are different approaches, which is database dependent, or maybe database independent, or library dependent. And library, I'll not get into the details of these approaches. You can have a discussion if you're interested in. But this is how the technology runs. So in clinical proteomics, basically you have two approaches: the expression proteomics and the biomarker discovery. So biomarker discovery is one area which is extensively explored by proteome tools. It is translational in nature, and the characterization of the proteome of normal or diseased tissue or biological fluids is what people start with. So in the expression proteomics, you are detailing and quantifying the protein differences. As for example, in the normal patient cohort and the disease patient cohort, what are differences in the protein levels, and the quantitative and qualitative differences you are listing out. And that actually eliminates pathobiology, improves disease qualification. There you can go for new therapeutic targets. And in the biomarker discovery, which I'll be slightly elaborating upon, you identify peptides, proteins, or post-translation modifications, which supports early disease detection, facilitates diagnosis, and information on the disease and guide therapy. You can do a lot of things on proteome expression. You can go for gene annotation. You can go for differential expression between the two cells, two tissues, two developmental stages, or maybe, as I said, the most common in the disease proteomics will be the normal and the disease tissues. And you can also go for absolute quantitation. You can actually, mass spectrometry is typically not a quantitative tool, but recent developments have made it quantitative in nature, where you can even go for label-free quantitation of the proteins and peptides in a given sample. So you can accurately tell how many copy numbers of a given protein is there in a given cell, and correlate that with the transcripts present in that. So you can describe a lot of things on the regulations that are going inside. You can go for temporal dynamics in the developmental biology, the different stages of development, what proteins are being expressed, at what stage, and then try to look for the structure function correlations. Localization is another area that you look for, that where these proteins are actually localized. As I said, post-translation modifications are actually critical for the function of the proteins, and they do have some correlates on the disease. Certain diseases have post-translation modifications on some residues of a given protein that you will be looking for as a marker of the disease. Maybe a set of post-translation modifications or a combination of different peptides that you can look for. AI has actually given a lot of strength on the biomarker discovery on all aspects, starting from the the digestion to the liquid chromatography, ion mobility, MS1, peptide fragmentation, MS2, protein identification and quantitation, and largely on the developing the algorithms to analyze the data. So generating data on MS is not that difficult, but to use the algorithm to make them meaningful is all that important. And he has recently played a lot of role in this regard. So you have different approaches. As I said, you can have top-down approach where you start with the intact protein and then fragment it down, or you can have a bottom-up approach where you digest the protein using it protease, generally trypsin, and then use those peptides to identify the proteins and see what are the differences and what are the sequences present over there. So you can have a discovery or a shotgun approach, which is the data dependent acquisition mode, or a SWAT MS approach. I'll just give briefly, I'll not get into the details, what is DD and DID and all that. I'll just give you a feel or a flavor that how the proteomic tools goes. And then you have a targeted method where you can actually go in the data independent acquisition mode and go for either SRM, MRM, or the SWAT MS. So when you go for the discovery or shotgun mode, what you're actually doing is you're flying all the peptides into the mass analyzer and generating the first MS scan, which is known as the MS1. So this MS1 gives you, these are the peptides and their masses present over there in your given sample. And from there, you select the most abundant, maybe generally 20, 50, or maybe many as you can choose these days on the spectrometers. And then this abundance of precursor determines the MS2, where you go for the sequential fragmentation or maybe fragmentation together these days to determine the sequences of these peptides. Then you can also have the targeted approach where you have a very predefined peptide that you know that this is my marker peptide, or maybe a set of peptides which you think is my set of marker peptides, and develop your assay where this peptide and certain daughter ions after their fragmentation become transitions. They form the transition. So this peptide mass and their daughter ions, say 3, 4, 5, are making a transition that is unique to this peptide. So you can straight away take the patient sample, go for screening these peptides present over there along with the daughter ions. The transition is scored yes and no, and you tell that this is the marker present over here. This gives a lot of advantage over the antibody-based approaches, because you can have antibodies, say, for 2, 3, 4, 5 agents. Here you can have the assay for 500 peptides or 1,000 peptides in one go. So the current is the SWAT MS, which is the data and dependent acquisition. This combines discovery and the targeted approaches, where you have the 25, almost 25, or these days even lower, 25,000, 25 Dalton window, where you're pushing all the peptides, which is coming after LC separation, sequentially in nanoseconds and few milliseconds. These from 400 to 425, 425 to 450, these many masses will go in bunch, and they are fragmented in the collision-induced dissociation, and they are scoring the fragment ions. And the algorithms are able to tell you which daughter ion is coming from which precursor, and they give you the sequences of these peptides. So you are, in a sense, sequencing the complete proteome of a given cell or a tissue type in this SWAT data independent acquisition. So you can also use this technology for the targeted processing, where this is now a digital picture that is generated on this SWOT, where you have the fragmentation of precursors, ions, and the daughter ions for all the peptides that you have scored. And the several fragment ion chromograms for each of the peptides of interest are extracted from this digital map. This is just one example where they have covered the complete yeast proteome by SWAT-NS and gone up to 40 copy per cell of the the abundance, the sensitivity that even they have sequenced proteins which were as low as 40 copies per cell in the given sample. So in the biomarker discovery or the clinical research, it is possible to assess the complexity of human proteome and uncover disease-related subproteome. So what you are looking for is making disease correlates, and you are having enough replicates and cohorts in the control or the healthy and the disease state. And then you're trying to go for the early and accurate diagnosis. And obviously, the protein becomes a better marker as compared to the others like RNA or DNA because of the simple fact that they are dynamic. They are much more dynamic and they can give you more information about the disease state and the disease progression. So there is a need for reference maps for generating your assays on the marker discovery or maybe many more areas. I'll specifically talk about the biomarker discovery where the reference maps are generated. They are collection of the fragment time spectra of peptides for the predicted proteins and sequences of the genome. So this is essential prior information for the targeted or SWOT-IMS type of the measurements. So what you are generating is the all essential coordinates of the informative peptides. You have already defined the informative peptides in your discovery mode, and this information will range from mass, charge state distribution, retention time, and M by Z of the precursor ions, charge state distribution, relative intensity of the fragment ions, and this all is now in the digital form. Now you need not push your sample again and again iteratively to look for new information. Once you have this digital format, this can be exchanged between the laboratories and you can extract data of your interest as you wish, retrospectively. This reference map is generated in two approaches via deep shotgun sequencing of the natural proteins in a suitable sample or via the sequencing libraries of the synthetic peptides. So in the first approach, you are generating the suitable relevant spectral libraries to serve as a map. So as I said, that you have sequenced every peptide and that's your reference map in the reference state. And the second approach, you are defining that these are my peptides of interest, and then you are synthesizing those peptides and generating the reference maps. So these reference spectra generated from fragment time of chemically synthesized peptide library is actually in progress for several tissue types and several disease states. So what you are doing here is you are ideally defining the prototypic peptides. So these prototypic peptides means these peptides are present only that protein of that given organism. So you can define that if this peptide is there means this protein is present over. So this you define a set of prototypic peptides, and then you have your marker studies that can be further validated. As an example, for Saccharomyces, more than 97% of these synthetic peptides have been generated. And for Mycobacterium, 13,000 unique synthetic peptides have been generated and the reference spectra have been made. So this is the scale at which they are working now for generating the reference maps. And once this reference map is made, it is useful for all, and they can be exchanged across. So this in the spectral library, you have different, different websites where they provide different. There are different tools where you get these libraries and informations that you are looking for. peptide atlas, SEM atlas, PASL, human proteome media, and that can be shared electronically. So, and when you talk about the MS reference map for clinical research, you have the human blood plasma proteome that has been completed now almost. It includes 1,900 non-redundant protein sequences. So this all has been sequenced. And if you're looking for some changes in these plasma proteins, your reference map is already there to quantify your study. And then you have the cancer proteome for different types. You have human cancer cell lines, which have been mapped, for which the complete data is available. There are more than 7,500 proteins from colorectal cancer tissue, of which about 1,800 proteins with high significance differences, that they are different from the healthy tissues. There are the cumulative coverage of human proteome, which was 69 when I extracted the data. It has gone much more. It has reached more than 90% these days. There are US FDA approved protein markers, about 1,172 proteins, which have been selected as cancer associated proteins, for which the SRMs and MRMs have been developed. For many of them, the SRMs and MRMs have been developed where you can quantify these peptides and proteins which are already known to be cancer associated. MS assays for synthetic peptides are for complete human proteome is near completion. And there is another area which is coming up these days, this is single cell proteomics. There was earlier we were using the whole tissue for the proteome analysis, or maybe multiple, the bunch of the cells from the cell line. Now you can have single cell studies. We can generally the laser microdissection is used to separate one cell, and then followed by the insol digestion, or maybe digestion, tryptic digestion, and then you have the sequential MS analysis where you can subsequently correlate by these special terms that this cell has come from this tissue and this place that I'm looking for. So this is the state of art, which has got potential applications from basic biological questions to precision medicine. And this is just to give a glance, nothing to do with the disease proteomics, but this is the area which is coming up where you actually have multiple questions that you can address. Metaproteomics in large scale identification and quantification of proteins from microbial communities, and they provide direct insight into the phenotypes of the microorganisms into the molecular level. So when we talk about One Health, this metaproteomics is actually coming up for the environmental characterization and deep sequencing of different environmental samples, what is present over there and what is getting changed over time or in the climatic changes. This is just to summarize these metaproteomics. There are different questions: who is there and in what abundance? What is expressed and what is the metabolic pathways? How do community members interact and how the different metabolites are being used? What are the carbon sources and assimilation pathways, and so on and so forth? You can have multiple queries on that. And the current mass spectrometers are really handling these issues where you can have the complex mixture in your hand and then bin every peptide to the protein and protein to the taxon that this has come from this organism, and this organism is present in this much of abundance. Just to briefly rush through what we have been doing using the proteomic tools, we have used the proteomics for the post-exposure recovery and analysis of biological agents in the simulated biothreat scenario, where we think that if there is deliberate release in the form of aerosol, are we able to detect that and characterize that what is the agent present over there from the foliage or from the surfaces and so on and so forth. So we have also used for elucidation of the biomarkers for the select agents. When we say select agents, they are pathogens or toxins which are of biothreat significance. So you already know what is biothreat, so I need not emphasize on that. So we have also used for multiplex protein detections. As I said, we have developed MRM assays for several toxins, which can be analyzed using this tool in a simulated biothreat scenario that, yes, there is an aerosol release. I can collect that and tell what toxin is present over there using the tandem mass spectrometry. We have used proteomics for elucidation of vaccine candidates. This is the one of the examples where we have used it for elucidation of a potential candidate for gas gangrene, and for the elucidation of the biomarkers for epsilon toxin exposure, where this is the toxin which is very important from biotech perspective. What really happens when the mouse is exposed to this toxin at the proteome level that we have described. And this is all I wanted to say, and to tell you, to summarize in a nutshell, that proteomics has great potential in the days to come. I don't know, questions are allowed in this session, otherwise. Yeah.
Thank you very much, sir, for very informative lecture on proteomics advancement and approaches for the processing of technology. Biomarker analysis through the developed techniques, really a good approach for the proteomes. Thank you, sir. Now it's time to express our gratitude, and here we have a token of appreciation in the form of memento. We request our dignitaries to kindly felicitate to our honored guests. I request Honorable Pro-Chancellor Lieutenant General V.K. Sharma, Ati Vishish Seva Medal, to kindly felicitate our chief guest, Professor Dr. P.S. Bisen. I request officiating Vice Chancellor Professor Dr. Anil Vashish to kindly felicitate guest of honor Dr. A.K. Meena, Institute in charge, Regional Ayurveda Research Institute, Gwalior, with a moment. And now request Pro Vice Chancellor Research Professor Dr. M.P. Kaushik to present a memento to our keynote speaker Dr. S.I. Alam. Thank you, sir. Now, I would like to call our Director, AIB, and the convener of the International Conference to give the formal vote of thanks.
Good afternoon. I stand before you with a profound sense of gratitude and honor as we conclude the inaugural session of the Third International Conference on Recent Advances in Biotechnology and Nanotechnology, INT BioNano 2024. Today, we have witnessed the convergence of brilliant mind and innovative ideas in the realm of biotechnology and nanobiotechnology, and it is indeed a momentous occasion. Firstly, I would like to express our sincere thanks to our Pro-Chancellor, Lieutenant General V.K. Sharma, Ati Vishish Seva Medal, for gracing us with your esteemed presence. Thank you, sir. Your unwavering support and commitment to the advancement of education and research have been instrumental and in the success of this conference. We are truly grateful for your inspiring leadership and vision. A special note of appreciation goes to our esteemed officiating Vice Chancellor, Professor Dr. Anil Vashish, for your continuous guidance and dedication to fostering an environment of academic excellence. Your efforts in promoting interdisciplinary research and global collaboration have laid the foundation for events such as INT BioNano 2024. We are honored to have our distinguished Pro Vice Chancellor Research, Dr. M.P. Kaushik sir, with us today. Your passion for research and commitment to pushing the boundaries of knowledge have been pivotal in making this conference a reality. Your guidance has been invaluable, and we look forward to continued support in our pursuit of excellence in research. We are privileged to have our distinguished chief guest, Professor Dr. P.S. Bisen sir, and guest of honor, Dr. A.K. Meena, with us today. Your presence has added immense value to this gathering. Your expertise and insight are invaluable and we are grateful for your contribution to Int Bio Nano 2024. A heartfelt thanks to our keynote speakers, Dr. Mukesh Dubey and Dr. S.I. Alam, scientist F, whose illuminating address have set the stage for what promises to be captivating and intellectuality stimulating conference. Your wisdom and expertise have enriched our understanding and sparked new avenues of thoughts. I would like to thank IT team for smooth conduct of the functions. Thank you once again and wish you all a productive and inspiring conference. Thank you very much.
Thank you, sir. And thank you all the faculty member, HODs, HOI, and all the students. May I now request all the members of the team kindly join the stage for the group photograph. Dear all, thank you. And you all are cordially invited to join the high tea and get back to your respective session at 1 p.m. Thank you.
Amity Institute of Biotechnology welcome you all in the main session. For first main session, chairperson is Dr. Pranav Kumar Mandal, Principal Scientist, National Institute of Plant Biotechnology, LBS Center, Pusa Campus, New Delhi. And co-chairperson is Professor Dr. Vinay Dwivedi, Amity Institute of Biotechnology, Amity University, Madhya Pradesh. For the first main session, today's first invited speaker is Dr. Pranav Kumar Mandal. Dr. Pranav Kumar Mandal, he is Principal Scientist in ICAR National Institute of Plant Biotechnology, Delhi. He did PhD in Plant Biochemistry and Molecular Biology from IARI New Delhi in 1996. He has 33 years of experience. He served in ICAR ARS Scientist from 20th Feb to 90th Feb 2004, Senior Scientist from 20th Feb 2004 to 90th Feb 2010, and as a Principal Scientist from 20th Feb 2010 to till date. His research area of interest is enhancement of nitrogen use efficiency in cereals. He has awarded by many awards like Adjunct Professor and the Faculty Council member of UBKV, Cooch, Bihar, West Bengal, Editor, Board member of SAARC Journal of Agriculture, Editor of Journal of Plant Biochemistry and Plant Biotechnology, IBSC member of Bayer Crop Sciences, IMC member of many ICAR Institute. He has gold medal in MSc from IARI New Delhi for outstanding academic performance in 1991. He is CSIR ZRF, CSIR SRF, NET, ICAR ZRF, and SRF and NET qualified. He has more than 90 research papers published. Scientific review articles is 4, edited book is 17, and edited newsletter is 16. He has published more than 32 publications, manuals, popular articles in conference and conference proceeding. He has awarded by best paper in last 5 years for 5 times. His H index is last 5 years 18 and I-10 index is last 5 years is 23. I welcome you, sir. On behalf of Amity Institute of Biotechnology, kindly continue.
Thank you so much. I was actually waiting from 12:40. Is it audible?
Yeah, it's audible, sir.
Yeah, actually, I'm having a bad health as well as bad throat. So I was actually worried till yesterday whether I'll be able to talk because the voice is not coming properly. But then I think I'll manage somehow. And I thank Amity Gwalior. I had been there once. Otherwise, also overall Amity I am aware of because Noida Amity I have 5 students did PhD from here. It's a good opportunity. Raghavendra is there, who is basically like our student. So with that, we came to know about the Amity Gwalior, and I saw some of your work also doing good, because being a academic institute and mainly for teaching, there are quite a few good papers what I saw. In this session, maybe I may not be able to continue for the entire period because I have class from 2:30 onwards again. So what I'll request my co-chair to continue after I leave. But then let me share the slides for presentation, and I'll try to be brief. 20 minutes time is there. I'll try to keep within that. So few of the slides I'll just skip, and then I request, because the area is quite vast, and of course it is a recent technology. So my email is there. If anybody want any kind of work together or means jointly doing later, we can do that. So let me share the screen. Co-chair, anyone, anything you want to say now? Sir?
Good afternoon, sir,
Yeah.
and welcome. I'm audible, sir?
Yeah, you are audible.
Yeah. Uh,
So you want to make any comments, or we'll start?
no, sir, we can start, sir. We can start.
Yeah.
Thank you, sir. Thank you.
Right. So is it visible?
Yeah, it's visible, sir. It's visible.
Yeah, let me go through
Yes, sir, it is
this.
visible.
So the topic is gene editing for enhancing nitrogen use efficiency in cereals. Basically, I'll talk more of nitrogen use efficiency, and then of course gene editing is quite a large area, So once you know the gene, you can edit them. So the target here, it is an example how you actually identify the genes for your traits. And then once you know the genes, you can edit it as you wish. There are different things you can do. In fact, if you want now, the transgenic can be completely replaced by gene editing, which we are telling as SDN3 approach. So to start with, basically, I'm in National Research Center for Plant Biotechnology. This is Biotechnology Department or Division of Indian Agricultural Research Institute, where from we have studied and we are actually teaching now. So coming to this nitrogen use efficiency, we say nitrogen use efficiency, basically the priority is, it is a nutrient use efficiency part, but more than nutrient use efficiency, the bigger importance at present and priority is nitrogen use efficiency for mitigating the climate change. And ultimately what we want is we want to improve the nitrogen use efficiency so that lesser amount of nitrogen will use, and
yield will not be affected that much. And the crop which I target is rice and wheat. Of course, I work on rice, wheat, maize, all three. But maize is not on nitrogen use efficiency and not in my lab. Basically, the students where I'm advisory committee and quite a few students are working on maize gene editing. Now, coming to nitrogen as a plant nutrient, it is very, very important. It's not only for the plant, but even for the animal, nitrogen is extremely important macromolecules, and it is required for its development. It is present in amino acids, protein, nucleic acids, hormones, almost everywhere. So it is very, very much required. And if you want to increase the productivity, naturally you have to increase the fertilizer. That is what is a general trend. And from the time the yielding crops have comes, mainly the rice and wheat, nitrogen use has increased quite high. The interesting part is, which is not the part of the talk, that nitrogen actually helps in overcoming many of the other stresses, which we observe. We had a dual stress along with nitrogen stress. We have given a drought stress. The plants which gets affected more when you have a higher dose of a nitrogen, they actually combat that. Similarly, many of the diseases, basically nitrogen, especially parasites. Just we are talking with one of our colleague. In case of brassica, there is there's a nitrogen, it helps. So basically nitrogen use is rampant, you can say. And if you see this is taken from the net, and if you see that rice and wheat, they take the major part of the nitrogen use, total nitrogen fertilizer what we use. And these are the three major fertilizer: nitrogen, phosphorus, and potassium. Of that, the blue is nitrogen, and it is always much, much higher than other two. Now, the problem with nitrogen is the amount of nitrogen we use, 50% is the maximum level where it gets used, and most of the things get lost. So they can be lost in a different way. It can get lost almost up to 80% through ammonia in the air. 3% go by denitrification, 20% can go for nitrification, and 20% of the applied nitrogen is lost as a nitrogen gas itself. And into the water by leaching, it goes almost around 25%. It's a BASF slide, this part. Why BASF is doing this? Because they wanted to stop a urease inhibitor, so that, or nitrifying inhibitor, so that they can stop this loss a little bit. But that is apart. When you are using the normal urea, this is a big, big loss. And in a quantitative term, there is a huge loss. So if you say this is 37 trillion grams, here 22 trillion grams denitrification, leaching. So it's a huge loss, and not only monetary loss, but this losses causes a huge amount of environmental pollution. Now, what is the purpose? By this time you understood, we have to use less nitrogen, minimum yield compromise. Naturally, for that sake, we have to increase the nitrogen use efficiency. And nitrogen use efficiency, if somebody is working on that, or somebody goes through the literature, you will get around 14 different definitions. Somebody says that how much of nitrogen you use means per unit of nitrogen, how much of photosynthesis it does. Physiologists usually they say, agronomists say how much biomass. Similarly, there are many, many definitions, but for us, practical purpose, we say how much of output, in the sense, basically we are talking output in terms of yield. So how much of nitrogen fertilizer we are using, or you can say that how much of available nitrogen in there in the soil, and how much yield we are getting. So to increase this nitrogen use efficiency, the part where we are coming to genome editing, before that there are other approaches, and long time back people are using. One is management, how we use the nitrogen fertilizer. Second part is biological nitrogen fixation, which is by microbes. And third part is our concern, where the plant perspective will be looked into. Now, coming to the nitrogen management, what you say, it all depends upon what type of soil, what type of climate, what are the different types of fertilizer. Like at present, all the urea what you get is neem coated. Neem oil is having urease inhibitor. So urea becomes ammonium carbamate, then it becomes ammonium carbonate, and then from there onwards, it's actually start getting lost. So if you can, and it is highly soluble. So if you can actually use a urease inhibitor, natural urease inhibitor is neem oil. So then you can reduce that. Then how you apply, like nowadays they use daily also, like a fertigation. But these are nothing to do with crop rotation. They are nothing to do with the plant's perspective of gene. So that's why we call it as a management. And by management means large amount of nitrogen saving can be done. So they are doing a great part of the job by nitrogen management. The second part is biological nitrogen fixation, that is by microbes. You know, in case of legume, there are symbiotic microorganisms, but other than that, there are a lot of what is called free living organism like azotobacter. We had actually done a genetic modification of a bacteria, and it is able to save 25% of the nitrogen in terms of area in case of wheat. So free living you can do. You can improve the symbiotic bacteria out of different, suppose rhizobium you are seeing, which is much better like this. And in this symbiotic, there are actually, we have a project where we are quite ambitious and we have gone little bit headway you have gone, but unfortunately the person who was working, he expired in this project which I am coordinating. There we wanted to make the rice fix nitrogen. So what we want is we take the gene from the legume, put it in rice, and want to see why rice is not able to fix nitrogen. And at present we have the transgenic plant where rice is showing that it is going to develop the nodules kind of a thing. And the last approach which we thought of, but probably will not be able to do because he is not there. There we wanted to transfer the genes of legume, means NIF genes, into the plant itself. We don't want the help of any symbiotic organism. That's okay. That is another part of that. And then when we are going, this is a paper last year it came. We wanted to see what legume is having and what rice is not having. Because of that, rice is not able to do the nitrogen fixation, because there are many symbiotic diazotropes which are present in rice, but they are not able to form nodule. They are called endophytes, and very little quantity it is there. So last year we got good hints, and we are still continuing with this work. But then coming to the last part where we are talking about the identification of genes, you know. So here we are talking about the biological nitrogen use efficiency of the plant. So there we are looking for the genetic potential of the plant. Same amount of fertilizer, same plant, same environment, same soil when you put, some plants are able to give better yield, some plants are able to take more nitrogen, some are not. So we want to see why this it is. Okay. So basically, if you see overall, this is the outline. Here, just one review has got accepted. That's I didn't give the reference, but we just wrote in that. We had lots of work on that. Of course, this is the nitrate transport, this is ammonia transport, and then when it goes inside the plant, after that, this part is uptake, then assimilation it happens by GS and GOGET, and finally it forms a particular amino acid, and that is the major amino acids in that. And these are the pipelines you see. In case of nitrogen use efficiency, you can work on nitrogen acquisition, assimilation, utilization, remobilization. And there are list of genes and pathways which you can look into. Now, we did little bit of thing. We are just showing on that. Coming to that acquisition part where root is most important, we have done a thorough study on root, and we have seen how different types of root is actually affecting the nitrogen uptake. These are all on root. Same root when you put in hydroponics, when you put in soil right perlite, when you put in soil, they behave completely differently. There are some good papers came out of that. And here, natural variation of root architecture in diverse wheat genotypes grown under different nitrate conditions and root coat media. So we have, as I told, there are a lot of work we did on root, and this is a major parameter. Very recently, one article has come where they say the depth of the root is a big factor for nitrogen use efficiency in case of rice. But then which gene you will be doing for that? Now coming to the assimilation, as I told, the first amino acid is glutamate, and whatever we produce, exceptions are of course there, but whatever we produce having nitrogen, they are all coming from the glutamate. Glutamate to hemes or chlorophyll, glutamate to nucleic acid, glutamate to other vitamin biosynthesis, everything starts from the glutamate. And these are the known genes or known enzyme: nitrate reductase, GS, GOGATE, GDH, AS, and this is called C-page. So these are the major player which it does. But interestingly, they actually does not do so much of change because they are ubiquitous. Now, if you say somebody is respiring very good and some is bad, that usually doesn't happen in case of a human being because this is ubiquitous. This is very, very essential. So these enzymes are mostly very crucial and ubiquitous type of enzymes. So not much of variation or not much of change in this can make a great change in nitrogen use efficiency because they are essential. If they are not there, plant will not survive. Now, remobilization, while we are talking, there the plants die. Then there are sometimes phosphorylation happens where the ammonia, whatever has been assimilated by amino acid, they get again released and they get utilized. The plant which can make use of them better, they are more better nitrogen use efficient. Now, all this process is regulated by few transcription factors, few proteins, and few microRNAs. This is all, most of the cases, they are all known. And overall, we have a good work on this on DOF. There is a DOF transcription factor, which regulates quite a few enzymes which are related with nitrogen use efficiency. And that is the first time, not us, but then they did in Arabidopsis and told that nitrogen metabolism is not only important, but carbon metabolism is also equally important because this GS GOGATE, which produce the amino acid, it requires a carbon skeleton to attach that nitrogen into that. So for that sake, basically, you require carbon metabolism also equally efficient. So we had a good work on that, DOF is actually responsible for nitrogen use efficiency. Now we characterize few candidate genes. This is first time we identified the gene. These are all summary picture of the publications. There are around 5 publications where all the candidate enzymes, nitrate reductase, nitrate reductase yet to be done, GS, GOGATE, CS, and ICDH, which is carbon metabolizing, which we have seen. And in case of a wheat, the interesting thing is one wheat plant is a 3 plant. Their A, B, D, 3 genomes are there, is hexaploid. So how each genome is working, which we characterize. So this is again, it is done in case of a rice as well as in case of a wheat. I'm just going fast because we have to come to the genome editing. So finally, when you know that these are my genes, then you can start with that. But then how do you identify these genes? These are all candidate genes. And most of the candidate genes are essential, as I told, they are ubiquitous. So once you know, you have different strategies. You can do transgenic, you can do mass, or now what I am telling is you can do through the genome editing. Now, identification of genes, this is the pipelines. You can identify genes that somebody else has done through the literature, we do. You can do transcriptome, proteomes, genomics. Then you have to actually, once you find out both the cases, you have to functionally validate that. And then you have to see a genetic variation analysis that yes, it is actually the genes are working like this. Then do a comparative genomics, then genome-wide study, and then gene expression study in a set of genotype. Now, in this, what we did is basically when you are trying to do a genotype, when you need a contrasting genotype, it takes around 10 years to find out a contrasting genotype. Recently, we published a paper in which I have not cited in Agronomy and Crop Science. The work started in 2013-14. And developing a field with low nitrogen is another challenging thing. Nobody will give a field that year after year, you will not be putting nitrogen into that. So we did it almost consecutive 4 to 5 years. And finally, we selected this. We started with 300 genotypes. Then we selected to 60. Then next year, we came to, there are different doses of nitrogen to that. These lines are all different genotypes. So this is the variation. So from there, if you take this and this, two extremes are two different, means contrasting genotypes with respect to different parameters. Then those contrasting genotypes we have to grow in control condition. whether they are behaving similarly. And similar thing we did in case of rice. And rice, since it grows in hydroponics, it was wonderful. This is the first time anywhere in the world we did nitrogen, under nitrogen stress, full grown rice, 100 genotypes. Almost it is a huge area. Almost it is around, you can say around 400 square yard of space, full hydroponics. And this is my PhD student who is from Nepal. So we did that. And from there, we found some of the genotypes, which is very good in uptake, very good in utilization and nitrogen use efficiency. Then after that, you can develop a mapping population, the people for the breeders, and you can, this is the pipeline, once you have a mapping population, you can do genotyping, phenotyping, analysis, QTL analysis, find out the genes, then you can validate the genes. From this part onwards, gene validation, use and product, you can actually go really for gene editing. Same thing we did, we actually found out the contrasting genotype of rice from there. And we did a QTL analysis from a mapping population. At the same time, we have done transcriptome analysis of these contrasting genotypes. We overlap both. We call it as a co-localization. And we identified 20 genes which are completely novel, not those genes which I was reporting that nitrate reductase and all. Nobody knows what are the genes. Now my job is to validate them. Yes, functionally validate them. There are two ways of functionally. This is the list of 20 genes. So functionally validate them means there are two ways. You have to make it a gain of function. You have to make it a loss of function. So gain of function, you can overexpress and do, but loss of function, you can use knockout, and there your gene editing works. It is not for the product. It is just to say, yes, this is the gene which is really having a function on nitrogen use efficiency. We already picked 4 genes. All 20 we characterize, and 4 genes we picked, and we are going for nitrogen use efficiency. Sorry, functional validation. Parallelly, of that, 3 genes were negative regulators. Please try to understand what is negative regulator. They are negative regulator of nitrogen uptake. When the nitrogen is there, they say you don't take up so much. It is not required. So those are the inhibitors, the proteins, they are the inhibitors of nitrogen uptake. If you can stop them, then the nitrogen uptake will be more. And if the nitrogen uptake is more, when suppose the plant is taking 40 kg, when you are applying 100, actually it could have been by 60 kg also if the uptake efficiency is more. And if you are able to knock out those negative regulators, your uptake will be more. So we found 3 actually in case of rice, and we found the ortholog of those 3 in case of wheat also. Of that, one is not related with wheat because it is ammonia transport. It is important for wheat, but this is in disease resistance that they work, because ammonia is not the preferred nutrient for wheat. But then in case of wheat, basically we found other 2. So now we are trying for gene editing directly for developing a product, because if we are able to knock them out through gene editing, we will be able to get the plant where the nitrogen uptake will be as much as it is required, but the nitrogen application will be less. Hence, we will be using the fertilizer. Simply, as I was telling, in case of rice, we have to use 100 kg fertilizer per hectare. Now, it takes around 40 kg. If the efficiency is more, if the inhibitors are gone, probably we can reduce to 60, 70 kg. So that is actually a great saving. We are hoping for that. Plans are there. Now, for coming to the gene editing, many of you know, and if you don't know, this is the pipeline basically. It is identification of target, in silico designing of guide RNA, then designing of Cas system, transformation of transformation vector into explant, selection of transformants, then regeneration. There are lots of things are there on that. In the background, what you are working, just like that, you cannot take a gene from somewhere and then do, because in the background also you have to check that gene is same or not. So it looks simple, but it takes a lot of time and a lot of things. So selection of transformant, then regeneration of genome edited plants, means transgenic plant will be with the CRISPR-Cas. Then you have to check whether they got edited or not. Then confirmation of gene edited plant. Then you do either selfing or back crossing of them, then make them free of transgene, because we don't need any more this transgene. Then selection of transgene free edited plant, After that, you phenotype. Then you go to the field testing. If it is SDN 1 and 2, directly you can go just by information to IBSC, and then you can go for releasing a variety. And it is for any trait, but then in nitrogen use, we are almost another maybe 2 years of the last step. And this is what basically it happens. There are SDN1, SDN2, SDN3. There are two ways. Principally, there are two ways of any kind of gene editing. It happens. ZFN is there, TALEN is there, CRISPR is there. We are talking about the CRISPR here. It is more preferred than anything else. Of that CRISPR also, SDN1, SDN2, and SDN3, SDN3 almost equivalent to a transgenic plant. In case of SDN1 and 2, this is a small mutation. Since it is a small size mutation, it is approved. Means it is like you can change, if you know that one plant is having a better allele, you can actually by SDN2 or by prime editing, you can change 1 or 2 or 3 or 4 nucleotide base and alter the allele just like that. SDN1 is basically we do just knockout. So they are not considered as GMO and it can be done. Now the bigger problem comes, what we are facing is for one particular negative regulator, we have 75 plants. And how do you screen them? This is a huge, huge task. The people who are doing on genome editing, really, it is from superficially, whether it's a DBT expert or whosoever, they say, aise kar lo, aise kuch nahi hota hai. Because when you have two guide RNA, that itself selection is an extremely difficult thing. So you have to isolate the genome edited plant, then you PCR confirm, then you do southern hybridization. People say, why do you do southern hybridization? That's because copy number is important. Why copy number is important? Suppose one copy of the transgene means your cassette has gone, or two copies has gone. If one copy has gone, when you are making transgene free by selfing, the probability of becoming transgene free plant is 1:3. Hence, it will segregate as 1:2:1. But if it's 2 copy, it will be 1:15. So that it will be like what is called like double hybrid type. So if you have a plant which is having a single copy, knowing by the southern blot, you will be processing for the next generation or taking the F1 seed for that. Then you go for Sanger sequencing, and you can do the next generation sequencing. In that sense, you can do complete gene sequencing when you got the plant, because once you get a plant, whole genome sequencing for rice is only 20,000 now, 20,000 rupees. Now, this is a slide. What I wrote is given in the slide. Basically, this is a pipeline, what we say. Actually, there are a lot of nitrogen is being used after the green revolution. And a lot of work has been done of genomic, transcriptomics, proteomics, metabolomics, and NGS technology. Gene have been identified. And then basically, it is lots of work is going on gene editing and its delivery method also. Finally, by this way, you can get a product. And if you have to do a functional validation of many of the genes, then those genes, once you have functionally validated, those genes can be used in different approaches also. There are 2 more slides. I think 2 or 3 more slides are there. And this is typically, it looks when you have a gene edited plant where you will be having enhanced nitrogen uptake, which I was taking because the inhibitors are gone. So plant is to be healthier, and unused nitrogen, when it is a wild type plant, it will be like unchanged as it was. And basically, this is a overall picture if we say. So the main thing of the work is identifying the gene, and after getting the genome edited plant, screening them and finding out the product, because it's a huge task. So you actually in silico design the thing. If you have a wrong design in any case, suppose you have designed the primer and you have not checked the background. Background means the plant which you are going to edit, there is a mutation in the PAM sequence. The entire effort of 3, 4, 5 years will be waste. So you have to spend considerable time in design the guide RNA. And then usually we go by the functional domain. If you have a target gene having 3 functional domain, we usually target at least 3 guide RNA. And then you have to find out a suitable transformation vector where you put, usually vectors are available with the Cas9. And of course, the IPR issue is still bothering whole Indian government. But then still we are using that. Probably we'll negotiate with the people who have those Cas9 vectors. Mainly we buy through AdGen, where it is given as a repository. Then there are different ways of doing things. We have used both biolistic as well as agrobacterium mediated. And finally, you get the plants. And then, as I told, you have to do a large number of screening and you get the transient finally with the sequencing. You are confirming that, yes, it is my gene edited plant. And this gene edited plants is having all the phenotypes which I am looking for. And then you can actually go for the releasing. But the last slide is, it is not that nitrogen use efficiency alone, but beyond nitrogen use efficiency, any traits, if you have a gene, especially if you have a negative regulator, straight away you can go by SDN1, which is most easy. Just it will be knocking out, and you can develop a much improved plant that per se. Now, this is my duty to acknowledge all the people who have funded for last 10, 12 years, all ICR officials, DVT, CIMIT. We had an Indo-UK project, all the partners from UK, as well as different collaborators of different institute. And Dr. H.K. Das, who was actually the Raghavendra may be knowing his time, he was there. He is very, very close to me somehow. We had work on quality of wheat also. He was head of department of JNU. He had the patent on this azotobacter. Now we did in case of wheat, and we saved 25% of the nitrogen. But at present, government is not having a guidelines for releasing the genetically modified microbes. So thank you so much for the opportunity.
Thank you so much, sir, for enlightening us, including participants and students, on gene editing for enhancing nitrogen in cereals and various applications of genome editing. Thank you so much, sir, once again.
Sir, I
We
have
will
one
take,
curiosity. Ma'am, yes.
yes, sir.
Sir, I have one curiosity. The similar kind of things can we also do on legume crops because they have already rhizobium, so nitrogen is already being there utilized by these atmosphere.
So far, legume. So
Legumes
far,
also.
I'll come to that.
Use efficiency.
Legume so far means if you take 10 years back, nobody was talking nitrogen and legume because legumes are not applied for nitrogen. Most of the cases, basically, we do a crop rotation of cereals and legume so that in the other season, legume will be fixing nitrogen, and in the soil, they will enrich with the nitrogen naturally. But of late, last 10 years, agronomists, they say that actually the yield of nitrogen, yield of legumes means pulses are less. That is because it is feeding the bacteria. आप वो बैक्टीरिया के ऊपर डिपेंड मत करो. That's what they say. You apply nitrogen, and the amount of yield you will get means at the cost of the plant, the bacteria are getting fed and they are giving nitrogen. He's telling, I don't want this bacteria. So now the work is started on legume on nitrogen use efficiency. So they are doing in a different doses of nitrogen, at least some amount of nitrogen they are putting in case of legume. And yes, legume also nitrogen use efficiency is presently people are working. But then cereal takes the major chunk of nitrogen.
Thank you, sir.
Thank you, sir.
Thank you.
Sir, we will take all questions in panel discussion. Now, our next speaker is Dr. Tanusri Kaul, researcher and group leader, Nutritional Improvement of Crop, International Center for Genetic Engineering and Biotechnology, New Delhi. Dr. Kaul completed her PhD in botany from Delhi University. She has dedicated her career to advancing understanding of plant molecular biology and biotechnology. Her research focus on developing innovative strategies to enhance the nutritional value, stress tolerance, and yield of crop plants with the aim of addressing global challenges such as malnutrition and food insecurity. Dr. Kaul has been recognized for her outstanding contribution to the field, receiving prestigious awards and fellowships such as the BioCare Award, Rapid Grant for Young Investigator Award, and Postdoctoral Fellowship from various institutions, including ICGEB and the Indo-Israel DST Fellowship. She has authored numerous publications in peer-reviewed journals, showcasing her expertise in areas such as genome editing, stress tolerance mechanism, and plant biotechnology. She has started as group leader in 2006 and at present she is group leader in nutritional improvement of crops. She has published more than 15 publications in different international and national journals. I welcome you, ma'am. Please continue. Tanushri, ma'am.
Yes, I'm here. I'm just trying to share my PPT.
Okay, ma'am.
Am I
Yes, ma'am, it is visible and you are audible, ma'am.
audible? Yeah. Once again, I thank Dr. Raghavan.
Ma'am, one thing, ma'am, your camera is off, I think. Yes, ma'am. Now you are also visible, ma'am. Welcome you, ma'am, once again. Please continue.
So I thank Dr. Raghavendra and the Amity University director and all the entire team that invited me for this talk. Thank you again. As the stage has been set by Dr. Pranab Mandal, and it is very nice to see him after so many days. So this is what I wanted to present what we have done in the last 7 years in this field of crop improvement with special focus on nutrition. And in this regards, we have developed several genome edited lines. I would be taking up a few of them and describing some of them. However, we have in all, worked upon nutritional enhancement and trait improvement in maize, rice, pigeon pea, arar dal, tur dal, and as well as soybean. And we are also trying to improve the nutritional traits as well as crop improvement traits with respect to herbicide resistance in mustard as well as cotton. And we've also improved the cotton yield. Yield has been increased through genome editing. We have developed tomatoes with increased lycopene content as well as increased vitamin D, and we are about to increase the other traits like GABA, etc., together simultaneously. And we've also developed tomatoes with reduced allergenicity. So we knocked out 3 genes that are actually creating allergenic proteins that affect around 20 to 30% of the population, which cannot consume tomatoes. So giving this a brief view of what we have developed, eventually we know that crops produce food, feed, fuel, and other consumable resources for human life, thereby contributing to the progress of human race and society. The world is predicted, as you know, to hit 9.6 billion by 2050. And to feed the entire global population, what is predicted is that 100 to 110% increase in the crop yield is to be attempted, and thus to feed and nourish the burgeoning population of the globe in the face of climatic changes and decreased arable land, as well as severe drought, as well as water shortages. There is a pressing need for innovation in crop breeding technologies to increase agricultural
Excuse
productivity
me,
as well as accelerate.
sorry for interruption, ma'am. Your slide is not changing.
Yeah, I haven't changed it as in. Yeah. And it, I will change it in due course. Is it changing now?
No, ma'am, it's not changing.
So there is a problem, I think. Is it changing
Yeah.
now?
Yes, ma'am, now it's changed.
Yeah. So there is a pressing need for innovation in crop breeding technologies to increase agricultural productivity and accelerate sustainable agricultural development. in this regards, I wanted to highlight that soil is an important fundamental source, and we are treating it as a resource. We need to stop treating it as a resource and start treating it as a source, because as Dr. Mantal said, even if you are knocking out the negative regulators of nitrogen uptake or iron uptake or whatever, phosphorus uptake, eventually all the uptake is through the soil. And if the soil is deficient of nutrients, there would be no nutrient in our food. Hence, even though we do genome editing and we try to knock out these genes that are negative regulators of nutrient uptake and efficiency, we need to see what is there in the soil. So what is being proposed is that at least 3% to 5% organic matter should be there in the soil, and that has to be into the policy of the Indian government has to put it in the policy, where farmers are given some incentives that they maintain this kind of organic matter, which is essential for microbial population growth. Hence, this is what highlights the brevity and the depth of the situation, that every 5 seconds a soccer pitch of soil is eroded, and it is estimated that by 2050 around 90% of the earth's soil would be degraded. And what does this mean for the people? This is what said. There is there are more microbes in a tablespoon of soil than human beings on this earth. So we need to take care of this microbial population in the soil in a way where we can balance the nutrients there, and we do not take away the nourishment, and by just adding fertilizers would not help. So in this case, we need to develop certain technologies technologies, as well as go for situations where we do not perform monoculture or be dependent upon few crops. So at present, there is a lot of biodiversity, although it's decreasing every day. However, we are still dependent on 14 to 15 crops. However, this is leading to depletion or deficiency of nutrients in the soil. Whatever genome editing, whatever soils we try to maintain, we need to first of all change ourselves, our eating habits, our diet. So this is what situation is there. And every year Costa Rica is lost almost. So because we are continuously expanding our cities, and also most of the farming in most of the areas throughout the world is being done for family consumption. So this also is leaving us only less than 2% or 1% soil for cultivation and for sustainability. So sustainable soil management could produce up to 58% more food. This is what I need to press here. By my lab is therefore focusing on developing different kinds of foods and diversification of diets that would enhance actually replenishment of the soils, in addition to employing the new breeding technologies like CRISPR-Cas breeding. And this would eventually lead to boosting the cultivation of different kinds of crops that would be micronutrient rich. For example, the underutilized or orphan crops, for example, Vigna umbellata, we have tried to extract the importance by genome editing. We are trying to bring this into mainstream. So this is one way to tackle micronutrient malnutrition. So as we all know that nature has been editing and rewriting and altering genomes for a very long time, with providing enabling natural variants both in animals and plants to survive. And moreover, humans have been employing artificial selection and via domestication of crops for the past 10,000 years. And the quantum of crops that we presently consume have been domesticated from their wild relatives around 12,000 years ago. For instance, the present day corn that we consume is generated, or it has arisen from the wild ancestor teosinte. So we need to see that we are not doing anything extra than imitating the nature. So artificial selection has several methods in modern agriculture that have been identified, and amongst them, as you all know, is crossbreeding, mutation breeding, transgenic breeding, as well as transgene breeding. All these are time and labor intensive, as well as they are also known as the first generation, second generation, and the third generation technologies. However, the amongst these, this crossbreeding and mutation breeding are quite labor intensive. It takes several years to develop a variety, and also this also leads to narrowing of the genetic diversity by cultivating crops cultivating crops with certain traits that are selected, whereas the other traits are lost in time. So the narrow genetic diversity of cultivated crops is the prime cause for yield plateaus observed in the crop breeding programs nowadays. So according to a recent report, we found that 120 essential rice genes governing quality and yield traits revealed 28 genes having a single haplotype in a 3000K rice genome panel. And this could be reversed through genome editing. So this is what the latest technology is, but it is not a standalone technology. It needs to be supported by other arenas also to reconstruct this genetic diversity in the beneficial genes and alleles. And currently the fourth generation breeding technology has emerged as the precision design agriculture, wherein it is basically an integration of deep exploration on crop functional genomics and popular precision editing technologies, as well as big data mining, all synergize into the breeding strategies that can reconstruct the genetic diversity which is not there, or which is there but is lost, or which is not there. For example, in mustard, the oil content cannot be increased through breeding. So people are going towards these technologies. Hence, acceleration of domestication of wild species can be done through genome editing in a de novo domestication fashion, where the starting with the elite material, annotation of the genes, editing of those genes, breeding, bringing it into breeding, and crossing it with the current high variety mega varieties that are being grown throughout the country, and then field evaluation, which increases the elite traits in the crop, rather than going for direct domestication, which has led to land races but taken thousands of years to develop, and eventually cultivars which have limited selection of wild plants, and it's basically dependent on monoculture. So this technology is basically, mankind is on a verge of genetic revolution. It holds huge promise not only in crop sustainable agriculture, or it has importance in human health, somatic genetic engineering, gene therapy, cancer immunotherapy. Has recently Russia has also developed certain vaccines for for controlling cancer. Infectious diseases can be controlled. Simple C to T, A to T mutations in sickle cell anemia or progeria, or C to T mutations can be reversed back through prime editing, base editing, and through other tools that have come up. So this is how it will alter the food that we grow, medicines that are produced, animals that are modified, and also bioplastics, biofuels, chemicals, all would be modified, and it would salvage us in the face of climate change, weeds, poor nutrient levels, and pathogens. So these are the important products that have been already generated. As you all know, the MLO gene has been used for barley and wheat. Mutation in the MLO gene led to mildew, powdery mildew resistance in barley and wheat. That Huang Longbing disease that is in sweet oranges, which is affecting California, Texas, and Florida, has been controlled through genome editing. Cavendish bananas have been resurrected. So this is all, even different kinds of animals have been generated, and this is going on. Eventually, those traits that cannot be introduced through breeding could be introduced through genome editing. So here you can see the heterosis being introduced through genome editing, or knocking out of these 4 mutations in these 4 genes. Then you can see different haploid inductions. Double haploidy can be introduced. Entire metabolic systems or devices, synthetic devices, can be introduced in plants. And de novo domestication of the wild tomato has been done through these mutations in 4 genes. So this has been the past. So this is the technology, how genome editing has been evolved through all these stages, where previously it was precision breeding, ancient domestication, crossbreeding, mutation breeding, eventually transgenic breeding, and then marker assisted selection, as well as then coming up with the genome. So the first genome edited product was produced around 2019, wherein Calixed, a company in US, had developed high oleic soybean, HOLL trait, high oleic low linoleic acid, HOLL trait. It was produced. So then eventually a storm or a flood of genome edited crops came up, mostly in China, USA, and other parts of the world. And it is more precise than the random mutations that generally people had, like Mueller and Orbach had proposed. It is specific, targeted, and not random. Hence, it is more efficient. And it was started. The foundation was laid by Mario Capicchi's, the invention, the discovery, where he could strategize to enrich the embryonic stem cells in which homologous targeting events had occurred. So he devised a double selection method to select the targeted mutations. So this was then eventually taken up by these two ladies. And as you all know, it requires a target sequence. A target, it's a single guide RNA which is combined of two molecules, a target sequence, as well as a CRISPR RNA, as well as a tracer RNA. So the CRISPR RNA of the target sequence along with the tracer RNA eventually guides the Cas9 endonuclease to create a double strand break. And some latest tools have come up where double strand breaks are avoided, like prime editing tools. So this is how it was taken over, repurposed through bacterial genomes, microbial genomes like Streptococcus pyogenes. There was acquisition of the specific virus, piece of a viral genome was taken into the bacterial genome in the Cas loci and transcribed. Biogenesis was then eventually taken up by hybridization. So this guide RNA could be used as a hybridization tool, an RNA DNA hybridization tool, which would recognize the sequence of the viral genome and create a double strand break, therefore leading to destruction of the virus and hence protection of the bacteria. So this is how it was repurposed. And amongst all the existing programmable site-specific nucleases, CRISPR-Cas system has emerged as an explicitly prominent and efficacious tool that enables precise and targeted genome modifications in diverse organisms. You can see previously meganucleases came up, then zinc finger nucleases, then talons, then eventually CRISPR. So talons and zinc finger nucleases were mainly based on protein engineering, where you need to develop a specific protein for every different gene, and that was cumbersome and time consuming, and money, a lot of funds required for this. But when CRISPR came in, only to see a simple DNA sequence could eventually create a double strand break. And basically the mutation is introduced via repair. The endogenous repair machinery introduces the mutation. It is either through non-homologous end joining or through homology donor repair, where the homology donor repair mechanism is a precise one. So these are the it's a slide showing that CRISPR tool is really less cumbersome and dramatically less tiresome for researchers, and they can develop the experiments within a week. They can develop their synthetic guide RNAs and work for the plant only if the transformation protocol is standardized in the specific plant. These are the specific editing tools that have been latest tools that have been available, like cytosine base editors, adenine base editors. These are base editing where they use cytosine deaminases and adenine deaminases, whereas prime editing is another latest one, which it requires a reverse transcriptase along with a Cas9 or a nickase gene. Sorry, not a Cas9, but a nickase gene, where a single, not a double strand, but a single nick is made, and then eventually the sequence that you give for the template that you send in as a primer with PEG RNA. It's called a PEG RNA because it's a prime editing guide RNA. This would then be used as a template by the reverse transcriptase to develop the sequence that is desired, and hence replace the old sequence. Eventually, these all are available. It can be taken care of, as Dr. Mandal said, gene, but we have synthesized our own vectors indigenously. Hence, we are not going back to gene for this. So these are the different characteristics of nucleases used in crop breeding. I can send this slide to Raghavendra ji for the use of students. It's there where you can choose. Even after CAS 9 there is CAS 12A or CPF1, then CAS 13 came up, now CAS 14 has come up. It has different requirements and less stringency, so it can be utilized efficiently. And as of now, CAS 9 and CAS 12A or CPF1 have been utilized for, these are the main relevant plant genome editing tools, type 2 Cas9 and type 5 CPF1 or Cas12A. So type 2 and type 5 have been utilized as of now. And we have utilized Cas9 in our lab because of its simple and efficacious nature of engineering and adaptability for multiplexing. And you can see eventually it's a GIF where you can see you can replace a small fragment with a desired fragment easily through this technology, and we have done that for herbicide resistant traits in our crops. So this is a slide wherein we could see that if you want to start genome editing in your lab, you need to have the whole genome sequence of that crop in your hand. If it is not available publicly, you need to genome sequence it, because when you design your guide RNAs, initially you need to use those softwares like ChopChop and Cas or Finder or CRISPR-Cas. To all these tools generally require total complete whole genome sequence in the background, where these guide RNAs are actually blasted against, and they give you a list of target sequences or guide RNAs from which you can choose which is most efficient without any loops or bends or dimers formed, and with no mismatches throughout the genome. Hence, you reduce the off-targeting problem of genome editing. And in case of single traits like grain amylose content and quality of oil in soybean, so grain amylose content in rice can be through one gene it can be targeted. Starch bunching enzymes, if eventually one gene which is actually producing grain amylose can be targeted, like waxy gene has been targeted for making waxy maize and waxy rice. Similarly, in oil, increasing the oil content to increase the oleic acid content, FAT 2.2 isoform of fatty acid desaturases have been knocked out to increase the oleic acid. So similarly, you need to be careful on choosing the targets, because they should not be the housekeeping genes, because that would lead to the affecting your growth and performance of the plant. So need to select an appropriate genome editing approach. So in our lab, we have the first product that I want to highlight is the iron and zinc enriched rice, but not just in the allurine, it is enriched in the endosperm, which is actually what we eat after polishing and milling. So we have knocked out 3 genes together, one cadmium transporter and 2 iron sensing and zinc sensing genes, that have eventually led to the increase in iron and zinc content in the endosperm. This slide just shows how the more than 2 billion people are affected by iron deficiencies and anemias. Basically, they are from the populations where children are affected, school children, 43% of them, 38% pregnant women, and 29% non-pregnant women, all menstruation, young teenagers, they all affected by anemia. And anemia also contributes to 20% of the maternal deaths. So this is how it is more disastrous than other diseases like HIV and all, because a child will not grow enough old to die of HIV or any other malaria or any other disease, TB. It would eventually within 5 years would not survive. So it is more dangerous than other diseases that are prevalent. And also summing up, phytic acid enhanced enrichment in the seeds, also seeds and legumes, cereals and legume seeds, is also a problem, because even if you enhance the iron and zinc or micronutrients in the grains, this phytic acid with the 12 negative charges chelates away all your micronutrients. So all the positive cations like calcium, iron, magnesium, all are chelated, even proteins and starters. So to reduce the hidden hunger and to increase the micronutrient nutrients to billions of people, we need to have a biofortification strategy by 2025, where stunting and anemia has been highlighted to be reduced, has to be reduced by 2025, the WHO SDG goals. 2 and 15. So stunting mainly happens through zinc deficiency and other aspects, and carbohydrate deficiencies, and anemia because of iron. So this is how we've shown that eventually what we eat has only 2 to 4 ppm of iron, and what we require is around 50 to 30 ppm of iron, and zinc around 28 to 40 ppm to provide, in order to provide 30 to 40% of the dietary estimated average requirement. So in that case, and also cadmium is a problem for Indian soils, where not much as of now for India, but eventually it would. So that could, we also wanted to reduce by this strategy. So this is how we multiplexed, and we got a inspiration through we predicted a proof of concept before we could edit these genes through machine learning and AI. We could see that these expression of cadmium was specifically in the vegetative tissues like leaf blade and leaf sheath, and the iron sensors were specifically expressed in the embryos and the endosperm. So we tried to construct models to quantify iron levels in rice grains, and to achieve this we actually combined all the gene sequence data available, synonymous, non-synonymous, synonymous mutational data, gene expression profiles, protein interactomes, metabolite quantification profiles, descriptor profiles, all together. We put them together just to create a whole network of rice genome, wherein we could keep these three genes at the least neighborhood centrality, so that if we knock out these genes, we need to see what is the influence of this on the entire network. So we could see that once, if we knock these three genes out, there would be no major problem in the rice plant, with homeostasis would be regained. So this is a wild type model, and you can see that ferritin and nicotinamide synthase genes have been used for overexpression for last 35 to 40 years, where they have failed to develop enough iron and zinc in the endosperm. But by just knocking out these three genes, you could see that in the edited model, you could see these black dots are all other genes that are involved in iron and zinc uptake, transport, and storage. So when we knock these three genes out, you see there is enhancement of all these 18 to 27 irregularities that are involved for iron and zinc uptake, transport, and storage at grains, including NAS genes and ferritin genes. So this is how we made a ranking matrix of the data, and we could see that how ferritin gene upregulated it. The ranking went from 7575 to 521, and here for NAS gene it went from 11,000 to 1250. So this is how we arranged this, and we use a diffusion matrix to combine all the data, and we ranked all the gene regulations. Then eventually, based on expression profiling, in-house data, and geosets, we converted the entire upregulated gene intensities into ppm through this formula, which is termed as IBAC, or intensity based absolute quantification, which led to our 15 to 30 ppm increase. This was a proof of concept, and eventually we selected the guide RNAs, we knocked out the genes, then we assembled we created the cassettes, we developed the indigenous vector for expression plant expression vector we developed indigenously. And then this is a sequence of how a CRISPR RNA guide RNA is being formed. It is mainly a sequence that has a 20 nucleotide sequence of which resembles your gene which you want to knock out, along with a PAM sequence that is there in the end. And eventually then there is a 12 nucleotide CRISPR RNA repeat, and there is a 4 nucleotide tetraloop sequence, and then there is a 64 nucleotide tracer RNA, which basically is like a handle where Cas9 gets attached or settles. So these are the vectors that we used for NICTK1.
Excuse me, ma'am, we are running out of time. Please conclude. Thank you, ma'am.
Yeah. So this is how we developed the transformation protocol for Samba Masuri. We developed the T0 lines. We checked the Cas9 expression through southern blots. Then we eventually moved ahead to collect the T1 seeds. We developed the T1 positive lines. We checked the genes, whether the mutation is there or not. And eventually we got the T2 lines. Again, we checked for Cas9 sequencing, and then eventually for the 3 gene mutations. Then we segregated the populations. We created a T2 population, and eventually we had these T2 lines and three lines where Cas9 is not there anymore. These 8 lines, and these are the mutations that were identified in these 3 genes. And this is the basically the frame shift that has happened in the protein. And then they were the expression was down regulated in all the lines. The 3 genes were, through real time we found out the fold decrease in the plants, and on all the parts in the flag leaf and the node in the stem flower, everywhere we saw that. And we checked for these 27 18 genes which expressed highly, and this led to, because these are all genes that are related to uptake, iron and zinc uptake, transport and storage in the grains. So it was like all were overexpressing. Just by knockout of 3 genes, you could overexpress 18 genes in the rice endosperm and the rice plant. So this is the histochemical test that we did. So we removed the aileron layer and then we stained. So in respect to the wild type, you could see this iron staining in the endosperm with Prussian blue, and then the zinc staining with DTZ, and the cadmium staining. And this is for the T2 lines, and this is for the Cas9 3 lines, 8 lines. And then we checked the intensity, the content of iron and zinc through ICP-MS also, and we got like 15 ppm to 64 ppm iron we could get, and for zinc we could get from 20 ppm to around 70 ppm, and cadmium too. So morphogenesis, phenotyping was done in the fields. You could see that the yield is not affected. It's as good as a wild type. The grains are also as good as a wild type, but you can see a slight yellowing, but it is not any impact on the, it's not like black or, it's just a little bit brownish than the control. And the morphology was also tested with different parameters for T0 lines, then T1 lines, and then T2 lines. And eventually we have these lines, and we are giving them to Indian Institute of Rice Research, Hyderabad, where Dr. C. Nirja would be taking care of the phenotyping, and eventually in the ACRIP trials next year, it would come into the farmers field. So this is what we have developed for glyphosate-resistant maize, rice, pigeon pea, and we have a patent on this. We see how herbicide-resistant weeds create a problem. Eventually, with every generation, they increase. Hence, we need a sound strategy for weed management. Hence, we developed the genome-edited glyphosate-resistant maize, as well as pigeon pea and rice. I'm showing the maize part where shikimic acid pathway, the EPS-based gene, is the source center for glyphosate binding at the PEP binding site. Instead of PEP, the glyphosate goes and binds. So we try to modify this conserved region so that the glyphosate cannot bind and the PP binds in through genome editing. And this is to knockout and homologous donor repair. And we developed these lines where through, this is the native sequence, this is the mutated sequence, including the mutations that are seen. And this is the homologous donor repaired template that we use. And this is the strategy: you knock out the piece of DNA through designing 2 guide RNAs on the flanking regions of the conserved region. The conserved region has 2 flanking guide RNAs to remove this, and eventually you give a donor template with your desired mutations, and then that is being replaced to homologous donor repair, because these left and right arms are as good as the gene. And after that, this recognition is done, the entire new mutation part is thrown into the gene. So we get a precise sequence modification. Similarly, for astolactate synthesis, we have done the same thing for this strategy. So this is the the small video where you could see the mutation site, active site, PEP binding site in the EPSPS, and we introduced 3 mutations. One is the G2A, which actually is involved in recognizing, glyphosate recognizes this site through G, and we mutated it to A, and the rest is T2I and P2S, which is basically found to the weed resistant to herbicide, so that we introduced, which reduced the cavity region and it did not allow the glyphosate to bind. So this is how we developed these lines. We checked it on media, 4 millimolar with 4 millimolar glyphosate, our edited lines survived. Then we checked through Cas9 PCR. When the EPSP's mutation region was PCR amplified to nested primers, sequenced, and all the 3 mutations were there. We have 13 such lines, but 7 of them were the best, giving us higher yields. And this is how we tested through through Roundup spray. The treated lines are, edited lines are happily growing. And these are in the greenhouse here. We, in comparison to treated control, you see all the edited lines are significantly having higher photosynthetic efficiency and growth parameters. The EPSPS activity was also working well in all 7 mutant lines. And the shikimic acid was utilized, which is the starting point of the EPSPS pathway, the shikimate pathway. So it was utilized as good as the untreated control, but in the treated control, it wasn't being utilized because the EPSPS activity is hampered by glyphosate. The aromatic amino acids were checked, and that also increased, probably because the mutation that we did increased the binding efficiency with the PEP, and hence increase in the aromatic amino acids. And this is how the COPs could be seen. So there were no developmental defects. And this is a paper that we had published. This is in this year, Plant Physiology and Biochemistry. We did the similar situation for rice, and a student from Bangladesh, Sonia, helped me with this. And this is the transformation protocol we standardized for Samba Masuri. This is the native sequence of EPSPS. We designed 2 guide RNAs, and this is the target sequence that we need to remove and replace with the desired region. So this is the homology donor template. This is the vector that we can indigenously develop vector that we put the vectors, the cassettes of Cas9 as well as the guide RNA for EPSPS here. And this is the homology donor vector where we select the homology digested and shoot it along with the guide RNAs through ballistic mechanism. So these are the vectors that we have developed based on T-Cambia 1300 series, and these are the selection screening process. And eventually the calli were selected on glyphosate. It was standardized, and you could see the selection. Southerns were done, then sequencing was done for these 3 mutations were there. Then the leaf disc assays also showed that the leaves could tolerate chlorophyll degradation was less in higher concentrations, up to 4,500 ppm it could tolerate. So this is how T1 lines were selected. Again, we checked the aromatic amino acid levels in T1 lines, and then we did the southerns for T1s, and then again we grew these T2 lines, and we put them in through the assay, glyphosate assay. Then we did the evaluation in the field, and we could see that within 45 days the plants could recover, our edited lines. 15-day-old plants, 15 to 20-day-old plants, so 30-day-old plants were sprayed, and within 45 days they could recover, but the wild type could not recover. And then again we for the T3 lines we did the IRGA and all. These are the same experiments repeated for these T3 lines for EPSPS activity. For glyphosate, these 3 aromatic amino acids. So these are the lines which are basically Cas9 free. We chose those lines, and then we saw the southern Cas9 free southern, and then we have these seeds now for field trials. They are ready, and we are going to give it to Indian Institute of Rice Research again. So this is basically, so this is the dose dependent glyphosate spraying, and we could see how you can, it resists, the edited lines resists and tolerates Roundup. So this is the weed assay, competition assay. Your weeds are, when they are sprayed, the weeds are dying, but your edited lines are not dying here. after 14 days of spraying. So these are the pollen that are viable that in these lines to show that it is all is okay, the flowering is okay, everything is okay. So I take so we published this in Frontiers in Plant Science last year, and these are our partners. And this is my team. Thank you.
Thank you, ma'am. Ma'am, kindly unshare your screen. Thank you, ma'am, for very informative lecture on CRISPR gas technology for crop development and the genome editing. That is actually the essential for the development of the new crops. Uh, all the participants are requested to drop your message on chat box. So at the end of the panel discussion, we will discuss the question answer session, ma'am. So that time, ma'am, we will discuss. Also, all the invitees are requested to complete the talk within the 20 minutes. Now, next. Now, I'm welcoming Dr. Siddharth Tiwari, Scientist F, National Agri-Food Biotechnology Institute, Chandigarh. Dr. Tiwari leadership at NABI has propelled the transformative research initiative aimed at enhancing the nutritional content of staple crops with a particular emphasis on banana and wheat. His team pioneers work in banana genome editing utilizing CRISPR-Cas9 technology, has garnered international recognition, notably in augmenting provitamin A level to combat the malnutrition. Dr. Tiwari collaboration with leading institutes worldwide, including Queensland University of Technology, Australia, underscore his commitment for fostering international cooperation in agriculture biotechnology with over 50 peer-reviewed publications and multiple patents. Dr. Tiwari research continue to drive innovation in crop metabolic engineering, offering sustainable solutions to address global food and nutritional challenges. Tiwari sir?
Yeah, ma'am. Thank you so much. Yeah. Can you allow me to just
Sure, sir. So
put
you
my
can share
slide?
your screen.
I just
Yeah.
Are you able to see my that full
Yes, sir. Your
screen?
slide is visible, sir.
And my voice is clear?
Yes, sir. Your voice is also
Okay.
clear.
Thank you so much, ma'am, for giving me an opportunity. I am really thankful to Professor Dwivedi and my good friend Dr. Mishra for allowing or inviting me to just give brief introduction or brief glimpse about my work on plant biotechnology applications in crop improvement or crop biofortification. So in the first slide, I would like to just briefly introduce about that genome modifications or plant biotechnological approaches, those usually utilized in the laboratory. First one is like conventional plant breeding. As you know that it's a traditional methods where you can be cross two varieties, contrasting varieties. One variety usually contain the trait that you would like to transfer to the donor variety. And after subsequent generations, like 5, 6 generation, you will get that traits will be transferred into the variety that is desired. But in this case, you will get undesirable traits also, because during the crossing or breeding process, undesirable traits also be transferred from the allied variety to the donor varieties. And it is time-taking process. Second one is that plant nutrition breeding. It is very commonly utilized for the crop improvement programs. There are several programs usually uh, used to develop that crops by using mutation breeding, where you can be use that chemical mutagenesis or radiation mutagenesis like gamma mutations. And based on that, you will generate the populations and desirable phenotypes you will be judged based on that performance of plants, and accordingly you will select. Again, in this case, you will get several undesirable mutations in the plant crops, and that maybe have some negative effects also. But when you will talk about that plant genetic engineering or plant transgenic approach, where you can take the gene across the taxa, there is no limits. Any living organism gene that is helpful or you can say beneficial to the crop plants for any trait improvements, you can take this case. And that trait, that gene you can be transport into the plasmid, and plasmid can be utilized for the transformation in the plant system by methods like agrobacterial transformation or particle bombardments. So in this case, you will generate the crop plants, but again, in the possibility, because the random mutation will be happened in the crop plants, so that sometimes what happens that desirable traits can also be disturbed. So in this case, there is a one solution. You have to be generate many lines, maybe 100, 200, 500 lines. And then after screening, you can be judged that there should be any promising lines. There is no negative impacts on the plant physiology or morphology or molecular studies. So based on that, you can judge the plants. And fourth one is very novel or very recently, what we can say evolved, that is a genomating, and also known as a new breeding technique or new breeding tools. So in this case, genomating, you know that mutation is very precise, and most commonly utilized method is CRISPR-Cas based. My previous speakers, Dr. Kaul, has already been explaining details about this technology, so I will not go in details. But you know, it's a very precise technology, and it's one of the very recent advancements in plant biotechnology, where you can improve any traits in a specific manners. So in my talk, I would going to be discuss mostly about these two technology, plant transgenic as well as genomating approaches, and their most implementation in case of biofortification of plants. So if you are going to be implement plant genetic engineering approach for improving any desirable traits, there are some standard steps. Those are known, and you can see over here. You can see over here that first you have to be optimize the protocol of regeneration, then that you have to be select the explant from the plant source, then you have to be select the transgene or gene that you would like to be incorporate or edit or silence in the crop plants. Then after you have to be design the construct and binary vectors, and you have to be optimize the protocol of genetic transformation and their regeneration. And after generation of transgenic plants or regenerated genetically engineered plants, you have to go for molecular or biochemical analysis. So these steps are based on that several methodologies. There is a mixture of methodology, yeah, like you can see for that selection of explants and their regeneration, tissue culture is involved. Then when you are going to that selection of targets, then bioinformatics plays major role for that. Then designing of construct, their genetic transformation and generation of plants, biotechnology and tissue culture plays major role. And finally, when you generate the plants or transgenic events, then again bioinformatics, molecular and agronomic and biochemical analysis plays major role. However, in case of genome editing, bioinformatics plays very important role, especially in two stages. First one is that selection of target gene, and second one is that after generation of events, you have to be go for their analysis. So in this case, what happened that there are several tools have already been available where you can be designed by your guide RNA, and accordingly you have to be develop your vectors. And after that, that plants can be transformed and generate edited lines, and those edited lines, there is a editing or not, those lines are edited or not edited, then you will go to sequencing, and their sequence can be analyzed by using bioinformatics approaches. So in these approaches, I am working last 20 years. I started my journey in 2002 doing my PhD, and still I am working on all these aspects in depth about how to we can be implement that biotechnological tools to improve the crop plants. So during my PhD, my first major objective or achievement is the development of insect-resistant groundnut plants. You know that one of the major insect known as the tobacco cutworm or Spodoptera litera is a notorious pest in case of groundnut, and it is reported more than 70% yield loss in southern part of India, especially Karnataka. So considering those kind of issues, we have optimized the protocol of regeneration where groundnut seeds cotyledon, these are the cotyledons we had considered as a source of explants, and these de-embryonated cotyledons where embryos have been removed, utilized for the multiple shoot induction. You can see that in the proximal end of the groundnut cotyledons, we have generated 100 of shoots from the single explants, and this efficient protocol of regeneration we have used for genetic transformation for CRY1 EC gene. This CRY1 EC gene basically responsible to give protection against to this insect known as a Spodoptera litera. So after transformation and generation of transgenic plants, so you can see that our transgenic leaves, when we have performed insect bioassay, were completely, you can say it is not damaged as compared to the control plants. even in the late instar of this larvae stage. Even in that advanced stage, that insects are able to chew leaves, but not able to survive, because when we have used the promoter, those are induced when insect is going to be attack in the plant system. So that known as individual promoters, and we have attached this promoter along with our Criban EC gene. So this technology have been demonstrated in this case of groundnut, where we have protected against to this insect, and we had published several research papers during my PhD. And for this work, I got that Indian Science Congress Young Scientist Award in 2010 from our Honorable President, Dr. Abul Kalam Azad at that time. My second objective is to plant use for the vaccine production, because plant has so many advantages if you are going to be used as a bioreactor for vaccine production, because easily you can be multiplied. It is a low cost as compared to the cell line based system. There is a low possibility of human or animal pathogen contamination, and most importantly, it is like a eukaryotic post translational modifications. So the protein, whatever the end product is similar to that, it is required for eukaryotic systems. And if especially you consider that plant seeds, that has another several advantages, because it's a natural organ for protein and synthesis and storage. The protein content is high, so if you are expressing the protein or that any vaccine candidates, the content of protein will be high in the seeds of plants. It has low protease activity, so there will not be any degradation of protein those you are going to be expressed, and it can be stored long time. There is no need of cold chain kind of things, especially if you are considering plant seeds. And I considered groundnut seeds to develop the vaccine against to cholera as well as rabies glycoprotein. This fusion we have used and expressed in the tissue-specific manner in the groundnut seeds, where we have used seed-specific promoters, legumin promoter, and the fusion of cholera toxin B subunit along with rabies glycoprotein, we have transformed into the groundnut seeds. And after that, we have expressed this vaccine, and based on that, we estimated if that groundnut seeds can be grown in a 1 acre land, those are expressing 1% of total protein of this vaccine. So you are able to immunize million of pupils. That work was published in Biotechnology Advances 2009. So based on that, we have developed the groundnut seeds and performed some assays in Hyderabad against to rabies vaccine, rabies viruses, you can say. And we got that some kind of protections, and very recently you have seen that this technology where plants can be used as a bioreactor for vaccine production is showing very promising results, where COVID vaccine has been developed in tobacco plants, and Canada is the first country in the world those have been used this plant derived COVID-19 vaccine for adult immunizations. So based on my work on groundnut seeds to use as a vaccine production unit, I got another Young Scientist Award 2010, that is a BioSR BioAsia Innovation Award. So after that, in 2010, I joined NABI, that is a National Agri-Food Biotechnology Institute. It is located in Mohali, and the vision of our institute is very clear: to provide food and nutrition security to all through agri-food biotechnology research and innovations. My institute is basically working on 3R principles. First is research and development, second is resource generation, and third is reach to community. So considering the vision and the mandate of my institute, I started biotechnological approaches for biofortification. Biofortification means to enhance the nutrition quality of plant crops by using two approaches: gain of function, where we can be used that overexpression of a certain genes, or second one is a loss of function, where you can be use RNAi approach and also a genomatic approach. So my approach to enhance the nutrition quality in banana crops by considering both transgenic cisgenic approach as well as loss of function genome based genomatic based loss of function approach. So why we have to be consider plant genetic engineering for biofortification? And there is a three main reasons that we have to be work on biotechnological approaches to enhance the nutrition value of crop plants. First is population, second is hunger, third is inherent hunger. When you will see that population in the global status, that I have taken this information very recently in March 2024, you can see that the world population has already been reached more than 8 billions. So in the world, there is more than 8 Arab peoples are living, and India is the largest populated country in the world. Just China is behind to us, and that target initially was estimated to achieve in 2025, but that was the old prediction. And but we have now the largest population, and that has been reported in 2023 only. So in 2023 January, we have surpassed China and become most populated country in the world. So you have to know that how much food is required, how much land is required to provide sufficient and nutritious food of this population. That is one of the question. And another fact is about hunger. You can see that India positions very well regarding hunger index reported in 2020-22. Our neighboring countries like Bangladesh, Pakistan, Myanmar, Sri Lanka are in the far better as compared to the India. We are in the 107 positions out of, you can say that there is a country is considered like 150. So across world, it is reported more than 750 million people are hungry. Those are undernourished. That means those are not getting sufficient food in their diets. And India accounts for the highest, that is nearly 224 million undernourished people in any single country. So that is the biggest questions that we have to be address the problem of hunger, that is known as a zero hunger, given that targets to UN by 2020, 2030, that every country in the world has to be a zero hunger index. But it is looking difficult without implementing biotechnological approaches. So third one is about hidden hunger, also known as a micronutrient deficiency. That is a global problem and reported more than 2 billion people worldwide suffer from the hidden hunger or micronutrient deficiency. These micronutrients mostly covered vitamin A, vitamin C, iron, zinc, iodine, and folate. Basically, micronutrients constitute a small fraction of the balanced diet and plays essential role in human metabolic processes. These micronutrients you can get from diverse dietary sources, but country like India, there's most of the peoples are dependent on staple crops like rice, wheat, maize. Then it is very difficult to provide these nutritions from these particular staple crops. And these micronutrient deficiency also responsible for several abnormalities like impaired brain development, stunting growth, mortality, and also responsible for the diseases like diarrhea, malaria, and measles. So how we can we overcome these problems? There is a one solutions that can be governed by plant breeding approach, but plant breeding, as I mentioned, it is a limited that by several kind of things. First one is that there should be a variable that germplasm should be variable, and there is a desirable gene has to be present, especially for disease, pest resistance, or nutritional improvements. So these genes might be, or we can say most of the times are not available in the germplasm of related varieties. So breeding is not possible in these cases. So future breakthrough will depend on the creating additional variability and inflow of desirable genes from related or unrelated species. In this case, modern biotechnological approaches like genome editing and transgenics will come in the picture. So based on this background, I started work on biofortification of banana and by using genome editing and transgenic approach. So for that, we have chosen the target of provitamin A. It is also known as a beta carotene. And this beta carotene is a basically precursor of vitamin A, usually provided present in the plant crop systems. So in this case, for improvement of beta carotene, we have developed the collaboration of Australian University known as a QUT, Queensland University of Technology, Australia. And that project was supported by IREC and DBT, and the objective is very clear to develop disease resistance as well as biofortified crop plants. And we have different Indian partners in this collaborative project. BARC started working on iron biofortification, TN and UIH working on disease resistance, NRC working on trial sites, as well as NABI is working on the provitamin A biofortification. And also we are the national coordinator of this project. You can see in the picture our prime minister is taking so much interest in this project. In 2024 he also visited to our collaborator institute in QUT University, where that James Riddle is our collaborator. So this project is running in the first phase from November 2012 to February 2021, where we have developed the crop plants. Those are enriched with nutrition quality and extended next 3 years, where we have to be analyze those plants in open field conditions by following the DBT biosafety guidelines. Parallelly, we also started that our own project where we are explore the gene promoted combinations apart from that QT transfer technology. And after the screening of banana germplasm in India, we find out certain genes and promoters those might be responsible for enhancing or carotenoids contained in banana crop plants. So the overall objective of both the projects is to development of beta carotene enriched biofortified Indian banana. So why banana we have chosen in our study? Here you can see banana is a tropical food crops cultivated more than 130 countries. It is the second largest produced food just after the citrus. India is the largest producer of banana and contributing nearly 26% of the world banana productions. The important is that banana fruit is available throughout the year at the reasonable price and most suitable and choicest fruits for all age group, including infant, old people, as well as in adults. So why we have to work on biofortification using biotechnological approaches? Because banana is genetically a sterile crop. The genome is triploid, so breeding is not possible. So we are dependent to enhance the nutrition value in case of provitamin A or any genetic modification in banana plants. We have to be dependent on biotechnological approaches. So that's why we have chosen banana for biofortification of provitamin A using biotechnological approaches. So vitamin A deficiency, you have to know it is one of the serious issue. That's why we have considered to enhance the vitamin A provitamin A enhanced banana, and it is reported more than 6.5 lakh children die per year just because they are not getting sufficient vitamin A in their diet, and another more than 3.5 lakh children go permanently blind. Vitamin A deficiency is responsible for mortality, blindness, night blindness, impaired immune system development, and impaired brain development. In case of India, there is a reported in around 57% of children, those have less than 5 years of age, are deficient with vitamin A. And it is also reported more than 3 lakh children die per year just because they are not getting sufficient vitamin A in their diet. How you can get vitamin A you can get from two major sources. One is animal origin, second one is a plant origin. In animal origin you can get directly in the form of retinol or vitamin A, but if you are eating or you are vegetarian, then you have to get pigmented carotenoids, and these pigmented carotenoids are alpha carotene, beta carotene, or gamma carotene. Those are known as a provitamin A, and beta carotene has the highest activity to convert into the vitamin A in our body. Why we have to be dependent on plant source? Because approximately 375 million people worldwide and around 30% of Indian population is vegetarian. So we have to be considered plants as an important source to enhance the nutrition value. In case of banana, we have how much vitamin A we have to be increased. We have calculated based on the ICMR report. It is reported to get 1000 microgram vitamin A in our diet per day. The conversion ratio of beta carotene to vitamin A is 1:6. So if you are getting 6 molecules of beta carotene, then the body is converted into the 1 molecules of vitamin A. When we screen Indian banana Janpad, we found out that one of the most commonly cultivated variety, Grand 9, contain only 400 to 500 microgram per 100 gram of beta carotene in their pulp. So if we would like to get 100% RDA, that is a requirement of 6000 microgram, then we have to be increased nearly 15-fold high content of beta carotene in single banana. So that is our objective, and based on that we started our study. And these are the carotenoids biosynthesis pathway. You can see in this pathway that DXS is one of the genes that provides substrate flux in the carotenoids pathway. Subsequently, generalized diphosphate is converted into the lycopene by the successive enzymatic reactions, and this lycopene is diverted into the two direction. One is the lutein or alpha carotene biosynthesis, and second one is the beta carotene biosynthesis. Beta carotene our target molecules. So based on this carotenoids pathway, we designed our approach to use gain of function approach, where DXS gene and phytin synthase gene we are considered to overexpress in banana, so that these genes can provide substrate flux for the biosynthesis of beta carotene. And for loss of function based on CRISPR approach, we had considered PDS gene. This gene is basically we have used as a marker proof of concept that I will let you know in my research slides. And then lycopene epsilon cycle is this LCYA gene used to silence or edit so that lycopene resources can go to synthesize beta carotene only. Then BCH and CCD is considered to stop the degradation of beta carotene by using CRISPR approach. So this is the base of our study, and accordingly we started work. First, we optimized the protocol of regeneration to banana cultivars, Grand 9 and Dasthali. You can see that this is a very efficient protocol, and embryogenic cell suspension culture in liquid media we have used and maintained for a long time, and this culture basically utilized for the genetic transformation of banana. The protocol is very efficient, where we can generate thousands of plants from 1 ml of packed cell volume of embryogenic culture. So these are the steps. First, we usually use to perform in silico analysis. After identification of genes by using this in silico approach, we have to be characterized in bacterial systems. After characterization or functionality of that gene, we have to perform tissue culture transformation in banana cell suspension culture. After transformation, plants have been generated and shifted into the greenhouse and subsequently into the net house. We have very big net houses, as considered banana as a tall herbaceous plants. And after that, fruiting started and fruit has been taken to perform HPLC analysis. So based on that, this approach we have standardized in our laboratory, and the conditions for GM banana or the transgenic or genetic engineering approach, we followed DVT biosafety guidelines. So we had published several research papers regarding these. If somebody is very much interested to read about our research, they can go and read these publications. That plants and more than 500 events have been initially generated. Those were maintained initially in the greenhouses. Then those plants were transferred into the net houses and fruiting was started, and those fruits were analyzed in our laboratory. So I am starting from the first approach, genome editing. You can see, as I already mentioned, we have used that PDS gene as a proof of concept. Then lycopene epsilon cyclase gene has been edited. That work was published in Metabolic Engineering. Then second target is CCDs, is responsible for beta carotene degradation, edited in banana lines, and the work was published in Journal of Experimental Botany. We had also published two very good review articles very recently. If somebody would like to go in more details, they can read this review article also. So regarding first target PDS, that gene I already mentioned is responsible for the chlorophyll biosynthesis as well as beta carotene or carotenoids biosynthesis. If you edit or silence this gene, you will get albino phenotypes because chlorophyll biosynthesis has been stopped. So this gene is very commonly utilized as a marker for that RNAi or CRISPR optimization approach in any crop plants. So based on that, we had designed the CRISPR guide RNA and construct for genome editing in banana. And you can see after editing of banana embryogenic cell suspension culture, these embryos looks like albino phenotypes. There is no chlorophyll biosynthesis as compared to the control. That plants have been generated, those have been again shown albino phenotypes. And when we have taken these leaf tissues for DNA isolation and subsequent sequencing, we noted that there is a addition and deletion in the sequence, and based on that some stop codons has been generated in between, and based on the stop codon generated, that this gene is not going to be translated into the protein. So this gene is not synthesized at carotenoids as well as chlorophyll. Based on that, we had performed chlorophyll estimation as well as carotenoid estimation in leaf tissues, and you can see as compared to the control, there is a drastic degradation or a reduction of that carotenoids as well as chlorophyll. Based on this work, we had published this work in functional integrative genomics. This is the first report in the world that has been shown that gene editing is possible in banana. Then other target is lycopene epsilon cyclase. As I mentioned, it is diverting lycopene into the other sites, alpha carotene biosynthesis. But our target is beta carotene, so that we have edited this gene so that all resources can go to synthesize beta carotene. For that, we have designed the CRISPR gene construct that you can see over here. And that protocol for regeneration and transformation has been performed. That plants have been grown initially in greenhouse. After that, transferred into the net houses. Then fruiting came out. At that time, we had performed that sequencing analysis to make sure there is editing, and editing has been confirmed, where you can see that deletion and addition has been shown in this chromatogram. After sequencing, T7 endonuclease also have been performed to know that there is a homozygous or heterozygous editing in banana genome. When we perform the carotenoid estimation in fruit pulp, you can see that the beta carotene has been increased nearly six-fold content that is reflected into the dark yellow colors of fruits after cutting, because beta carotene has property to give that dark yellow color or golden color. So we had also performed some agronomical or agromorphological analysis in the net house grown plants as compared to the control, and that work was published in Metabolic Engineering, and that work was the first report in banana to improve the any nutritional traits by using genome editing approach. Then my second target is CCDs. It is responsible for the beta carotene degradation. As I mentioned, the CCD gene, 5 CCD genes have been found out in the banana genome. Those genes have been performed for HPLC, HPLC analysis and real-time expression analysis in two contrasting cultivar, Rastali and Nenden. Nenden cultivar is known for high beta carotene content, while Rastali known is for low beta carotene content. So after the screening of these two germplasms, we had performed expression analysis in different tissues of like leaf, pseudostem, root, finger, brackets, ripened pulp and unripened pulp. So after analysis of expression, these two contrasting cultivars, we find out that CCD4, CCD1 and CCD8A are present in abundance in fruit pulp of both cultivars, Rastali and Nenden. So based on this, 3 genes have been selected to analyze their expression in the fruit pulp of different stages like Rastali and Nenden. We had collected fruits from 3 weeks to up to the ripening stage. And when we had performed that HPLC analysis, you can see that content of beta carotene or carotenoid alpha carotene is very less in case of Rusthalia at various developmental stages of fruits. While in case of Nandan, you can see that the carotenoid content has been very high as compared to the Rasthali. It is a, or when we had performed that what we can say the expression study in these developmental stages of these three CCDs gene, we have noticed that CCD4 is showing very high expression at 6-week intervals of in Rasthali fruits. So it is hinted that CCD4 might be the potential target for beta carotene degradation in Rasthali cultivar. So based on that, we had cloned this CCD4 Rasthali and performed bacterial complementation assay. It shows that beta carotene is degraded. And subsequently, we also overexpress this gene in Rhodosis thaliana, and again in the seeds of Rhodosis thaliana lines also shows that beta carotene is degraded. So based on that, it is sure that this beta carotene, this CCD4 from Rustali has activity that is negatively regulated the beta carotene biosynthesis. We have also performed that GCMS analysis by for that enzymatic assays, and this CCD genes purified from Rustali and perform enzymatic assays with different substrates, and we find out that CCD4 is responsible for the degradation of beta carotene 10 and 10-FO beta carotenol. So based on that, CCD4 gene has been designed for the transient expression and as well as that transient expression for particle bombardment, because this gene construct has no left-right border as well as no selection marker, so that this construct has been transiently expressed in the banana protoplast. And after that, we had generated certain lines. Similarly, we had also performed particle bombardment assays of this CCD4 constructs, and we had generated number of lines. Those lines were screened by sequencing, and we find out 2 lines have been shown addition or deletions of the what we can say mutations, and we have also confirmed there is no integration of CCD Cas9 gene in the genome. So these lines have been considered as a non-transgenic banana lines. That work was highlighted by DBT in their website in 2020-22 when we published this work. And we also analyze that these carotenoids in leaf tissues as well as the root tissues of edited lines, and it shows that there is a increase of beta carotene 2 to 3 fold as compared to the control. So this work was published in Journal of Experimental Botany in 2022. And plants I have reached in the fruiting stage. Now we are going to be analyze these plants. And our second objective is the overexpression study. DXS gene we had considered that might be responsible for the providing substrate flux in the carotenoids pathway, so that beta carotene has been increased. And here you can see that Rasthali and Derin cultivars we had considered to see that which CCDs are responsible. DXS gene is responsible for that high beta carotene depositions. You can see DXS2 is showing very high expression in case of Nandan cultivar. This cultivar is the same cultivar, those are containing high amount of beta carotene. So based on that, we had cloned these DXS genes, and we have again noticed that DXS2 is showing very high beta carotene depositions in bacterial systems. We had designed the construct for overexpression, and we had generated that lines of overexpression. And when we cut the fruits, you can see it is completely looking like a golden in color, and it shows that very high depositions of beta carotene, that is compared to control, that is 20 to 38 fold high beta carotene as compared to the beta control foods. You can see that color is very dark in yellow, and that's why it's known as a golden banana as compared to the control normal banana. We also performed that Keiko cell line based that biology study, where we have taken that banana fruits, and we have performed that bioavailability study in cell line based systems. And after that, we have concluded that single banana is sufficient to provide 100% recommended dietary alliance. And now we are in the second phase of this project. In the second phase, we are performing multi-location event selection trials to see the agronomical performance of our biofortified lines. And for that, we have considered 5 trials at National Research Center Banana, that is in Tirchi, Tamil Nadu Agriculture University in Coimbatore, Navsari Agriculture University in Gujarat, Assam Agriculture University in Assam, as well as in Nabi. These 5 locations we are performing event selection trials, and BCIL is supporting us for that compliance and field trials and data generation by following the DBT biosafety guidelines. We have got the all clearance, regulatory clearance like RCGM, GSA, and Institutional Biosafety Committee. And based on that, we got permit letters to perform our event selection trials. So our journey started in 2013-14 from the optimization of tissue culture protocol. Subsequently, we have designed the gene construct and performed transformation. After performance of transformation, we had generated our lines, and those lines we have analyzed, find out that some of the lines showing very high depositions of beta carotene, and now we are performing event selection trials in different places of India. So very recently, it is showing that in the articles published in Times of India that after mustard, genetically engineered banana, rubber, and maize on the way. So we are on the way, and most probably in the next 3 to 5 years, we are able to reach for the commercial cultivations of banana. Those are enriching with beta carotene or carotenoids content. So you can see the current trial stages status in different places of India. In Nabi, you can see that trials is going in the open field conditions. Navsari Agriculture University, Gujarat, plants are in the vegetative stages. The Tamil Nadu Agriculture University, they are still in the greenhouse stage. Assam Agriculture University and Assam plants are also in the juvenile stage, and NRCB plants have been reached at the fruiting stage. So in the next 6 months, we have concluded our that agronomical performance in different locations in India, and accordingly, we conclude that the most promising lines will be considered for further environmental reviews. So in this layman language, you can see that this is a normal ordinary banana available in the market. It is saying that, oh, if you would like to get 100% idea, then you have to eat number of fruits. But in case of our golden banana, it is saying, I am single sufficient to provide sufficient recommended dietary allowance for vitamin A. So this is just a pictorial representation for laymen so that they can understand the promiseness of this technology. And we have also given the interview in that Pallab Bhavla very recently. If somebody would like to go in details and see how we had performed the experiments in the laboratory, and this laboratory outcomes reached into the field conditions, they can see this interview and get more awareness about our work. So I am really thankful to my current lab members, especially my old team members, and those most of them have already been settled well in the different countries. Also, though most of them have got permanent jobs in India, and without their hard work, it is not possible to reach at this stage where we can say confidently that by using biotechnological approaches, we have product in our hand that might be going to be released very soon for that commercial cultivations. So with this, I would like to say my sincere thanks to our collaborator, QOT, NRCB, BARC, TNAU, IHR, NAU, AU, and NAU, our research team. Those are working day and night to reach at this stage. My funding agency, BIREC, as well as Department of Biotechnology, my all executive directors of NAVI who have supported from the inception of this project. So with this, I would like to conclude my presentation. Thank you so much.
Thank you, sir. Thank you very much, sir, for very interesting lecture on biofortification. And that also you have taken the good initiative for the malnutrition individual by changing the classical method in the new technology. Thank you so much, sir.
Thank you so much.
So question answer session we will discuss later.
Sure. No, sure. Thank you so much.
Thank you, sir. So now you unshare your screen, please.
Sure. Just a minute.
Now I would like to call our next speaker, Dr. Rajesh Patel. He is a professor, Department of Biosciences, VNSGU, Surat. Dr. Patel completed his doctorate degree in the field of microbiology in the year of 2003, and since then, he has been imparting his valuable knowledge to the students with teaching experience of over 21 years. He has also been guiding scholars for their research work. Dr. Patel has feathered of several publications in his hat of renowned national as well as international journals that include the 45 research papers, 4 conference proceedings, 5 books, and the 4 book chapters. He has been working in the area of microbial diversity and its biotechnological exploration from various extreme environment of Gujarat, including saline desert and hot spring. He submitted 4216S rRNA gene sequence to NCBI and completed whole genome annotation for several bacterial species, isolated and studies more than 30 actinomycetes from saline desert environment. For anti-MRSA activity, isolated and studies more than 20 endophytic isolates for biofertilizer application from saline desert. Patel sir, I welcome you.
Am I audible?
Yes, sir, you are audible.
So my screen is visible.
Yes, sir, your screen is visible.
So good afternoon, everybody. And first of all, I would like to thank the Amity University to give me the opportunity to have the interaction with you all. So basically, I was listening to three lectures which are gone earlier to that. And more or less, we are trying to manipulate and we are trying to convert the nature in our favor only. And in the race of this favoring the nature and changing the nature only, we have changed our soil and our environment only. So my topic is about unveiling the secrets of the nature, metagenomics and metabolic exploration of the natural farming input for the sustainable agriculture practice. So basically, the problems and issue which is leading us to go for the natural farming includes the soil degradation. So we have gone for the very heavy farming practice. We have just removed the forest. We have added the fertilizer, pesticides as per our need, and this has already increased our production of the crops. But at the same time, that is also lead to the degradation of the soil. Similar to that, as we are observing that whatever our crop production is there, that is highly depending upon the different kind of the chemicals, and this is leading to the unsustainable practice. The heavy amount of the chemicals that has lead to the food safety concerns to us also, and that is also reduced the farm profitability. And due to all only, now we are just again going back to our natural way of doing the farming, and that is one way of natural farming. This is also known as the zero budget natural farming. The basic principle of this is we are just eliminating the need of the external input. So whatever is present in the site, the zero budget natural farming is promoting the use of that. So that can be improved the maintain the soil fertility. That will encourage the beneficial microbial activity in the soils, and that will also promote the plant growth and pest resistance. And the key principle in this is no chemical fertilizer or pesticide, and we have to believe or we have to use all the natural methods. We can have the different kind of the natural farming input, but broadly suppose you are observing the natural farming input, and that is the locally sourced material that will avoid all the external inputs, and that is relatively inexpensive, easy to perform in the case of the farm. But at the same time, this natural farming can be a more labor intensive, and at the same time, that is also taking a very long time to see the results. So that is the reason that we are not that much exploring the natural farming input in comparison to the other farming practice which done conventionally. So normally we all aware about the composting, made up of the compost. So whatever the organic waste material is there, that is converted into the compost. But in this natural farming input, along with the composting process, we are also adding the cow dung and cow urine. And due to that only, the different preparations like the jivamrut, kanjivamrut, and other kind of the things are there. And that will be having the addition of the urea. And this urea will be toxic for the harmful organism. At the same time, that will also have the very bad smell and pungent nature. And that will be also repelling the different kind of the pest. So that creates a combination. And along with that, to increase the activity of that, we are also adding the neem and other kind of the plant material which is available locally. So that will also work as an insecticide. This will be also leading to the biofertilizer, but this biofertilizer would be a live organism, and this organism would be helping the recycling of the soil nutrients. So that will be not focusing only on the nitrogen, phosphorus, like normally other organic fertilizer is done, but this is normally utilizing the live microorganism, and that will overall increase the soil fertility, and that will help us to fix the nitrogen from the air, solubilize the phosphorus, and so many other things whichever is required for the plant growth. Along with that, we have seen that we are using a lot of weedicide and other. So currently people are using the plant covering method. So they will just put the sheet of the plastic just to avoid the growth of the weed. But in this natural farming, they're just using the intercrop. And whatever the other kind of the weeds are there, that is inhibited by addition of the other crop between the two lines of the main crop. So that is the way you can just go for the cover farm. So these are the some of the very common practices that will include the Bijamrut, Jivamrut, Achadan, Botaps, and plant protection. So due to the limitation of the time, we'll be not discussing all of this, but we'll be just going through few of the things. So as initially, when the plant is going for the seedling, we are using the Bijamrut. So this Bijamrut is a mixture of the cow dung, cow urine, water, and that will also have the small amount of the soil from that. And that is basically acting like a soil treatment. This is the entire preparation by which it is done. So you have to mix all these things, and after that short level of the fermentation, we are going for the seed treatment. The 20 liters of the Bijamrut is sufficient to treat the 100 kg of the seed. And if your seeds are there in the form of the saplings, then you can also use it in the rhizome stage root and others. And with this way, you can just have the addition of the Bijamrut in this. This is the nutrient content which is normally present in the Bijamrut. So that will have the total nitrogen, phosphorus, potassium, zinc, copper, iron, manganese. And this will be just supplementing all this mineral element, micro element, and macro element for the rapid settling development of that. And suppose you are checking the microbial population in the Bijamrut, then the Bijamrut you can see that that is having the short level of the fermentation. And after the fermentation, it will also have the different levels of the beneficial bacteria, fungi, actinomycete, nitrogen fixers, and phosphosolubilizer. So instead of using one or two organisms, this will be a mixture of all the beneficial organisms that will be enhancing the seedling growth. So this Jivamrut is basically a fermented liquid organic matter. It will promote the immense kind of the biological activity in the soil and make the nutrient level available to the crop. So it is a rich source of the beneficial microflora. That beneficial microorganism would be having the trait as we have discussed earlier. So the Bijamrut is basically applied during the seedling, but after the getting the proper growth, people are using the Jivamrut. The preparation of the Jivamrut is also similar to this only, but that would be adding the jaggery, and along with that, that would be also having the additional of the pulse flour. Along with that, that will have the cow urine, cow dung, water, and even the local soil would be added to this. And this will lead to the fermentation approach. And after that, again, you can see that you can have the high CFU, and the CFU would be increasing in almost for the bacteria, fungi, actinomycete, nitrogen fertilizers, and even the phosphorus solubilizer. So that is the days after preparation, and that way that will become a live preparation, and that would be normally spreaded in the field. So that can be done by the entire spray method, or you can also use the drip irrigation method for that also. And after this method of the application of the Jivamrut, you can see that that is increasing the growth of that, and that is a normal thing which is done along with that. This Jivamrut cannot be stored for a longer time, so to avoid that, only people are also producing the Ganjivamrut. And the Ganjivamrut, the solid part of that that is done. Similar to that, to inhibit the pest, people are using the Nemastra, so that will be addition of the neem in this entire preparation. And like that, only so many different kind of the things are done. So in the Brahmastra, some other kind of the alkaloid containing plant materials are added. So these are the few preparation along with that only. Agniastra, these are the few example of that. And even people are developing the Dasparni extract. So instead of only 2-3 plant material, they are using the 10 different kind of the plants for that. And that is again the preparation and how it can be utilized for the further purpose. In this, normally we are taking that this is the entire process which is depending upon the cow. And in India, currently people are using this cow-based therapy, but the people are using the cow breed, especially the Gir cow for this. And we are focusing on the two other breeds of that. And one Dangi cow is there that is normally found in the South Gujarat region. And this is the cow which can be just stocked available. It is of course giving the lesser amount of the milk, but it can survive in the such condition where other cow cannot be kept. So this is a new thing about the our project, that instead of working on the Gir cow and other popular breed, we are working on the less explored Dangi cow. And this Dangi cow would be also taking the same kind of the inputs we are using for that. We will be using the cow and other. And especially in the South Gujarat region, the Dang region is declared as a natural farming district by the government. So we are also analyzing the soil condition in the different part. And in this only we can see that we have taken few of the sites where the commercial or the conventional practice is done, and these are the green part where normally the natural farming input is done. So you can see that normally all the nutrients and the availability of the nutrients is slowly getting increased by use of the natural farming input. Similar to that, this is the lab preparation of the Jivamrut, because whatever the Jivamrut people are producing in the field, that is more or less done by the traditional method, but to standardize that only we have gone for the standard method of this, and we have gone for the physical chemical analysis of this in our lab, and from that only after that we have gone for the microbial and genomic analysis of that, because as we have seen earlier that this can be a live biofertilizer, and during the fermentation it is going through the succession of the different communities. Whatever the initial communities are there, that is going to be changed after the fermentation, and even the level of that is getting changed. So we've gone for the isolation of the microbes from that, and at the same time even we've gone for the DNA isolation, metagenome isolation from that, and we have also gone for the metabolomic analysis of the Surti buffalo along with the Dangi cow. So the Dangi cow is a cow which is utilized in the Dang region, but along with that in the Surat, normally for the milk production, people are using the Surti buffalo, and this Surti buffalo is not explored for that, because normally the community which is keeping the Surti buffalo, they are associated with the milk production, but they are not associated much with the farming practices. So in that we have just explored the possibility that what kind of the metabolomics and metagenomics differences we can have when we are using the buffalo and the cow in the preparation of the Jiva. So we've gone for the sample preparation. I'm not discussing much about that, but basic process of the metagenome isolation was done. And from this, along with that, to analyze that only, we have also gone for the GCMS analysis of the natural input preparation. And whatever the results you are getting from this cow Jivamrut, the 0 day, and again after that 8 day, we have gone for that, and we have compared the both of that. And we will find out that some of the compounds are getting disappeared, which are present in the earlier after the fermentation, and some of the new compounds are coming after the 8 day of the preparation. In same way, it is also compared for the cow and buffalo for both of this. So these are the few important compounds that is remain and that is present after the 8th day of that. And these compounds would be also, some of them are also found in the case of the buffaloes, right? So these are the compounds which are the key metabolites, you can tell, that is increasing the crop productivity and even the soil productivity in that one. Along with that, when we are going for the metagenomic analysis, as we have seen that both of them, they have the human flora, and the rumen flora is less explored compared to the other, and that is naturally having the lot of uncultivable microorganism also. So even though we are just finding out so many different methods, but we are not sure that all the microorganisms which are present in the cow dung, we are able to transfer them in the lab condition only, and for that reason only, we have gone for this geometry preparation as we have discussed earlier. And from that only, we've gone for the metagenome analysis. We've gone for the shotgun genome analysis, and this is the overall comparison of the shotgun genome analysis data. So we have taken the cow dung, because basically the major source of the microorganisms are the cow dung. And after preparation of the cow Jivamrut on the 8th day, what are the changes that we have observed? So normally you can see that the number of bacteria is getting increased. At the same time, you can see that the archaea is getting decreased. Fungal species are having the lesser change, and even we can see that the number of the viruses are also remaining change in both of this. So that way, overall, you can see that the entire microbial community at the level of the bacteria, archaea, fungi, and virus, that is totally getting changed after the fermentation of the 8th day. So these are the broad picture. At the dominant level, you can see that overall, whenever we are going for the cow dung and buffalo dung, and after that, when we are taking the cow geomrut and buffalo geomrut, almost you can see that the taxonomy is giving some kind of the elevated level. So you can see that the increase of that. The same way, you can also see that at the kingdom level also, you can see that the different bacteria are getting changed, and even in the other also. So for the better explanation, we are just going for this only. Is that in that, but after the 8th day of the geometry preparation, the diversity is getting less, but specifically the lactobacillus and other kind of the probiotic bacteria, that is going to be increased. Same way, suppose you are observing in the case of the buffalo dung, and in the buffalo dung also the significant amount of the lactobacillus is getting increased, and same way the diversity is getting decreased. So this is giving us the indication that at the genus level also some of the harmful bacteria or some of the non-productive bacteria are getting decreased, and whatever the material is that is getting recycled and that is getting utilized by the lactobacillus species and other. So this is about the fungal. In the fungal also you can see that the shifting is done, the diversity is getting decreased, but specifically the beneficial microflora of the fungal that is getting to be increased in this. Same way, suppose you are comparing the viral diversity, then in the viral diversity also we can see the quite shift in this, and a lot of viruses are getting disappeared, and only few of the viruses are getting increased after the treatment of this. Same way in the archaeal diversity also you can see that especially archaeal diversity you have to see with the level of this methanogens and other, because normally whenever this fertilization process is going on, that will lead to the formation of the methanogens, and these methanogens are basically responsible for creating the greenhouse gas also. So typically we are also planning to go for analyzing the harmful effect of this fermentation process also, and we'll be planning to go for the microbiome engineering in such a way that along with the methanogenic bacteria, there will be also the presence of the methyl utilizing bacteria and MICP bacteria. So that will also utilize this greenhouse gases and give a sustainable practice in this formation of the Jivamrut also. Along with that, we have gone for the analysis of the WGS data from that. Whatever the taxonomic profile is there, that overall we have analyzed even with the help of the 16S RRN, because whatever the observation we have seen earlier, that was utilizing the shotgun approach. But suppose we are taking the other part, then we can also analyze them with the help of the BBBRC. And in that also more or less we are getting a similar kind of the results. So these are the overall part of that. And suppose we are taking the core biome of the cow dung and Jivamrut at a genus level, then we can find out that even both the species are different, but even they are having some kind of these species which are common. And specifically the azoospermum and some of the other bacteria which are quite beneficial for the plant growth that is present in both of this. So that way there are much differences, but at the same time, the presence of the beneficial bacteria is present in both the cow dung and even the Jivamrut preparation. Similarly to this buffalo dung and even Jivamrut preparation, so you can see that whatever the core biome is there, in that also that is present, but in the Jivamrut preparation that is going to be increased. So that you can see by this, that when we are going for the core biome of all the sample at the Jivamrut, then we can find out that specifically this lactobacillus species are getting increased. So what is done inside that is, out of the total biome, whatever is present, the beneficial microorganisms that are getting magnified during the process of this Jivamrut preparation. Similar to that, even the core biome of the buffalo and cow dung samples are done. So in that also we are getting the similar kind of the increment of the beneficial part. Suppose you are comparing the cow biome, buffalo and cow Jivamrut samples, then we can see that a lot of bacteria which are present in a beneficial level that is becoming common in both of these. So that way, whatever the commercial breed of the Surti buffalo is there, that can be used alone, or that can be also used along with the cow preparation together. So that can also can give similar kind of the results. So that are the results we are planning for. Now, depending upon the organism which you have seen, we have also gone for the plant growth promoting function of that, depending upon the phenotypic trait, with the help of the Magen analysis. So that is given that whatever the cow dung and buffalo dung is there, that is initially having the lesser kind of the trait, but after the preparation of this, the different traits like the biofertilizer, bioremediation, phytohormones, stress control, plant immune response stimulation, colonizing plant system, competitive exclusion. So this all kind of the trait containing bacteria are getting increased. So that way it is not only giving the nutrition, but at the same time that is also helping the plant to resist the stress and even for the plant protection against the adverse conditions. This is the comparison of the plant growth promoting functions in both of this. So these are the cow dung and cow Jivamrut. So you can see that in the cow Jivamrut preparation, whatever the level of the earlier is there, that is getting magnified. So that is getting increased. Same way, even in the case of the buffalo also, the fermentation process is normally increasing the entire part which is beneficial for the plant. And similar to that, suppose you are comparing the cow Jivamrut and buffalo Jivamrut, then you can find out that some kind of the trend is common between both of this. So that is the way that even at a genomics level also, this entire metagenome is shifted in such a way that whatever the beneficial genomes are there, that is beneficial genes are there, that is getting overexpressed in this preparation of that. For analyzing that, we have utilized the biobacteria workflow, and even we have gone for the pathway enrichment, and we have seen that pathway. So these are the detected pathway in the cow dung. So we had around 1400 pathway in the cow dung, but when we have gone for the cow Jivamrut, almost 2900 pathways are getting operated. So that gives the indication that whatever the metabolic pathways are there, even though the number of organisms is getting decreased, but this entire metabolic diversity is getting increased. And similar to that, even in the case of the buffalo dung and even in the buffalo Jivamrut, we have seen that almost 2700 different kind of the metabolic pathways are getting enriched. And this is giving us the indication that a lot of plant beneficial metabolites are synthesized, and that is normally provided with the preparation of the Jivamrut when we are applying in the field. These are the detected pathways in the cow dung and cow and buffalo dung and Jivamrut. So you can see that almost we are getting the increased number of the detected pathway after the preparation of the Jivamrut for both of this. Besides that, even we have gone for the WGS2 based gene prediction and annotation. So whatever the beneficial genes are there, that we have just gone for the locating them, and whatever the genes are there that is related with the different kind of the plant growth promoting trait. So this is the annotation of the gene and their corresponding pathways. So we can see that different kind of the pathways are getting increased and different kind of the genes are getting increased, and in that way you can have the increase in this. So this is the differential express of the detected in the cow sample. So you can see that in the cow dung, the total gene were around 4,000, and that is increased in the cow Jivamrut, that is around 6,000. And similar to that, in the genes which are getting regulated in the cow Jivamrut, so we are getting upregulation of around 3,400 genes and downregulation of around 2,600 genes only. So that way, you can see that even there is a downregulation and upregulation, and this upregulation is supporting the plant. After that, even we will just fish out all the genes, and even we try to find out that what are the genes which are differentially expressed and what are the roles of that. So you can see that this all are the beneficial genes and which are getting increased, which is normally present at a zero level in the cow dung, but after the preparation of the Jivamrut, the numbers are getting increased. So whatever is shown over here in a red color, that is giving the indication that that is getting increased. So that way, even at a Jivamrut preparation, at the gene level also, we are increasing the number of the plant beneficial genes. Similar to that, this is given for the buffalo genome, and in that also we can see the increment of the entire part. So that is the way that instead of in the organic farming, normally we are just supplementing the cow dung or the buffalo dung, but that will give the limited kind of the effect. But when we are going for such kind of the fermented preparation, that will give the additional benefits at both of this. So these are the differential expression between both of this. These are the few of the PGPR genes which we have just fish out from that, and this is the function of the PGPR, and these are the example on which it is acting. So these are the phytohormones, that can be also the nutrient transporter gene, that can be also stress tolerance genes, defense signaling gene, and even the can be a gene for the nitrogen fixation and so these are the some of the genes which are increased, and that is having the role with reference to the stress tolerance genes. So the stress tolerance genes can be also giving the better amount of the stress tolerance to the plant. So that will give the systematic induced resistance to this stress. Similar to that, even biocontrol genes are also getting reported. So these are the pathogenicity related gene, defense related, plant defense, and so many others are reported in a similar way. From this, we also created the database of that, and still right now it is in preparation, because in our project we are not comparing only these two breeds, but we are also comparing the other breeds of the Gujarat, and slowly we'll be coming out with the full detail of this entire database, where you can just find out the genes and their potential role and then detected from this part analysis. So that way, in future, we can have the much more part of this. So basically, the entire strategy is targeting the precise natural farming technique. So basically, natural farming input, which is done right now, that is a crude way of doing that, and that is done with the natural and traditional methods only. What we are planning to do in the next part is we'll be develop the precise natural farming techniques depending upon the metabolomics and metagenomics information. We'll be also going for the microbiome engineering, and from that only will be design a strategy by which we can maximize the benefit the soil biodata, and we can increase the plant metabolism in a better way. So this metagenomics and metabolomics can unlock the secret of the natural farming, and that will enhance the understanding and that will lead to the targeted strategy for the sustainable agriculture. Because in the earlier lecture, we have seen that the people are working manipulating the plant, but we are not having any other mode to manipulating the soil and the environment in which all such good plants are supposed to survive. So our basic role is to maintain a good environment in which the forthcoming new genetically modified plant and even our natural plant can survive better, and we can have a sustainable agriculture practice and good food and good health everywhere. So thank you very much from my side.
Thank you, Dr. Patel. Thank you very much. This is Dr. Vinay Dwivedi, Director AIB. Thanks for your valuable talk. And after the last session, there will be a panel discussion. So we will invite you again for the panel discussion and the group photograph.
Thank you, sir.
Thank you.
Thank you, sir. Yes, sir. Now, I would like to call Dr. Bhaskar Venkadasamy, faculty member, Savita Dental College and Hospital, Savita Institute of Medical and Technical Sciences, Chennai, Tamil Nadu. Dr. Venkadaswamy has made significant strides in advancing knowledge and innovation in dentistry. His research interests encompass a wide range of topics, reflecting his multifaceted approach to addressing key challenges in the oral healthcare. Some of the areas in which Dr. Venkateswamy has made notable contribution include oral health, epidemiology, dental material sciences, implant dentistry, and oral cancer research. Dr. Bhaskar dedication to research excellence and his commitment to improving the oral health outcomes make him a highly respected and sought after expert in the field of dentistry. His participation and international conferences serve to enrich the academic discourse and foster collaboration among researchers and practitioners worldwide. I welcome Dr. Bhaskar to deliver his lecture. Sir, please.
Thank you very much, ma'am, for your kind introduction. Can you hear me?
Yes, sir.
Okay.
We are able to hear you, sir. Also,
Okay.
we can see your slides, sir.
Yeah, okay. Thank you very much, ma'am, for your kind introduction. Like, a very good evening to one and all. And I would like to convey my thanks to Professor Raghavendra Kumar Misra for his inviting me to the international conference held at Amity. So today I'm going to present on the on the lecture relevant to glucosinolates, sulfur-based compounds, their synthesis and their application perspectives. So I will be finishing the presentation within like 10 or 20 minutes. Okay. So we can see what about the glucosinolates? Glucosinolates are sulfur and nitrogen containing secondary metabolites, and their presence are limited to the order Caprales, which belongs to Brassicaceae family, and we know that which includes the model plant called Arabidopsis. Okay. So far, there are more than 130 glucosinolates that have been reported structurally, and it is mainly due to the different biosynthetic pattern that occurs due to secondary side chain modification in the last step of glucosinolate biosynthesis. Most of these glucosinolates has been present in brassicaceae vegetables, as I said in the earlier, like Brussels sprouts, cabbage, cauliflower, broccoli, mustard, turnip, etc. So among the different glucosinolate so far exist in the nature, the main highly studied glucosinolate is glucoraphanin, which produces sulforaphane. That is the derivative of glucoraphanin, which is one of the highly due to the presence of potent antioxidant inducing capacity, because this sulforaphane is able to induce phase 2 enzymes, which is responsible for the conversion of toxic metabolites. So next we see the biosynthesis. So these are the vegetables which contains higher amount of glucosinolates that we commonly consumed in routine days. So biosynthesis of glucosinolates has been taken place by three different steps. First one is side chain elongation. The second one is core structure formation. After side chain elongation of the amino acids will undergo core structure formation. Then the core structure will undergo numerous secondary side chain modifications. And the secondary side chain modifications are essential for the production of huge number of different types of glucosinolates. And based on the amino acids utilized for the biosynthesis of glucosinolates, they were classified into three different types. The first one is aliphatic glucosinolates because they are synthesizing from cysteine, methionine, valine, leucine. Similarly, the tryptophan, the indolic group of amino acids, can be used for the synthesis of indole-specific glucosinolates. So the phenylalanine amino acid can be used for the synthesis of benzoic glucosinolates. So apart from the synthesis, the regulation of glucosinolates are strictly controlled by a complex regulatory network system, that is R2, R3 MIB transcription factor regulatory network system. In Arabidopsis, the aliphatic glucosinolates can be positively regulated by three different types of MIB transcription factors, that is MIB 28, which is a major regulator, then MIB 29 and 76, which are the minor regulator of aliphatic glucosinolates. So the next one is indolic glucosinolates, which are positively controlled by three different types of MIB transcription factors, that is MIB 34, and another one is 51, 122. So this slide shows about the simple structure of glucosinolates. So you see it contains glucose, nitrogen, and sulfur compounds. So this is the schematic diagrammatic representation of biosynthesis of glucosinolates in plant system. So you see the left one, it starts from methionine and it produces the aliphatic glucosinolates. The center one, it starts from tryptophan, which produces indolic glucosinolates. And the third one is phenylalanine, which undergoes side chain elongation and then which forms co-structure formation. Finally, it produces benzyl glucosinolates. So there are three steps. Amino acids will undergo side chain elongation. After that, it will be converted to form co-structure formation. Finally, it undergoes numerous secondary side chain modification for the production of diverse number of glucosinolates. Generally, these intact glucosinolates are inactive in nature because they doesn't have any biological functions. However, due to the presence of action of myrosinases, the myrosinases are thioglucosidase enzymes which are exist nearby side of glucosinolates in the tissues of plants. Upon the action of this myrosinases, the glucosinolates will be hydrolyzed to produce numerous number of secondary active compounds. And also, when we take these glucosinolate containing brassica vegetables, like after digestion, it will be hydrolyzed by the intestinal microflora to produce numerous biologically active compounds like isothiocyanates, thiocyanates, nitriles, etc. So you see, these are the brassica vegetables contain higher amount of glucosinolates. How these glucosinolates are gets hydrolyzed to produce active biological compounds. You see, this is the structure of glucosinolates. There is a glucose moiety. So upon the action of intestinal bacteria or by the action of myrosinases, the glucose moiety will be removed from the glucosinolate. In turn, it produces numerous active isothiocyanate and nitriles, thiocyanates, etc. And these biologically active compounds have numerous biological functions like cancer prevention, and also in plant system, it gives protection against diseases and insect resistance. So these are the intact glucosylate as well as their derivatives and their natural resources. For example, the first two, cinegreen and glucoraphanin, are intact aliphatic glucosinolate. The cinegreen will undergo hydrolysis to produce allyl isothiocyanate, which are commonly present in the mustard. The second one is the most highly studied glucosinolate. That one is glucoraphanin, which produces sulforaphane. The sulforaphane is a potent antioxidant-inducing molecule, which is highly present in the broccoli. So the next, third and fourth one is glucotrapalin, which produces benzyl isothiocyanate, which is present in garden cress, Indian mustard, papaya. So the fourth one is gluconastatin, which is benzyl glucosinolate, which can produce phenethyl isothiocyanate, also known as PEITC, which is present in turnip, watercress, etc. So the final one is glucobrasicin, which is an indole glucosinolate, and which produces indole 3 carbinol and 3,3-diindolylmethane, methane, that is DIM. So which are present in the brassica crops. So now we can see what are the application perspectives of these glucosinolate hydrolysis products. So apart from their there's certain biological functions, like it involves in plant defense mechanism, oxygen homeostasis, disease and insect resistance. Apart from that, in humans, it gives protection against different types of cancer, and it inhibits the growth of Helicobacter pylori, a causative agent of peptic ulcer. And how it is inhibiting the cancer or other diseases in the human system is like, so this sulforaphane can induce the NRF2 pathway. So in turn, it induces various phase 2, phase 2 metabolic enzymes like quinone reductase and glutathione transferase. These phase 2 enzymes convert the carcinogenic metabolites into normal metabolites, so that it governs protection against different types of disease. It also enhancing the tumor suppressor proteins, possibly via inhibitory effects on histone deacetylase. So that means it's stopping the silencing of tumor suppressor proteins. Finally, the sulforaphane can also give protection against heart from inflammation disorders. So our aim was to increase the genetic enhancement of aliphatic glucosinolates in Brassica repa, and we use two different types of approaches, like by modifying the genetic the biosynthetic genes. The second one is regulatory genes. So in the first approach, we were trying to increase the specific glucosinolate, that is glucoraphanin, which in turn produces sulforaphane. Sulforaphane is a potent antioxidant molecule, so that we were trying to increase the glucoraphanin in Brassica repa. Due to the Brassica repa contains very least amount of like this particular glucoraphanin content. So we were trying to increase the glucoraphanin in Brassica repa by genetically modifying the three different biosynthetic genes. As I said, the CYP79F1, which is involved in the formation of aldoxane. The next one is AOP2, which is involved in the conversion of glucoraphanin, the compound which we are looking at will be converted into alkynyl form. So this AOP2 considered as a feedback regulator of the pathway. The third one is broccoli AOP2. Broccoli usually contains higher amount of this sulforaphane, that is due to the mutant version of this particular AOP2 zone. So we were trying to know whether this particular mutant AOP2 will have some functions on this sulforaphane accumulation in Brassica repa. We were overexpress this particular broccoli AOP2 in Brassica repa, and we were trying to overexpress CYP79F1. Similarly, we were trying to knock down the expression of AOP2. So we made four single gene constructs, which include CYP79F1 and broccoli AOP2 overexpression line and RNAi line for AOP2. Similarly, on the combination of these four single gene constructs, we made four double gene constructs, and we started plant transformation individually for each constructs. And the main issue with this particular Brassica repa is recalcitrancy, and it's like the transformation efficiency is very low when compared with other Brassica species. Brassica repa has least transformation efficiency. So we were able to standardize the transformation protocol, and then we started plant transformation, and we could able to get transgenics for all the lines, and it was confirmed with PCR and RT-PCR followed by southern blotting. And interestingly, some of the genes have showed different types of phenotypic modifications, especially in the broccoli AOP2. So this is the second concept, like molecular characterization of regulatory genes, which is involved in the regulation of aliphatic glucosinolates in Brassica repa. So these MIB transcription factors are the positive regulators of aliphatic glucosinolates. They can activate several downstream genes of the aliphatic pathway. In Arabidopsis, these MIB 28 and 29 were exist as single copy. However, the plant which we are working is Brassica repa is a triploid genome which contains multiple copies. Based on the genomic survey, we found that there are three MIB 28, which named as 28.1, 28.2, 28.3, and three MIB 29 genes, which are referred as 28.9.1, 2, and 3. So we framed the objectives in order to find out the suitable candidate of this particular MIB for further metabolic engineering approach. So we analyzed the expression pattern of these MIB 28 and 29 from Brassica and the downstream genes in different developmental stages, that is different tissues. And also we studied the expression pattern of these MIB genes under numerous different biotic and abiotic stress conditions. And we studied the in silico regulatory motifs which occurs in the upstream region of MIBs and the downstream genes. Finally, we studied the glucosinolate profiling by using UHPLC. So, as I said, the glucosinolates are secondary metabolites which are strictly controlled by different developmental stages as well as different stresses, especially the biotic and abiotic stresses. So, we took 7 different developmental stages, that is tissues. For example, 1-month-old root, shoot, leaf tissue, then 2-month-old root, inner leaf, outer leaf tissue, followed by 10 days old seedlings were taken for studying the expression pattern of these MIBs and the downstream genes. Regarding with stress treatments, we included elicitors like methyl jasmonate salicylic acid and substrate like glucose, phytohormones like abscisic acid, gibberellic acid, IAA, followed by wound and low and high temperature were given to the plants in order to find out how these MIBs are regulated by these stresses. So the simple bioinformatics analysis, structural, functional, motif analysis of these MIBs, phylogenetic analysis, then cis-regulatory motif analysis were also conducted. So this is about the relative gene expression of MIB 28-29 parallax under different growth stages. And we see both MIB 28 and 29 parallax were showed higher expression in the outer leaf tissue. Particularly, among the MIBs, we could see the MIB 28 third parallax and the 29 first parallax showed higher expression with regardless to different types of growth stages. So then we followed to study the expression pattern of the downstream genes. For example, we took 2 crucial genes of side chain elongation pathway, that is MAM1 and MAM3. So in which we see that compared to MAM1, the MAM3 showed higher expression. And in both the cases, the expression of these genes were showed to be higher in the outer lip tissue followed by sealing tissue. Then we took to study the expression of core structure formation genes like CYP17F1, 83A1, SUR1, and ST5C. All these genes were exist differential expression pattern in different tissues, and most of them were showed higher expression again in the outer lip tissue and sealing tissue. So finally, we took 2 genes of which are the major genes of aliphatic glucosinolate pathway involved in the secondary side chain modifications, which are crucial for the accumulation of this sulforaphane. So these 2 genes also showed higher expression in the outer leaf tissue in the sealing tissue. So this is about gel picture. So the expression of all these downstream genes in different tissues. So we could see as compared with the post-structure modification, the secondary side chain modification expression genes are found to be very low. And most cases, the outer leaf tissue showed higher expression. Then we followed to study the expression pattern of these MIB parallax and their different phytohormones. And only the IAA and salicylic acid were showed higher response towards these particular MIBs. Particularly in IAA treated shoots of Brassica repa showed higher expression of MIB 28 and 29, whereas the roots of salicylic acid treated Brassica repa contain higher expression of MIB 28 and 29. Then we go for the expression pattern of MIBs under different biotic and especially biotic means we included wound stress. For abiotic stress, we have given low temperature and high temperature. And interestingly, the wound stress both the shoot and root tissues, the expression of MIB parallax were found to be very high as compared with all other like stress treatments, and followed by that low temperature and high temperature also induce the expression of this MIB parallax. So this is the gel image. So the expression pattern of MIBs under different stress treatments. So again we see the MIB 28.3 and 29.1 showed higher expression regardless of different stress treatments as well as root and shoot tissues. So this is the structural domain and pattern of MIBs, and we see that all the selected Brassica MIBs containing MIB binding domains. Apart from that, MIB 28.3 containing transmembrane binding domain. So then we studied the like regulatory motifs which occurs in the upstream region, that is 1 to 1500 base per upstream region of all the Brassica repa MIBs, and we could see numerous kinds of motifs which is mostly relevant to MIB specific motifs and also stress specific motifs, and especially abscisic acid responsive elements, drought responsive elements, biotic stress specific elements. These elements could be seen in the upstream regulatory region of all these MIBs. Finally, we could know how the aliphatic glucosinolates are controlled by these particular MIBs and the downstream genes. So we studied the glucosinolate profiling by using HPLC in the particular tissue, that is outer lip tissue, because outer lip tissue contains higher amount of expression of all these MIBs as well as the downstream genes. So that's why we took this particular tissue for glucosinolate profiling. And we could see there are 4 different types of glucosinolates in the outer lip tissue, among which the highest one is progesterone. Progesterone is one of the aliphatic glucosinolates, so their concentration is found to be very high. So which means that these glucosinolate regulators, that is MIB transcription factors and the downstream genes, are regulating the production of aliphatic glucosinolates. So we conclude that the extracted MIBs containing R2, R3 MIB binding motifs, and also in addition to that, it contains numerous stress-specific, tissue-specific, and MIB binding motifs. Like based on the gene expression analysis, we identified that MIB 28.3 and 29.1 parallax were showed higher expression in all the developmental stages as well as with regards to different stresses. So the outer lip tissue contains higher amount of aliphatic glucosinolates, which indicates that these aliphatic glucosinolates production were controlled by these MIB transcription factors. So finally, we conclude that this MIB 28.3 and 29.1 could be the right choice for further metabolic engineering studies. And interestingly, many Brassica MIBs were showed highly responded to particular stresses like temperature and wound stress, which indicate that they may have role in stress tolerance, and these genes could be used for stress tolerance studies also. So thank you once again for your kind invitation.
Thank you so much, sir,
Thank you.
for such an informative talk. With this talk, session 1 is concluded and we will continue for session 2. Before it, I request to all respected speakers and AIB faculty members, kindly open your camera for group photograph, please.
Ma'am, is there any panel discussion so that if someone wants to have any query, they can ask with the experts?
Yes, sir, they can ask, sir.
Sir, I have one query to Dr. Mandal, sir. Sir, can you please tell me what was the strategy to find out the novel genes in your study?
Actually, there has to be some discussion. Okay, I'll tell my part. See, basically there are many approaches. In our case, we started, as I told, we started from the scratch. Nitrogen use efficiency in case of a wheat and as well as in case of rice, we started in 2011-12 onwards. So we started, that's what I elaborated. But then in brief, we started with a very large number of germplasm. It's around 300 to 400, which consists of all the materials from all over India, having diverse kind of nitrogen use efficiency. Then we put consecutive 3 years. We narrowed down. That's what we are telling. And majority of the parameter which affect the nitrogen use efficiency we gone. Then the narrowed down material we have gone on a controlled experiments. Then we got the contrasting genotypes. And those contrasting genotypes, we made a mapping population. Incidentally, in case of a rice, earlier in IIRR, which is called DRR, they were working under a NICRA project, and they screened the material, which is N22 and IR64. So means they had they had quite a few contrasting material, but we had in NIPB as well as in IRI combined, we had a mapping population of means real lines of these two. So we did a QTL mapping. At the same time, since they are contrasting genotypes, we did transcriptome. Then we co-localized them. Then, because in transcriptome, you get at least a few hundred genes. At the same times, in QTL of a biparental mapping, you get quite a large number of genes. So when you co-localize them, you get narrowed down. And then pinpointing the genes, I gave a list of genes, around 20 genes. In case of rice, we what is called narrowed down. They're all unannotated. They're all novel. So now the job is what we are doing one by one. We are actually doing functional validation, both by loss of function as well as gain of function. Now in the same process, we got 3 negative regulators in rice, which are already been reported in rice as well as in Arabidopsis by RNAi, but they did not target for the product. They just wanted to say that, yes, they are actually negative regulator of nitrogen transport, means nitrate as well as ammonia. So we took all the 3. Priyanka, we took all the 3, and then basically what we did is we are all doing genome editing of different combination, because we could have done all the 3 together, but since it is 3, we did individually 3. Then we took 1 and 2, 2 and 3, 1 and 3, as well as 1, 2 and 3. So in this process, we got a large number of plants. So now hoping another 2 years, we'll be able to get some plants from there. So basically the strategy for us is identifying the contrasting genotypes, making a mapping population, find out the QTLs, at the same time do a transcriptome, co-localize them, and identify the genes, functionally validate them through a transgenic approach, and preferably in the same plant, means like in the rice, because in that way, sometimes you get the product also. Suppose it's a positive regulator, when you're going for a gain of function, you get a plant which you can actually release as a transgenic plant. Of course, you have to be a little strategic, because what marker you are going to use, they will be afterwards will be an issue for biosafety. So that if you are properly planning, you can do that.
Thank you so much, sir. You explained very well, and you emphasized that the negative regulators are knocked out in your study, and probably you are taking care of the macronutrient use efficiency, and people, they are working with the micronutrient use efficiency also. So
yeah, no, micronutrient use efficiency. More than that, they are trying that micronutrient, what is important for the human being, like biofortification. We have a program on that also, like more zinc and iron. Nabi is working quite a bit of thing, because they are working mostly on the food, means post-harvest part. So yes, we are, of course, here it is nitrogen, and more for environment.
thank you, sir. Thank you so much.
Shall I ask Dr. Tiwari one question? I have 2 questions actually, one for Dr. Tiwari and another for Dr. Rajesh. Yeah, Dr. Tiwari is there? Dr.
Is there,
Siddharth Tiwari.
is there any question answer?
Yes, sir. He is here.
Yeah, Dr. Tiwari, can you hear me? I'm able to see him, but then I'm not getting his response.
Yes, sir.
Meanwhile, I just asked Dr. Rajesh just now, who has presented about the glucosinolate. Right? Yeah, Dr. Rajesh is there from Chennai.
No, sir. This is Dr.
Dr. Bhaskar is there.
Yeah, sorry, not Rajesh. Bhaskar. Sorry.
Dr. Bhaskar is there.
Yeah, Dr. Bhaskar. Primarily, I want to ask this. Savita University is a dental, means basically a medical college. So, how you are working on brassica?
Actually, thank you for your question, sir. Actually, this is my PhD relevant work. Right now, I'm working as a research faculty here, not like academic faculty.
Okay,
So
fine,
this
fine.
is like a general university. So this is my PhD work.
No, that's fine. Now my question is,
Okay.
see, glucosinolate you are recommending for health benefit, right?
Yes, sir. Yes.
But then, you know, we, not only we, entire agriculture university as well as throughout the world, people are trying to reduce glucosinolate. See, there are 0 and 00 variety of mustard. 0 is where the erucic acid is very minimal or almost 0. And glucosinolate is very, very minimal. In this case, that's called 00. Where the canola has been sold in the India from Canada, that is because they don't have glucosinolate as well as what is called erucic acid. So when we are actually trying, we means entire brassica fraternity is trying for like reducing erucic acid as well as glucosinolate. Of course, what you are telling is correct. That actually both glucosinolase as well as erucic acid helps in biotic as well as abiotic stress tolerance. So the plant which has 0 and 00, they have problem from germination onwards. Of course, that is there. But the oil quality for human health perspective, they say that it is better. They say means there are release variety. There are many, they are called quality mustard. So 0 and 00. So this is bit contradictory, you know, what we do.
Like, thank you for a nice like details about this brassica. Sir, yes,
No, because we are all from
yeah.
agriculture system,
Yeah.
and typically we are working on brassica improvement. So anybody in brassica, if you ask, they are going for 0 and 00 varieties. So the other part is very, very correct, because the yes, Dr. Kaul, Tanusri.
I wanted to say one thing, Dr. Mandal. So these 0 glucosinolate varieties will be eaten only by animals, sir. Human beings would not like to eat it because without the pungency, the Bengalis would not take the.
No, that's okay. That there is a lot of arguments. See, the thing is, animal doesn't eat the mustard as such. Animal eats the cake.
Oil cake.
So the oil is no. The thing is, 00 variety is used so that the oil can be consumed by the human being, and glucosinolate is there mostly in the oil cake. So that is for the animal feed. Understood?
But the human beings would not take this oil.
No.
Nobody wants this 0 pungency oil. Nobody wants. You cannot sell it to anybody.
No.
You can try this.
See,
and
this is
the oil cake that is being developed in the 0 glucosinolate oil mustard, from the 0 glucosinolate mustard that Dr. Naveen Bisht has produced at NIPGR, that will be given only to feedstock for, because soybean is very crucial. It is very costly, not imported. We are mostly importing it, not producing it. China is mainly a competitor. Plus,
no, Dr. Tanusri,
Brazil and Canada. They are importing from there.
No, let me
That's
explain
why
this.
they want to replace the oil meal cake of from soybean to mustard using the 0 glucosinolating. But human beings, I bet they won't like to take this.
No, listen to me.
Even though it is whatever you say, cardiovascular diseases or whatever you say.
No.
There are so many other reasons where cardiovascular diseases happen, not just by mustard oil. So this is one thing people would not give up. This is their habit. They cannot give it up. You try. I've seen by discussing with many scientists. So they said this.
Dr. Tanusri. Yeah. So let me tell you, what you are telling is correct. Habit cannot be changed. The people who takes tobacco, they can't change it overnight. So that is an entirely different issue. But if you come to IRI, Naveen Bisht has done, I know very well, but then IRI, they have almost 12 varieties of 0 and around 15 varieties of 00 of mustard, which has been cultivated. And if you come to IRI, the quality mustard oil, what is sold from the pusa itself, is not having pungency, and we don't like it. That's a different thing, whether we like or not. But for the health sake, we are eating ragi. I don't like ragi biscuit, you know. That looks like ash, but then we eat it. I am just talking about the concern what people are showing. You are talking about the habit of the people. Habit cannot be changed. I know that very well. So that's an entirely different thing. But 0 and 00 varieties are being promoted, citing the cardiovascular diseases, and one for human, another for animal.
So in case of improving the diet, so we will go for olive oil rather than
No, that's okay. That's a
sesame oil
different
and
thing.
oil, rather
I'm
than
not
going for
talking on
mustard
oil. See, Tanushree, I worked 15 years in oil palm. Now PM is promoting palm oil. This all are entirely different issue. We are not diverting this. I am only talking
No,
about the glucosinolate.
I mean to say, if we want to go for good oil, which
No, I
has
am not
high
saying the
oleic
good oil
acid
question at
and
all. Dr. Tanushree, we are not discussing about which
we
oil
would
is
go
good. We
for
are
oleic
talking only
acid,
about the
olive oil, or
No.
sesame oil, or some other oil which
You
has
do whatever oil it is.
good saturated fat,
Madam,
unsaturated fats. So
Madam,
rather than
olive
going
oil.
for mustard oil without any glucosinolates, there wouldn't be any market for it. That's what I'm saying. And farmers won't grow it if there is no market. If I am,
No, listen,
that is
listen,
what I'm
madam.
saying. Whatever
Madam,
amount of reduction you can do, that would only go for animals. That is what
madam,
I'm saying. The
the
rest
quality
rest
mustard,
is up.
listen, quality mustard seeds are almost 1.5 times more costlier than the pungent mustard oil. Okay.
Right.
So you are
So
in
that is
different world.
another reason for not buying. Okay.
That's
So
the
that will
different
be
thing. Not
another
buying
reason for
question नहीं
not
है.
buying.
मैं ये
So
बता
in
रहा
this,
हूँ
in
कि
this way, I
you
can
come to the
argue
practical reality
that the farmers
of
won't grow it.
agriculture. Who told not do it?
You
They are
make
all
the
taking double
reduction
zero
in
mustard.
pungency, farmers won't grow it because there is no market.
How
And
many
also
farmers you
the
have discussed? I
main
know.
point is that mustard has a problem of reduced oil content when they grow in the field. Not enough
Tanushree, I'll
oil
have a
is.
different discussion with you on that.
Yes.
So,
So I abide by the conference.
now Dr. Siddharth is there. I just have a question. Siddharth is there, but then it is not there. If anybody can answer this, I have gone through the paper of Journal of
Dr. Mandal, actually, I would like to ask one question.
Yeah, please.
As the European Union is not much more enthusiastic about the transgenic crops, the same I think is with India also. So we are working on transgenic crops to improve the varieties and all these things. But if they are not interested in transgenic crops, because I think India is also not promoting much more the transgenic crops, then what will be the fate of this?
No, who told it is not? Which one you are telling about the fate?
Means like BT cotton and BT brinjal, and after that, I think they have not promoted much more about this type of crops.
No. See, there are different issues. They are all much bigger perspective. Let me tell briefly about this. See, transgenic is at present, even Dr. Pentel's mustard was put there, then it is in the Supreme Court for a different issue. You can't say it is not, government is not pro or government is pro or that. The thing is, when it is needed, you have to go for a transgenic. At present, we have in hand, there is a different technology which we are calling as genome editing, which government has totally opened, that SDN1 and SDN2, you can go for releasing the variety. So majority of the people are actually going towards the genome editing than the transgenic one, which takes much more biosafety and regulation. Now, if you say about the European Union, at the same time, American president is eating the transgenic food. It is all a perspective which does not actually justify the science. And then when there is a need, see, at present, we are almost self-sufficient, though the equity is not proper. We are self-sufficient in the food production. In fact, we are surplus. Right. So because of that, we are not much pushing on the technology, which is having a controversy. But suppose a situation comes where we are not able to produce and we have a transgenic crop which will be giving a better yield, actually we have to go. And at present, as I'm telling, last 3-4 years, the genome editing is, see, DBT is giving a different call for genome editing. ICR is giving a different call for genome editing. And people are working because the regulation is less. And it is all different kind of a politics, whether European Union is not everything. America, everybody is eating transgenic food. So they are all surviving. It is not that they are bad or
They are eating.
Yeah, that's what I'm telling.
But here, mostly this is ethical issues and politics, you can say.
Everywhere it is such kind, some or the other issues, but scientific part, if you say, nobody is telling that you don't go through the biosafety norms and you don't see the safety issues. You check the safety issues and do that.
Sir, there is one question also that the genome edited gene is also like, what is the chances of getting laterally transferred to other crops also? Means once Tanushree madam has told about the genome edited glyphosate resistant maize that has been produced through CRISPR-Cas9. So what is the chances of that edited gene to move to other crops? That is what I have a query.
Dr. Tanushree will reply, but before that, I say that, see, why they are telling SDN 1 and 2 is permitted, that is because the same allele is existing. It is only a mutation in the gene, you are not transferring a nucleotide piece from somewhere else. And that is the reason the biosafety is relaxed. But when you go to the SDN3, where you are taking a piece of a gene and then replacing that, there it goes equal and equal rigorous biosafety norms. Understood? See, suppose your A is changed to
Yes,
T,
sir.
and you get a different allele, which you give a better yield, or suppose your glyphosate resistance.
So, ultimately, sir, gene is mutated now. Ultimately, gene is mutated.
But your alleles are mutated only.
Right,
Your two
right.
different alleles are actually two different forms of mutations,
Right.
May I add one sentence, please?
Yeah, ma'am.
Dr. Raghavendra, your question is quite appropriate. The answer to it is that when we release it through DBT biosafety norms, when we release these gene-edited crops in the field, there would be no Cas9 gene there. So the main criteria is to make it Cas9 free. Only then it will be taken as a mutant.
Okay, thank you.
So just 1 second, if anybody can answer this, even Dr. Tanushree also can answer this. I had a query about Dr. Siddharth's question. See, their banana, they got edited, but then there is no transgene in that. How it is possible, I don't know, because anyway, we do the selfing and then we get it back. Yeah, please.
So there is a technique called ribonucleoprotein complex approach.
No, but they have not done by ribonucleoprotein. They have actually done through transformation. A cassette has been put there. This cassette is there in the Journal of Experimental Botany paper. You see, they had a transgenic cassette.
That won't go into the field, definitely.
No, that is what I wanted to know.
Now they are doing RNP.
No, RNP, I know. RNP, you are actually doing a protein, already Cas protein, you are just injecting, that is doing its job and getting disintegrated. Right. But here in this case, they have a cassette.
That won't go into the field, sir.
No, that is what I wanted to ask. You see the paper, Journal of Experimental Botany of Dr. Tiwari, his students, but there I did not get it. No, one thing can happen, I don't know really why I am asking is, we have wheat plants where we don't get the transgenic, but we got the editing. We had a lot of confusion why it is happening. So maybe this could have been substantiated, that's why I'm asking. I'll just mail him separately. Yeah.
I think it is over. Thank you, everyone. And it was great experience with you all. Thank you once again. Now, it is my responsibility to introduce our speakers of session 2. For this session, chairperson is Professor Dr. Mukesh Gupta, Director ICMR NAR FBR Genome Valley, Hyderabad, Telangana, and the co-chairperson is Professor Dr. Vinay Dwivedi, Director AIB. Now the speaker is Dr. Mukesh Kumar Gupta. He has earned his PhD in biotechnology from Konkung University, South Korea. He has more than 15 years of research experience at South Korea and National Institute of Technology, Rourkela. Throughout his career, Dr. Gupta has held various prestigious positions showcasing his leadership and expertise in academia and research. As the director of the National Animal Resource Facility for Biomedical Research at the Indian Council of Medical Research, he demonstrated his commitment to advising biomedical research initiatives. Currently, as the Dean of Sponsor Research, Industrial Consultancy and Continuing Education, and as a professor in biotechnology and medical engineering at NIT Rourkela. His work on modified solid surface vitrification is used worldwide for cryopreservation of pig oocytes and embryos. He has more than 100 publications in SCI Index journals and has 18 patents to his credit. I welcome you, sir. Please continue.
Thank you. Thank you very much for inviting me
to this presentation and share my data. Let me share the screen with you. Is my screen visible?
Yes, sir, it is visible, sir. Please continue.
Thank you. So, good afternoon to everyone. Currently, I am working as a director of ICMR National Animal Resource Facility, Director of Animal Resource Facility at Hyderabad. I am no more the dean of NIT Rourkela because I have come here only in. Just let me see if my screen can be. So today I'm going to share some of my data on development of hemostatic agent, nanofibrous hemostatic agents, which is based on the chitosan. And we are trying to show one product which is of great importance to both the civilians and as well as the military. So as we all know, the hemorrhage or the rapid blood loss is a very, very common issues in both civilians and militaries. It is responsible for 50% of the death in battlefield, and 31% of all the trauma accident that happens, the mortality of 30% of them is due to rapid loss of blood. It says that one death in every 4 minutes occurs due to trauma. And the way to solve this issue is in hospitals is to do either cauterize it or ligate the arteries. And there are certain commercial agents such as quick clot, Hemcon, and so on, which are either powders or in the form of a tablet or something which they use it for preventing the blood loss. So these commercial hemostatic agents are usually based on the thrombin or gelatin or collagen and cellulose or a combination of these agents. But these agents, whatever is available in the market, is a very big market. There's about $4 billion market in the world, throughout the world. It's a very big market. But the problem with this is all these products takes at least 30 seconds to clot the blood, by which time many of the preventable death still happens. And some of them, say, for example, what you see below here, quick clot is very difficult to apply. So when they say they use the applicator, it's a gunshot wound. When they apply, it's a very painful experience to them. And this limits their applications into this. So in the long clotting time, difficult to application, and some of them causes thermal reaction there, which can burn the tissue, nearby tissue and all. In very recent years, there have been new impetus on using chitosan as hemostatic agents, and we all know the chitosan has a lot of advantage, like they're biocompatible, hemocompatible, they have antibacterial activity, they can activate platelets, they can cause the addition, activate and cause degranulation. And therefore, a few products based on the chitosan is also available, like Hemopad, Hemcon, Celox. Celox is the most commonly used one, which is available both as a powder as well as the gauze. It is available in the market. But these products, if you look at the market, they say they take long blood clotting time, by which time the sufficient blood loss may have happened to cause the death. These agents also use toxic cross-linkers for them, which has a cytotoxicity. Some of them try to improve the blood
Sir, you're not audible.
Hello. Gupta sir, are you there?
I think he has lost the connectivity.
Yes, sir, I'm calling.
Yeah, I lost the connection. Let me share the screen again. So we took advantage of that self-assembly method. So we mixed the chitosan casein and optimized the ratio, and optimize the pH to have isoelectric point. So it has to obtain the positively charged chitosan and negatively charged chitosan. And then we dehydrated them by using the tersinebutanol so that they assemble into nanofibrous material. And then that can be used. So we mix that, wash with tersinebutanol alcohol, dried them in the powdered form, and characterize them both in vitro and in vivo studies. We did that. So as you can see from this, when we did that, we could obtain the nanofibers, and the proportion of the or the diameter of the nanofibers were directly proportional to the chitosan content in it. What you see here is CC30, CC50, and CC70, which basically refers to CC30 refers to 30% of chitosan, and CC50 refers to 30% of 50% of chitosan, 50% of casein, and CC70 refers to 70% of chitosan and 30% of this. So as you can see, we can obtain the fiber diameter of 25 to 67 nanometer, and that was directly proportional to the content of the chitosan. As the chitosan percentage increased, fiber diameter increased. At the CC30, which is 30% of chitosan, we could get a nanofibers material, which is without using the electrospinning machine. Just by self-assembly we could get. And this happened because, as you can see from the FTIR data, there was interaction between the positively charged the amide groups of the chitosan and the negatively charged casein's peak of phosphate groups. So both peaks you can see here at 1630 at the amide group and the phosphate group, they could interact and form a interaction between them, cause the nanofiber structures. And this also lead to the change in the surface charge. If you look at the zeta potential here, the CC30 surprisingly has a negatively charged, and as the chitosan percentage increased, they acquired the positively charged charge on them. So when we use this material and look at the hemostatic efficiency, as you can see in this graph, the CC30, when we try to do in vitro test using the goat blood, they could cause the blood clot formation within 9 seconds, 9 plus minus 2 seconds. And as the chitosan percentage increased, clotting time increases, but it was much less than the commercially available material. Celox is one of the, as I said, chitosan-based material, which is commonly used. It took 90 seconds, whereas our material was as low as 9 seconds. If you look at the INR values also were much lower compared to the Celox material. And this graph, what you see here, C, this refers to hemocompatibility assay. They all were highly hemocompatible. If it is less than 5% hemocompatibility value, we say it's highly compatible. So they were highly hemocompatible as the percentage were much less than 5%. And they could also cause the adhesion of platelets. So you can see in this graph, the quantitative assay, and on the right side, if you see, this is the scanning electron micrograph of CC, of a polyelectrolyte complex. So you can see the fibers and the fibers in which you can see the RBCs entangled into this, whereas we could not find such event in the Celox because they get dissolved into this.
And these clots which we formed, the clots were also stable. When we did the mechanical strength, they have about the 20 to 27 kilopascal of mechanical stress, the clot from them. So this strength 20, 25 means it's like a gel-like structure, good enough to prevent the rebleeding of the blood. So thereafter, we did the in vivo study in using the rabbit ear artery model. We made a 1 centimeter incision into the rabbit ear, applied our material, and then you look for the clot formation. So we can see that in rabbits also, we could clot the blood. It took in the rabbit the 37 seconds. So in vitro, we could clot the blood in 9 seconds, and in vivo, it took about 37 seconds to clot the blood. We have now optimized this, all the conditions, the pH, the quantum of hydrogen and all. Now in our new product, which we could clot the blood even at 9 plus minus 2 seconds, even in vitro, in vivo models also. So you can use in a rat model, and you use this. So you can see here, CC30 could clot the blood in vivo. The Salox took about, just a minute, I think I'll stop my video. So Salox took about 95 seconds, whereas our material as quick. So it's so fast. Within 10 seconds, we could clot the blood. And the clotting amount of blood that lost is nearly same as what we get with the commercial products are there. Very small amount, 10 milligram was sufficient to stop. This is a rat femoral artery model. So directly on the femoral artery, we made a puncture. And then, so what we saw is the product that we have developed was capable of preventing the blood clotting both in vitro and in vivo within 9 seconds. We have also used this product to make a gauze, hemostatic gauze, which we can use it as a band-aid or something for the surface laceration, or we can use it to pack the punctured wound. So we again prepared hydrogen casing polyelectrolytic complex as an optimized PS, mold casted them, and lyophilized them to prepare like a gauze, and did the detailed physicochemical characterization and in vitro and in vivo blood clotting assay. Plus, we also did the detailed safety assessment, both in terms of biocompatibility, immunocompatibility, as well as the soft safety toxicity assay. So what we did, we prepared the hydrogen casein at different ratios. And first we created a film-like structure. So you can see there that we tried to dilute them at different percentage to coat them on the petri plates and get the product by solvent casting. So different compositions we tried, out of which 80% was found to be the best to get the uniform gauze. And you can see that they are macroporous gauze we can get. You can say it a gauze, or you may also say it as a sponge. They are sponge-like structures. And in order to, we did one more thing here. In order to increase the bioactivity, we also incorporated into it the silver nanoparticles to see if it can have antibacterial. So if you want to use it like a band-aid in lacerations, it should give not only the protection against the blood loss, but also should have the wound healing, promoting the wound healing and provide some antibacterial activity. So we also incorporated the silver nanoparticles into this. So as you can see here, when we prepared the gauze, the structural integrity was not changed. They still had the complex formation, and there was no effect. Below this one, what you see, A1 is with incorporation of silver nanoparticles. There were no significant change in the structure of the both the materials without silver nanoparticles and with silver nanoparticles. And these sponges were porous, highly porous. If you look at the CC30 composition, they had the approximately 73% of porosity. And they were highly, the spongy nature to absorb the water. See, when we clot the blood clotting mechanism is such that if it can absorb the blood, it leads to the concentration of the clotting factors and therefore promote the blood clotting. So you can see there is roughly 10 times they could clot the blood. They could absorb the water fluids. And there was no impact of silver nanoparticles on affecting either the porosity or the water absorption capacity. Now look at the clotting time of the sponge. So clotting time, this was less than 30 seconds. About 30 seconds we could do, which is 1/3 of what the time is taken by the Silox material. And BCI index, blood clotting index also, if you see, it was a very good value of less than 30. Again, it's a biocompatible. You can see less than 5% is there. There were no effect of silver nanoparticles. If you see in A1, B1, and C1, there were no effect of nanoparticles either on clotting time or on blood clotting index or on the hemolysis percentage. And when we look at the antibacterial activity, chitosan itself has some antibacterial activity. When you look at here, when we had chitosan casein gauze without nanoparticle, and if you can see it here, those materials did not allow the bacteria to grow. Bacteria were there, but did not. This is what you see here is quantified data. This is the zone of inhibition test. So bacteria could grow. This is for each step aureus, and this for gram-negative E. coli. So both gram-positive one and gram-negative. We have taken 2 samples. In both the cases, what we see consistently, the materials, both with and without the nanoparticles, they were, sorry, both the materials were the bacteriostatic. They did not allow the bacteria to grow. So if you see within 24 hours also, the amount of bacteria remained nearly same. So bacteria, it suggests that the chitosan casein gauze were bacteriostatic. Now, when we added the silver nanoparticles, they become bactericidal, kill the bacteria. So the CFU unit has become 0. So this was true for both Staph aureus as well as the E. coli. So our materials were now not only bacteriostatic, but also bactericidal in the action. We also checked this effects on the blood clotting efficiency in vivo model in rat femoral artery. So you can see similar to what we observed earlier, within 30 seconds, they could clot the blood, both without nanoparticles and with nanoparticles, and they were much better than commercially available Silox. And the amount of blood loss did not differ significantly between them. So we look at the mechanism. So one mechanism, as we have seen earlier, they could absorb the water and cause the concentration of the coagulation proteins to promote the coagulation time. We could see here also, when we added this gauze, and the gauze could cause degranulation of the degranulation of the platelets and then cause the activation. So this transmission electron microscopy confirmed the degranulation by our materials. We also tested its biocompatibility. So we took this gauze, make an incision into the skin of rats and transplanted them under the skin. And you could see that the wound can heal normally. There was no adverse effect either on the growth of the animal or behavior of the animal or feed intake or the water intake or body temperature, no pyrexia. And you can see the healing where normal, proceed normal. And by day 14, we could see that healed completely in them. There were no antibodies applied on them, no antibiotics. And you can see there is no evidence of localized inflammation or no evidence of any infection there. When you took a section of that skin and is stained by the HND staining, you can see that by 14 days, we have a complete keratinization of the area. And they were also, when you did the mason trichome staining, you can see the normal collagen deposition in both the control and treated group, nearly similar areas. You can see control area still have some non-grown area where complete skin regeneration did not happen, but here you can see complete regeneration could happen. We also tried to check systemic toxicity because this material, nanofibrous material, there is a tiny possibility that through the blood stream, we applied this in a major artery, the femoral artery. There is a possibility it may go through the blood circulation, lodge elsewhere, and cause the toxicity by causing the emboli and leading to the thrombi formation.
So we took several organs, 3 organs we are showing here, 3 major organs, kidney, liver, and spleen. We saw no evidence of any toxicity in there. All look similar. We also stained them to see if there's any deposits of the collagen or something. We found no evidence of any adverse collagen formation anywhere in the kidney, liver, spleen. We did for several organs. Only 3 we are showing: skin, kidney, liver, and spleen. So it was no adverse effect. Then we also look at if it causes any immunogenicity. So when we look at, we use 3 markers: CD3, CD8, CD68, and these 3 markers: CD3 for the B cell, T cell, and the macrophages. This, what you see, is spleen as a positive control. So they are very strong positive, but no such evidences in the treatment group, whether it's without or with silver nanoparticles. When we look at the von Willebrand factor, which is for the vascular for neovascularization, you can see the neovascular formations both in the control group as well as here. So that means the product that we are using was non-immunogenic in nature. So taken together, we have shown, if you see, this is our material. This is blood in which we are adding our material. You can see it's how quickly within 10 seconds it is clotting the blood. You can see. Within 10 seconds, it clots the blood. That's in vitro. And if you look at if you look at Celox in the bottom, you see we have added this material and it takes a long time to clot the blood. So we show here a hydrogen-based polyelectrolyte complex, which in the form of powder can clot the blood within 10 seconds, both in vitro and in vivo, in rat femoral artery model as well as the rabbit. And we also saw we could make from there an hemostatic gauze. That gauze can be attached to an adhesive tape, and that tape could be used in future for surface lacerations, and also they could clot the blood in 30 seconds. And 30 seconds in major arteries. In lacerations, we have to remember that there is only tiny amount of blood that comes out. So it could be used for clotting within 30 seconds. And we have done in vivo assay. In vitro, it takes 27 seconds. In vivo, also it takes about 32 seconds. And they were biocompatible. They were non-toxic in our study. This is all for my presentation. These are the students who have been helping us. This is my laboratory student before I joined ICMR. Most of the work is done by my PhD student, Balaram Mishra, and Devendra Verma, my colleague, is the main brainchild when we started with the material science area aspects of it. And Dr. Devendra Pathak from Garhwal, Ludhiana, helped us with the animal experiments at that time in Garhwal. And of course, we should acknowledge the funding agencies by both ICMR. This work was supported by ICMR, BBT, CSIR, as well as the SARB. We have many other experiments to be done. We have done many other experiments, but due to paucity of time, I have shared only some of the data there. Thank you very much for listening.
Thank you so much, sir. The topic was very clear and informative and gave us information about activity of nanofibers. Thank you once again, sir. Now, our next speaker is Dr. Brijesh Kumar. He is currently an assistant professor and head in the Department of Chemistry, Tata College, Kolan University, Chaibasa, India. He received PhD in chemistry from the University of Delhi, India in 2009. He is a pioneer researcher in the field of green chemistry, nanosynthesis, environmental remediation, photocatalysis, nanomedicine, polymer, and natural product extraction. Dr. Kumar has several national and international fellowships to his credit and has worked as a faculty member in various universities throughout the India. educator, and South Korea. He is a holder of 2 registered patents as well as a member of American Chemical Society, India Society of Chemists and Biologists, and Indian Science Congress Association. He has also published more than 80 research papers at CI Scopus Index and is an active reviewer of more than 70 journals. He is also included in the top 2% of the scientist list in 2021, 22, and 23, prepared by the experts in the Stanford University, USA. Welcome you, sir. On behalf of AIB AUMP, please, sir, continue.
Okay, good afternoon, everyone, and thanks for nice introduction, ma'am. So,
thank you, sir.
Okay. Okay, is it?
Yes, sir, it is visible.
Okay.
Make it full screen, sir.
Yeah. So now, okay. So, actually, my work is on green synthesis of metals. First of all, I would like to say special thanks to Amity University and Professor Raghavan Misra for inviting me to deliver lecture in this online mode. For the last more 13 years, I'm specialized in nanoscience. Actually, I'm a chemist. So on the time being, as per the requirement, I move my specialization from the organic chemist to the nanochemist. So I'm going to start with the simple image. This is the image which says about the great Alexander. And everyone knows from the ancient times when all the peoples are coming to invade or conquer India. So at the age of 33, he died. And and what is going to happen if he when he was moving from west to east, means west part to east, a lot of always they have the different category of army. So the first army that is they are on the higher rank, and another one the simple soldier, and they are going to use, because it's there are some paper also written, this is my own interpretation, because those army they are on high rank, they always use gold and silver utensil for drinking water and for eating food. But for the soldiers who are normal, they are going to use iron vessels. And that time, our ancient means our all the earlier peoples also know about the importance of gold and silver. Like they are going to put in gold colloids or silver colloids. And when they used the iron vessels, they have a lot of, we can say that, because they have a waterborne disease. And this is the favor, because iron favors the growth of microbes. And not only that, this is the image of, this is also the medieval age image, and it clearly explains about the church in Western countries. They paint like a different color, but we're surprised to know that they are not used the pigments. They used gold nanoparticles. Means all the pigments, all the beautiful colors are form of gold. Means normally it is a yellow color, but when it comes in nanoparticle form, it seems like a different color. And it is a, so that time also we the ancient peoples knew about the nanotechnology, but due to the some we can say characterization technique, they are not going to say that what is this. So in normal, we can say that nanoparticles or nanoscience means any molecule which comes under the range of 1 nanometer to 100 nanometer, it comes under the nanoparticle or nanomaterials. So this is the main part. If this is the part of this, so 1 nanometer to 100 nanometer, and how can we synthesize? How can we make it? So the simple way for the chemical synthesis is, we can say that bottom up synthesis. Just we can take the atom molecule, we can reduce it, and we can make a molecule in the range of 1 nanometer to 100 nanometer. So properties of the materials change in nanoscale. So this is, and they have the different techniques. So as per the chemist, we can explain about the bottom up approach. Means we are not related to the just we hammering or microelectronics from top to bottom down approach. We are going to start with the atom, with the ion, reduce it, or make a group, and then we can keep it in nano size. And this is the general trend. It was taken from the internet also, that how the nano world is going to change. Means DNA, if we are going to say about the DNA, it's a 2 nanometer. They are going to come in the part of the ATP synthesis. 10 nanometer or 10 nanometer of diameter is there. And also, this is the important part about the nano world, or we can say about this. Come to the point on there, why the nanomaterials are attractive. The first question is that why the nanomaterials are coming to attractive, why they are going to use for different scope or application. So they are going to use for the information technology, they are going to use for the energy, health, environment, and they are going to for nano manufacturing. Why? What is the reason? So I'm skipping this one. And what is the reason that why the peoples are going to interested in nanoscience? Means if someone is going to say that we want to work on nanoscience, so it doesn't matter either you are from the chemist, you are biologist, you are physicist, or material science, because it is interdisciplinary. So first, whenever you have prepared the nanoparticle or nanomaterial, you can use in different application. So this is the nanoscience part. And why it is important? Because when an atom comes under the nano size, or molecules comes under the nano size, they have a reduced melting point. It becomes ultra hard. They have a optical properties. They can use any cosmetic sensing. They are going to, because so in an earlier time, because gold is yellow color, but when it comes in nano size, they are purple color, violet color, blue color. It is a intrinsic property of the we can say the nano materials. They have the high electrical conductivity, they have the high magnetic property, they have the self purification, and they have also the chemical stability. So the question always arises, means how can we screen nanoparticle or nanocells or nanoparticles for different application? So this is the first instrumentation technique, that if we are going to prepare a nanoparticle, we can first characterize by the UV visible spectroscopy. And then if it comes under the range of UV visible or UV visible range, and then near infrared, means 180 nanometer to 1100 nanometer, if some peaks are coming or the color is coming, so we can easily use or efficiently use in cosmetics and pigments, photonics and telecommunication. They are also used for the solar cell application. Right now, in current scenario, the world is always looking for the challenges for the food, water, and energy. So now in the solar cell, they are going to replace, or they are other one dye-sensitized solar cells. So they can use the pigments, and then they can use for the energy harvesting from the sunlight. And then they are also the part of the environmental sensing. Sometimes the pesticides are there, metal sensings are there, we can use for the nanoparticle synthesis. And this is the practical application which is in the market, means high performance skywax. They are going to use for sunglasses, they are used for the socks, for the using by putting the nanoparticles on there. And so my idea, means my work that I did in the lot of, I did the work for the synthesis of silver nanoparticle, gold nanoparticle, zinc nanoparticles, palladium nanoparticle, iron nanoparticle, and copper nanoparticle. So what is the basic things about this? Means if we are going to use for the gold, silver nanoparticles, means they have the very good antibacterial, anti-cancer activity. For the palladium, it is used for the good catalyst. For the organic synthesis, for dye degradation. For iron nanoparticle, they have the magnetic property. They are going to use for the anti-cancer. They are for photocatalysis or dye remediation. So, and then this is the same way. So the main issue is going to consider is that how can we prepare the nanoparticle? The first thing is that synthesis of nanoparticles. Second, nanoparticle means if you are going to prepare nanoparticle, how can we functionalize, or how can we wrap the nanoparticle with the different molecules? Or then how can we characterize it? And then the main issue is that reproducibility and the toxicity, because nanoparticles have some toxicity also. And then after we are going to implement. So my idea is that whenever we are going to prepare the nanoparticles, so we are going, why not we can, instead of chemical synthesis, we can prepare the nanoparticle by using phytochemicals or bioactive molecules from the plant. So this is the first impression, that it seems like a handprint or footprint, but this was my first paper in 2015 when I was in Ecuador. So it is the nanoparticle, gold nanoparticle was prepared by capsicum bacterium. Means it is they are going to say that's ahi. And why I choose this one? Because ahi is originated from the Latin America, and then they are going to transfer all over the world. And this is the nanoparticle which is directly prepared by the capsicum, means the phytochemicals which is present in the chili. And so now, what are the phytochemicals that are dominated in nature? The first one, we can say the flavonoids. So every day we are eating onion. And then second is the glucosinolates that we are in the brassica family. And in case of flavonoids, the most important is that we are, sometimes we can say that flavonoids and the rich source is onion. So sometimes in here, everywhere in the world, they can say apple a day, doctor away. But the some, if we are eating an onion a day, there is no, if it is also going to substitute apple, means onion a day and doctor away. And my idea is that why not we can use some phytochemicals for here for green synthesis, and with the looking about the sustainability, means always science needs for synthesis of materials with the efficiently, with the less environmental pollution. So, and another one, the carotenoids and anthocyanins and the chlorophyll, because a lot of peoples are working on a lot of peoples are working on nanoscience, and they are going to use for the plant pigments. So low cost and green technology is forcing nanoscience to develop a new methods to guarantee the sustainability. So my approach is that to keep the environment by using the environmental material, and for protection of environment by using the by degrading the different toxic dyes. So this is the correlation of the different plants, that the phytochemicals they are rich, and that phytochemicals, the most important is quercetin, and it is the present in the, you can see that quercetin and their glycosides are present in the most of the vegetable, and we can say that horticulture crops. And this is the correlation, means how the correlation means how the correlation and their importance means, if someone wants to use onions, so they have the majority the quercetin. So on looking all this, means correlating means whatever we are eating, whatever we are taking in our food, why not we can use for the preparation of nanoparticle and then modify it and then can use for different application. So this is the correlation again. One of these means they are the specific phytochemicals that is rich in this plant, like blueberry, pea, cranberry, and there are a lot of phytochemicals are there. And the good thing for plant-based nanoparticle synthesis is that we are, because if we are going to talk about the nanoparticle synthesis, we can use every part, but we prefer to use those plant material that is discarded one, means we can use it discarded one, for example the waste like peel, and we can use the seeds, sometimes we are not eating. So this is the normal strategy for the preparation of nanoparticle: gold, silver, palladium, copper, zinc, and iron. And then we have to use for characterization by using the simple UV visible spectroscopy. And then gold and silver, if it is prepared due to the change of optical property, we can also identify by these. And then after we can proceed for the next instrumental technique like DLS, STEM, XRD, FTI, TGA, etc. And then we can, as per the size and as per the size, we can characterize, we can use for the like catalyst, we can use for organic synthesis, we can use for chemical sensor, we can use for drug carrier, etc. So what is the basic funda behind this? Because if you look for the reduction potential, means reduction potential of the phytochemicals or flavonoids or polyphenolics is normally 0.8 to 0.3 volt, means we can say volt. So all metals which value is higher than 0.3, they can easily reduced by the phytochemical. So we can choose here, means we can easily prepare gold nanoparticle, palladium nanoparticle, silver nanoparticle, and using iron nanoparticle using ferric chloride, because they have the high reduction potential in comparison to the phytochemicals. So phytochemicals oxidize itself and then can reduce the metal. And the simple strategy is that the first the metal is going to reduce, and then after it can make a group, or you can make the nucleated, and then they can control. Because whenever we are going to prepare the nanoparticles, we have the two things. First is the reducing agent, and second is the capping agent. And the good thing for the phytochemicals is there is no need to add both separately, because phytochemicals, some phytochemicals, for example, flavonoids is a good reducing agent, and where the starch, or we can say the sugar, is very good capping agent. So in one single pot, a single step synthesis, we can prepare nanoparticle. And the main thing is that we have to control the size. So this is because always the peoples are going to use for nanoparticle synthesis of toxic chemicals like sodium borohydride, hydrogen, but the main thing is that they are costly. So we can substitute the costly or hazardous chemical by the waste material or vegetable or fruit pigments. So this is this is my work that I did on there. The first one is the citrus paradisi, that the peel used for the preparation of nanoparticles. Second was we can say that the blackberries. And the third one, this is the part of like hairy lychee, and then rosa cincia, and lantana camara, and sacha inchi, and prunus serotina, that is a capuli. So this is the main part that our group is already reported on there. So with the different variety of nanoparticles. And so this is the recent paper which was published by the Helion with the use of pollen of bee, because normally we are going to use honey, but in South America and some Central America, people are preferred to eat pollens, which is collected by the bee. And that time we prepare this gold nanoparticle by using pollen, and then we can use for the reduction of nitrophenol. And nitrophenol is one of the starting reagent for the synthesis of most of the dyes industry. So this is, you can see here, this is the nanoparticle that we prepared, because gold nanoparticle is specific surface plasmon resonance at 510 to 540. And this is and then we can easily analyze that the nanoparticle is prepared, and they have the different from the phytochemical. So next is another one, the same pollen for preparation of silver nanoparticle, and then we have used for the just identifying of the antioxidant activity. And we found that they have the similar, a little bit of higher antioxidant activity in comparison to the phytochemicals in here. So the third one, that is the same way that we are going to put on here the China rose, and they have the pollen. So because pollen have own significance, pollen is also a very good source of bioactive molecules or bioactive compounds. Then people are going to eat it, or they can use it for the different applications. So this is the rosa hibiscus, we can say that the China rose, and we can put the pollen, and then again it is used for the preparation of we can say that the silver nanoparticle, and then after it is degradation of the thioflavin. So indirectly we can say that whatever the nanoparticle we target to prepare is used by the plant, the different material or the phytochemicals, and then we can screen it for their size, stability, and then you can use for the some application. And this is another one that says that rambutan peel. It is one of the, which is also called as hairy lychee, and it is originated from Latin America, Indonesia, and Malaysia. And it seems like a lychee, but their peel is a rich source of, we can say, the anthocyanins. So anthocyanins are also there. So they have the light, but no one is going to care about this. So they can throw it, they can, and it is discarded material. So we prepared in the another paper, one of the paper in molecular liquids, we have prepared the triangular and hexagonal nanoparticles with nanoplates, and then we use for antioxidant activity. And another way, we can dope with the titanium, and we can use for the degradation of methylene blue dye. So it is the valorization of waste material to nanoscience. Another one, we can say that a rambutan peel in the third way. we can prepare with the silver, gold nanoparticles, and then actually, if we prepare the nanoparticles, gold nanoparticles with the rambutan peel, it is not stable, but it comes in the form of a group, means seems like it's gold nanoflower, and then that gold nanoflower we again apply for the is a part of the catalysis for the conversion of nitrophenol to aminophenol, and then means indirectly we can say that again we can say that different application on here. So I'm going to put on there, if this is a kapuli, it seems like a jamun, but they have the different effect, because sometimes always researchers are asking, if we are going to prepare a nanoparticle, what is the optimum condition? Means either we can heat it, either we can put the sunlight, either we can put the ultrasonication. So this is the one of the study which shows that if we are going to the same material with the silver nanoparticle, if we are going to put in here for the normal condition and we can expose to LED light, so they have the different morphology. There is a not big, but there is a change in there. So white, and then also their nanoparticle size is they have the monodispersement. They are not a variety of size, which they are in a range of size is means they have the collection, means we are going to range of specified, not very big. Otherwise, if you expose for a light or keep it in a normal condition, the nanoparticle size is growing, and then after 100 nanometer it settled down. So there is no use of that nanoparticle. So the main thing is that whatever we prepared, we keep it stable for 2 to 6 months. And another one again of the this one is the same with the gold nanoparticles. We found that this nanoparticle is again used for the degradation of dye, and then you can use for the same other application. We can easy. And then this type of nanoparticle you can see here means small nanoparticles. If there is not a stable molecule, they can collide and they can make a triangle shape, or they can make a hexagonal nanoplates. And this is the copper oxide nanoparticle that is prepared by the Kapuli, because it is a very good rich source of, we can say that, flavonoids. And then and if there is a flavonoids are there, the peoples are also drinking like a colada, means we can put in colada morada, means they can put some juices and they are in a regular diet, and they can also use for the degradation of thioflavin dye. So this is the Fiscus carica fruit. Always we are going to eat anjeer, and then the fruit. We can prepare the nanoparticle in another paper with the heating, with the ultrasonication. So we found that in case of ultrasonication, they have the size is not, they have the more selective size in comparison to the heating. So this image and UV clearly explain about the nanoparticle size. So another one is the nanoparticle with the iron nanoparticle. This is a very good paper that was published in valorization, waste and biomass valorization in 2021. And in this, actually, if we are going to prepare an iron nanoparticle, the main problem is that we can prepare iron nanoparticle, but due to the magnetic property, means when we have already we knew that the iron nanoparticles, iron is magnetic, so it is very tough to take the nanoparticles, because after some times all the iron is agglomerated to form, and then it is not easy to analyze the in time. So the main problem is that, so before analysis we can put the ultrasonication, then check the iron nanoparticle. But the good thing is that they have no toxicity, and then we can apply for a different way, because we are going to, and it is very low cost preparation. So in case of there, we can see that if it is an iron nanoparticle, and normally iron has no antioxidant activity when in our body, but when some molecules are surrounding or coating about the iron nanoparticles, they have some antioxidant activity. So iron nanoparticles are also used as a antioxidant or drug carriers. So these are some characterization technique, and the good thing is also that if you prepare the iron nanoparticle, there is no need to filter. Just to put a bar magnet, we can add some dye, and just all dye are adsorbed on the surface of iron nanoparticle, and then we can put the bar magnet and we can separate it easily. And this is the different application about the iron nanoparticle from the, we can say that using the degradation and then using the antioxidant activity. So this is the another story about the Lantana camara fruit, because Lantana camara is one of the, we can say that notorious weed that is all over that is found in all over the world, but there is no application, because some reports, there are some reports that people can eat Lantana camara, the green shade, and they have a stomach pain in there, but there is no use. So our target is that why not we can use this Lantana camara or discarded weed for some application. So we prepared Lantana camara with the silver nanoparticle, and then we checked the antioxidant activity, and we found that they have some antioxidant activity. Then we put the Lantana camara leaf with the gold nanoparticle, and then we can say, we can observe that Lantana camara gold nanoparticle, gold seems to be yellow color, but their nanoparticle is red color, and its size is in between 5 to 15 nanometer, and it behaves very good catalyst for the degradation of methylene blue, because methylene blue, methylene orange, some are all are synthetic dyes and normally used in the dyeing industry. So they are good. So it is also, it is important that synthetic materials must be, we are going to degrade by using the nanoparticle, and the nanoparticle which is prepared by the plant materials. So another one, that is a mortino berry, that is the mortino, or it is a, this is also the, it is a berry, it seems like a Lantana camara, but it is not a Lantana camara. It is originated from the Andean, like the Latin America, and they have the very strong antioxidant activity. Means they have a very good anthocyanin contents. And the peoples are also drinking, like I told you, like the Kapuli and Lantana, like the Mortino, they are used for the drinking Kulada Morada. Means in the some festival, they have the some festival at the time of Diwali in November, they can drink, and it is very good, these ones. So my student, Carla Vijuti, suggested, why not, sir, we can use it. So we prepared the nanoparticle, and it found that it is a very good monodispersed nanoparticle with size is nearly 20 nanometers. So we used for the degradation of methylene blue. Again, she modified with this nanoparticle with graphene, and she prepared the graphene.
Sir, excuse me, sir. Sir, sorry to interrupt you. Sir, please conclude as we are sorting in time, sir.
Okay, sure. So this is the application with the nano composite, because it found that the initially the silver nanoparticle, it is only going to degrade methylene blue, but with the graphene nano composite, it can degrade methylene blue and methylene orange, both dye, efficiently under the presence of sunlight. So there are some result is there. This is the one of the last one of the paper on there, the citrus paradise peel, because whenever we are eating, it is very good source of antioxidant we can prepare, and then we can use for the degradation of dye. And just I am putting on here. Okay, so this is the one of the plant, that is fruit, it is called as Juniper americana. If you are going to see in the Latin America, the peoples are going to paint their face with the blue color. So why not we can use the blue color, because it is a part of the genapine, and we prepared the nanoparticle with the gold, and we can use for the HeLa cell line, A5 cell line, and antioxidant activity. So from that Amazon to that pigments are used for the drug carrier, or we can use for the antioxidant efficacy. And finally, this is the most important plant, that is that after that I'm going to conclude. This is called as Plucinatia volubilis. It is called as one of the plant well known in South America. It is known for the omega 369 vegetarian omega 369, and their leaf, Incas people are using leaf for the green tea, their oil for using cooking food, and now they have the very good, they have the very good tendency for the preparation of different nanoparticles with there, and they are the highly stable, because if you want to prepare the cube type of nanoparticle, we can use the leaf, we can use the oil of the Sacha inchi or the Plucinatia volubilis oil in the under the exposure of sunlight. We can prepare the square shape of silver nanoparticles. So we are the first, our group is the first, we reported a lot of papers, a lot of works on the Plucinatia volubilis. So, and these, so all are published papers, on there. So now I move to, yeah, okay. So on conclusion is there, the phytochemicals open a new scope to substitute the use of toxic chemicals. Phytochemicals are efficiently acting as reducing and stabilizing agent for the synthesis of silver, gold, iron, copper, tin, palladium, etc. And phytochemicals are also functionalized and they can use for organic dye and the use for the catalyst. And phytochemicals, matter nanoparticle also have the low toxicity. They are going to use for the peroxide sensing, they are going to use for the antioxidant activity, and etc. So I am going to give the acknowledgement to my group and the collaboration with my collaborating. Okay, thank you. Thank you.
Thank you so much, sir, for a very creative presentation. Thank you once again.
Okay.
Now, our next speaker is Dr. Brijesh Varse. Sir, please unshare your screen.
Yeah, sure. Yeah.
Sir, please unshare.
Okay.
Yeah. Yes, sir. Thank you, sir. So our next speaker is Dr. Brijesh Varse. He is working as Director of Life Sciences, Food and Agriculture at the US-India Business Council. Prior to this, he served as Director of Indian Industry and Research Scientist at the International Center for Genetic Engineering and Biotechnology, New Delhi. He was associated with DBT Bioenergy Project. He is routinely dealing with intellectual property rights issues, specifically with the biofuel domain. He has published various research papers, review articles, and book chapters in national or international journals. He has a broad range of experience in protein chemistry, fermentation, downstream processing, and accessing biodiversity for identification of novel biocatalysts. In 2007 and 2008, he was awarded a prestigious MIUR fellowship for young Indian researchers by the government of Italy in the field of nanoscience and nanotechnology, where he gained vast experience in specific ion effects on biological molecules and the synthesis and characterization of nanoporous silica particles with various functionality and pore size. I welcome you, sir. Please continue.
Thank you, Dr. Neha.
Thank you, sir.
Introduction and before starting the presentation, I'm audible.
Yes, sir, you are audible, sir.
So I just wanted to thank the MIT University and Dr. Raghavendra and the team for inviting me for this talk. And since the whole event is based on the bio-nanotechnology, and since morning all the participants are listening about the nanotechnology and its advancement, so I'm now changing the theme. So my presentation is entirely different because now we learn about a lot of innovations, a lot of research is going on in the field of nanotechnology or biotechnology or plant technology and everything. But important, what is more important when after doing this research, what next? This question should arrive. So my presentation topic will be on, I'm sharing my screen. Just a minute, some sharing things. Just a minute, there is some issues. Can you see?
Yes, sir.
Okay.
Sir, minimize the screen recording.
So I'm just full screen. It's fine now?
Yes, sir.
Okay.
Please continue, sir.
So the presentation is like entirely different from the theme what you are learning since morning on the nanotechnology, but this is very, very important after the research, because there should always be a question what next, because we do a lot of research, we do a lot of innovations, so we need to understand the importance of the work we are doing it in terms of the R&D activities and all. So where the IPR plays a like important role for becoming an entrepreneur and then securing your innovations, and it is always giving the like leverage for the business development as well. So before starting and going for the subject matter, I just wanted to give the brief of my organization, because I just wanted to change a little bit of thought process of the audience. Like most of them are like students, so I just wanted to let them know after doing the masters and their PhD, there is not only the R&D is the only way to get your career ahead, and not the postdocs things, because people have this standard mindset: masters, PhD, postdoc, and then academics, or in the some kind of industry and doing the R&D work. But now the scenario has been changed. There are a lot many opportunities, and one of them I'm just telling that I'm working, because I had like close to 20 years of experience in doing research and PhD, postdoc, and everything, but I've switched myself into the different domain areas by having the experience of the life sciences. So this organization is basically a US India Business Council. We worked between the US and India corridor. We work on the bilateral issues of the United States and the Indian government, and then we link the government with the US-based multinational companies. So basically, this business council support all the healthcare companies, medical devices, life sciences, pharmaceuticals, every life sciences or healthcare relevant companies to grow their business by providing them support and advocacy and the regulatory and the policy development. So we have a headquarter in the Washington DC, and it's a 120 years old organization. It's not very new. So we are into the different countries as well. So it has 18 different business councils and the 18 different countries. So let's uh, come back to the innovation sites, and this presentation is mainly for the IPR. So I just talk a little bit of basic fundamental of IPR, and then we'll just discuss about the importance of this, why this is things. So I'll just give you the brief introduction of the intellectual property, because as it has the name property, so there are like a various different type of property which all has the value. So it includes the real, like land, home, personal, like your car, jewelry, clothings, and some societal property like railroads, utility, highways, and the important is the intellectual property. So this is mainly intangible in nature, so includes the patent, copyrights, trademarks, and other different types of the things. So, from the even entertainment, we learn about the things. We don't have to run behind the success. We have to make ourselves innovative, so that success will automatically come. You are doing good research, come up with the innovative models, innovative process development, innovative product development, then automatically the companies will approach you, and then you will become a successful businessman or successful tech transfer guy in this field. So, whenever we are talking about the IPR, intellectual property rights, people normally have only one word: patent is the IPR, but it's not. There are like various different type of IPR, and one of them is patent, and the rest is copyrights, trademarks, trade secrets, utility model, plant breeder rights, and geographical indications. So I'll just briefly cover up these topics so that you can have the like refreshment of your previous maybe it may be in your course curriculum, or it's just an additional knowledge for you guys. So whenever we are talking about the patent, so we need to understand what is a patent. It's nothing. It's just a legal document. Like if you have a house and if you have a car, you have a registration of the car with you, and you have a registry of your house in form of paper, and that is a kind of a legal document which protects your property. In the similar way, when you are doing any innovation and you are filing the patent, and then you are getting the patent approved, so then it becomes the legal document for your innovations. Or any product or process can be done in a newer way, uh, will eligible to get the patent, provided it should have some elements which I'll discuss later. And just remember that patents are the territorial right. It's not like that you have filed patent in India, then you will get a uh, uh, see, coverage in the whole world. No. So if you are filing patent in India, then you will get all the coverage in India. It's like a geographical in nature, and there is no concept of the international patent. So this is very important. Like people normally get confused with, they say that I have a patent, so I am the like a god for all globe. No. So wherever in country you are filing your patent, you will just get the protection in that particular country, right? So when I talk about this patent is a legal document which is coming after your invention. So when we are talking about the invention is nothing, is just a new product or process which involves some inventive steps, and the importantly it should have the industrial applications. So the ingredient of the patent is the novelty, inventive steps, lack of obviousness, and sufficiency of the description. And the important thing we need to understand that everything cannot be patented. There are something for the society benefit or some safety purpose. Some of the things are like out of this aura. And I'll just say that like simple discovery or idea cannot be patented, and some mixture of a rearrangement of the chemical compound cannot be patented, and the new use of the known proper material is cannot be patented. Plants and animals except microorganism are not patented, but I'll talk about the plants in the later stage, how you can protect your plant. Mathematical and business methods, computer program, and so and so forth, like atomic energy and this uh, uh, integrated circuit and traditional medicine like turmeric and all, you cannot go for the patent uh, under these things, because there is a Patent Act of 1970 under Section 3. These all are things are mentioned, right? So I'll just go through briefly what is copyright, and then the copyright is nothing, it's just a legal right granted to a author. If you're writing any paper, like you are doing research, you are writing a paper, and if you're writing a poetry or you are making some kind of a sketch and all, it automatically becomes your copyright thing. But uh, it's important to get it protected by filing the paper in the offices, intellectual property office, and get the copyrighted as a certified one. Otherwise, it's your own property. Even if you, it's not mandatory to get the copyright certificate from the office, but if it is your original work, you are secured, and you can enjoy your, new innovations on that. So the same something is not protected in the patent. In the similar way, something as well is not protected under the copyright, like idea, concept, and discoveries, some title names and short phrases and slogan and something like that. And the important, interestingly, the copyright patent has some limited uh, time period to enjoy your innovation period, but the copyright is just for the life of the author. Even the author died, then another 70 years will be given for their thing. Like if there is any musician or there any lyricist, if they write any songs and all, so they will enjoy forever of their life their copyright protection. And then even after they died, if some other person is using their creative work, they have to pay royalty to the concerned person, right? And same goes for the industries as well. They can enjoy the 95 years for the corporations. Now we talk about the trademark, and trademark is a word and it's a phrase, it's a symbol or design or combination. This is actual very, very important to have a trademark, because it shows your business value. And you can see the TMR, trademark, registered, and SM is service mark. So these are all the things that I'm just giving you some example, because if you have a good trademark, it shows your value in the market, and based on your value, your business can automatically be flourish. So if you see some very famous brand like Colgate, Nike, Pepsi, this Coca-Cola, McDonald's, Pepsi, something like that. These all are like a trademark examples you can see here. So it's very important to protect your trademark, because trademarks are protected under some laws, and trademarks are earned, not born. As soon you register your trademark or you are coming up with any kind of trademark and you are doing any business in the market, if you have a quality and the creativity, So it automatically become your value. So let's take the example, even if you're going for the market and buying any noodles, so doesn't matter in which brand you're looking at, but in your mind and your thought, you always thought about the Maggie because they have a value. They made it like this so that even the person is looking for the similar type of product, but having a mindset of the Maggie as a brand. This is called as a noodle. So this is the kind of example you can understand the importance of this trademark. Other than the trademark, there are trade secrets where you don't have to register, just you have to to keep a secret of your composition or of your business ideas or anything. So for trade secret is information that is important to the business or company and it's not open for the public. So in trade secret, normally, if you see the examples of a trade secret, then you can understand the importance of the trade secrets. There are like various ways to protect the trade secret. Like you have to restrict the information and you have to sign the non-disclosure agreement whosoever is known the business idea or the composition of your product. So you see, this is the famous trade secret. No one knows the formula of Coca-Cola. No one knows the this McDonald's special red sauce and green sauce composition. That is why they are having a global business and no one can replicate their things. Same goes with the KFC chicken. And this is important. WD-40 formula is anti-rust spray. So these all are the like a famous trade secret, and you can understand that they haven't filed any patent. They have not made it anything like which it discloses to the society and the public domain, but they made it as a trade secrets, and because of that the business is like completely very in a great way, and they are leading the globe on their business model. So, and other form of the IPR is the utility model and the design model. Utility patent issued for the invention for the new and useful processes like machine and the composition of matter or new or useful improvement thereof. So there is a very little different. The utility patent is for the machines and all, and design patent is for the ornaments like jewelry and other things. But you don't have to pay any maintenance fee for these utility and the design this design patent, but some of the nominal fee for the utility patent. Now, since this conference is for the like a biotechnology, so we talk about the plant breeder rights. Since plant and the animals are not protected under the Patent Act, but there is a plant breeder right wherein this right can be granted to the breeder if they are coming with the new variety of the plant that gives the exclusive uh, things, and then there are certain things like your plant should be very new. It has not been commercialized for more than 1 year in the country. It should be distinct, and the important thing, from generation to generation, there should be a uniform growth and the activity of that plant material, and it should be very stable. So if you are like complying all such parameters, then you will get the plant breeder rights. Like in the morning call, like you heard about this mustard oil, this cotton as BT cotton has got the new variety, and then now people are trying to get more and more new variety by doing the genetic engineering. So the plant biologists will get benefited if they are following all such parameters, so they can have their own uh, property in terms of the plant breeding right. And the last one is the geographical indication. This is also a part of the intellectual property, and GI is the name or sign used to certify the product which correspond to the specific geographies. And some of the examples of the GI is like basmati rice, Coorg in India, and Darjeeling tea, Alfonso mango, Kolhapuri chappal, Tirupati laddu, and there are many more new addition into the GI list like Pashmina shawl and all. Many things are like now included. And in the global level, there are like Canadian whiskey, Switch watches, Florida oranges, and champagne. These all are the geographical indication, and it has a value when you are doing the import and export business. You will get a great value of your product, and it reflects and it gives your country has an importance for that particular product, right? So here I'll discuss about the different forms of the IPR so that we will not get confused that only patent is the intellectual property. There are various different forms in the intellectual properties are there. So I'll skip this because these are the bureaucratic how you file the patent in India. Some bureaucracy like we do normally whenever we are submitting any application, you have to follow our such form 1 2 sub 26 for different things. I'll share this presentation with all the students so that they can read it and they can get more idea about it. So when we are filing the patent application, it only makes the difference because it is simply like the research paper where you have all the information. But the patent only makes the difference because in patent document, you are claiming something. You're claiming for new process, you're claiming for any product or any new thing. So this is the only difference between the patent application and the research article. So these are the like a procedure how to file. Like you have to file if you got an idea and you are like try to translate your idea into the product, then you have to file the provisional application where you will get a 12 months time to uh, complete your research work, and then you file the complete application. If you are not able to file within the year time, then your application will become a prior art and your application can be abandoned. But if you file the complete application, then the 18 months after 18 months your application will be published, and within after publication within you will get a 48 months to file for the examination. Then examination will be there. Once you get the examination report, then your examiner may accept your application or reject your application or may raise some of the queries. Then final after completing all the process, your patent will be granted and you have to pay the maintenance fee and you will get it.
Sir, sorry to interrupt you, sir.
Yes.
Please conclude the session as we are running out of time, sir.
I'm just concluding, right? So I'm just skipping this because that is the Indian patent system, and there is the PCT system where you can file one application and you can get the access to enter into the 160 different countries. There are certain things how you because that was the like a brief introduction of the patent, but you can understand you can monetize your IPR by doing the licensing. You can open the spin-out company and you can have some different business development activities. So this I've already covered in my things, the benefits. And simply like the IT, I just wanted to let you know, guys, that the IT was the like a main hub for these technology things, but now biotechnology is coming up as a boom. So there is a lot many opportunity if you are filing the patents, so you are going for the trademark and the copyrights and all. So there is a lot great opportunity for the biotech business in the country. And there are some breakthrough innovation. You can understand that how they have done the things like gene scissoring, CRISPR-Cas9. You have heard about the CRISPR-Cas9 presentation and the previous things. There are breakthrough. There is a. I just wanted to let you know that this is a Lachel smart shoe, the red color. It's an Indian-based company. They come up with the new smart shoes for the blind people. And some company like Adidas is using the spider silk to make the biodegradable shoes material. These are the new innovations. In the medical sciences, people has made the nanogels, which is like treating the burning in a very less time. And this is the high-end nanotechnology based research for the burning people because it's very great innovations has been in the market. There are many other things which comes under the innovation and the medical sciences by the biotechnology fields. And there are many various opportunity in the IPR as well if you don't want to go into the research. So you can go in the different field and make your career on that. With that, I just wanted to conclude and thank you very much.
Thank you so much, sir, for such an informative talk on IPR. With this talk, session 2 is concluded and we will continue for session 3rd. Before it, I request to all respected speakers and faculty of AIB, kindly open your camera for group photograph, please. Thank you all. Dr. Asha, please continue for session 3rd.
Thank you, ma'am. Finally, we are moving forward to the session 3, secondary metabolites and metabolic engineering. The chairperson of this session, Dr. Anurag Sharma. Currently, he is an assistant associate professor in the Department of Environmental Health and the Toxicology at NITI University Center of Sciences and Education Research, Mangalore, Karnataka. And the co-chairperson of this session, Professor Dr. Vinay Dwivedi. And the invited talks are Dr. Anurag Sharma, Dr. Pankaj Rai, Dr. Praveen Avasti. So one by one, we'll start the invited talks.
Sir.
First of all, I would like to call
Yeah, hello, sir. I'm audible. Oh, Praveen Avasti, sir. Okay, okay, sir. Okay, okay.
Dr. Praveen Avasti. The Dr. Avasti has a PhD in biotechnology from CSIR, Indian Institute of Integrative Medicine, India. Dr. Avasti academic journey is marked by excellence and innovation. With the extensive research experience spanning over the decade, Dr. Avasti has contributed significantly to the field. His previous role include the prestigious positions such as postdoctoral research fellow at the Institute of Plant Molecular Biology, Cranes Republic, and the project scientist at the National Agri Food Biotechnology Institute, India. Dr. Avasti research endeavor has been recognized with a numerous award and the honors, including being selected as an invited keynote speaker at the International Conference of Plant Genome Genetics Engineering and the Genome Editing at the Central University of Kerala. His dedication to advancing the plant science is the further underscored by his receipt of the postdoctoral fellowship from the Cranes Academy of the Science and the CSIR Senior Research Fellowship by the Government of India. I welcome to Dr. Praveen Avasti, sir. Dr. Praveen Avasti, are you there? Okay.
वॉइस ऑडिबल है मेरी? ठीक है।
ठीक है, स्टार्ट करो प्रवीण।
Okay, I'm just sharing the slides. Huh?
Yeah.
And okay. So,
Hello.
hello, slides are clear. Everything is okay.
Yes, sir. Your slide is visible, sir.
Okay,
Yes,
okay,
sir.
okay. Thank you. First of all, I want to thank Professor Vinay Dwivedi for giving me opportunity to present in international conference in nanobiotechnology at Amity University. So today I'm going to talk about my work which I done in Czech while working in Czech Republic on hop CRISPR work. So before starting with my work, I just want to give a brief introduction about my area of research. I'm working on CRISPR-Cas9 genome editing approach in plants. So when we talk of genome editing is basically a modern day tool in which is used to do the knocking in, knocking out of a gene. And that was previously evolved in 2000s, and generally previously people were using zinc finger nucleases in which a protein was fork1 enzyme was attached to the DNA helicases, but it was very difficult to design the target for that. But after zinc finger nuclease, people moved to talon, which was an advanced version of zinc finger, but also there was limitation in identifying the target. But in recent years, this CRISPR-Cas system, which use a Cas9 gene, is very efficient way of knocking out the gene to make disrupt the function of a gene in plant system, in animal, in like prokaryotes also. So this is a very widely used genome editing methodology. And when we talk of CRISPR-Cas system, Cas9, there are two types of enzymes which do this deletion, class 1 and class 2. Cas9, which we typically use in our genome editing approach, fall to type 2. And in type 1, we have a cascade of Cas gene, in which is a multimeric protein complex. So to get proper targeted mutation using type 1 and type 3 is quite tedious, and it's very is not feasible till now. But normally using Cas9, it is very easy. With Cas9, Cas9 make a double strand break, and we have also enzyme like Cas12, which is also known as CPF. It is also used for cleaving double stranded DNA. But the difference between Cas9 and Cas12 is that Cas9 produce blunt end in the DNA, whereas Cas12 create the staggered end in the DNA. And Cas13 has been recently also discovered, which is used for RNA editing. So what we can do with Cas9 system, first is knock-in and knockout. Knock-in, if you want to insert a particular gene segment in precise location of your choice. And in knockout, if you want to delete a particular sequence. So what we have, like we firstly design this sgRNA. sgRNA is contain the target which is complementary to the DNA sequence which you want to knock it out. And then we have a Cas enzyme. Cas enzyme and sgRNA make a complex known as RNA ribonucleic complex, and this bind to the DNA fragment. Then Cas enzyme has two DNA binding cleavage sites, and they cleave the DNA target at 3 nucleotide base pair from the PAM sequence, and this cleavage lead to this double stranded break. And when the double stranded break is made in a system, in a DNA, in a cell, what happened? It can go either through non-homologous enjoining methodology or homology directed repair mechanism. 90% homology directed non-homologous enjoining method is prominent in plant system. And if it goes to non-homologous enjoining method, it's lead to formation of indels. When we call indels, it means it can create insertion and deletions. And when these insertions and deletions are made, it lead to frame shift mutation and finally to non-functional protein, and that lead to a knockout of enzyme. But if you want to go through homology directed repair mechanism, in this, if you want to insert a particular fragment in this double-stranded break site, you provide the homology arms which are complementary to DNA sequence, and then you also provide this vector, and this lead to SDR, homology directed repair, and this lead to knock-in of particular gene of interest or particular sequence of interest. And getting this like a view of what is CRISPR-Cas system, how it is work, and this is like the two enzyme RUC and RHN. These are the sites which do the cleavage of DNA sequence. After getting this overview of what this Cas system is, how it's work, what are the different classes of Cas enzymes like class 1, class 2, we all credit goes to Jennifer Doudna and Carpentier, which we know that in 2020 they got the Nobel Prize in Chemistry in discovery of this CRISPR-Cas system. And it is said that this CRISPR-Cas system, after development of the recombinant DNA technology, it will be the fifth revolution in the science of genetic engineering. And that's been implemented in plant widely, and it's been impacting the human life in a very progressive way. So here are the few examples. Like when you take the from the market an apple, when you like cut it, and if you keep it for some time, you get this reddish brown color. This is basically because of excretion of TPO gene, polyphenol oxidase. So with use of CRISPR, people have blocked that, knock out that gene, and it has improved the viability of apple and increased the shelf life on apple. So this is already been in the market, and Vaxi gene of amylopectin in this corn, and similarly their report of this button mushroom of CRISPR-Cas, this including PPO, which has like brown patches in mushroom. They have knocked out this gene PPO, which lead to eradication of these brown spots in mushrooms. And these are another agro 8 variant genotype of CRISPR-Cas, which is used in maize and improving the mildew resistant genes. These are the few examples which are used, which are being developed, product had been developed using this CRISPR-Cas technology. And now this is the very important, this about very much, this is from Cold Spring Harbor Lab in collaboration with NASA. What they have done, they have just knocked out 3 genes in tomato plants, and you can see what different it has made to the plant system. A plant is full of tomatoes, so it has increases the yield with such an impact. So this shows that what an impact of CRISPR can have in field of agriculture, increasing productivity, removing hunger, increasing nutritional value, and lots more. So these are the few examples that already been USDA approved CRISPR edited plants for the commercial release. And this data is of 2018, and most of them are under, a few of them are already in the markets, like white button mushroom, waxy corn, Camilla for improved oil content, and drought tolerant soybean. So these are some examples of CRISPR-Cas. Now moving after getting the what CRISPR's application in plant system, what is CRISPR-Cas, how it works. Now I'm going to share my research, which I carried out in Institute of Plant Molecular Biology in Biology Center, Czech Academy of Science, Czech Republic, where when I was working as a postdoctoral fellowship of Czech Academy of Science, I was working in a hop plant, which is also known as Humulus luteus. It is a commercial and the main crop cultivation in Czech Republic. And the use of this hop crop is that the cone of this is known as cone of the female flower of hop. It is used in beavering industry in beer. And in beer industry, Czech is known for the production of various Pilsner and Budweiser's are the various beer industry in the Czech. So when we use this cone, because in cone they have lot of these dot you're seeing, these are lupulin glands, and they contain prenyl flavonoids and bitter acids, which give aroma and flavor to the beer. So the lab which I joined was working on increasing understanding the metabolic pathway of this prenyl flavonoid and bitter acid biosynthesis. So before when if we go by traditional breeding method, it took almost 9 to 12 years in hop to get one variety, and that to be a very untargeted way. Cone and what are the agronomic traits which people look for in hop breeding is like cone yield increase, number of cone in the plants, and secondary metabolite like bitter acids, prenyl flavonoids, and other essential oils should increase disease resistant. But the problem with hop is that incompatibility, because male and female pavlovas are different. So breeding is a very challenge in hop. So the only approach, viable approach left for improving this crop was through like genetic engineering. But so what this is basically prenyl flavonoids and bitter acid biosynthesis in hop. So prenyl flavonoids and bitter acids are formed from the phenylpropionate pathway. You can see this phenylpropionate pathway. This is xanthomenol, and this humulone and lupulone are the bitter acids is coming from this pathway. So before I joined the lab was working on this pathway, and they have worked on some transcription factors like MIB2, PHLS2, WDR1, WIRKI1, WDR2, MIB8. These were the transcription factors which they have already discovered and known their function, that they regulate the pathway gene. And but what the problem was that when they did the overexpression of this gene, they were seen ideally if you overexpress this gene, it will impact this pathway gene, and this molecule xanthomenol should increase. But when they do the expression in leaf, there was bit change in the content of metabolite, but in the cone there was no change. So there was something fishy going on in the cone, that why after overexpressing this transcription factor, there is no change in the cone. So we did the transcriptome analysis of lupulin glands, which is also known as trichome. And we found some interesting metabolite. We did a transcriptome and metabolite form. So we found some interesting enzymes which are regulating transcription factor. So firstly, we were upregulating this transcription factor, and we were expecting that gene should increase and with lead to a metabolite increase. But what was happening when we increase the transcription factor, there are some regulator gene which are regulating this transcription factor. So if we increase this transcription factor, this regulator gene are suppressing the expression of this. So there was some other transcription factor regulators also. And when we looked into literature and transcriptome data of our, we found that there are MED5 and 5B, which are known as mediator protein. Like gene are regulated by the transcription factors, and then this mediator complex regulate the transcription factors. So we found interesting paper from Arabidopsis that these two were found to be regulating biosynthetic pathway of flavonoids, and we were also working on a flavonoid pathway in hop. So what we did, we also want to study the role of this MET5A and 5B in hop. So when we discovered this mediator 5 complex, this we found that the discovery was firstly was made in yeast, and mediator complex was successfully, this is about the history of what mediator 5 and 5B had been done. And there are around 20 to 30 mediator complex which are reported in trans system, which work in regulation of transcription factor. Our interest was made of mainly on MET5A and MET5B. Why? Because when we find the transcriptome data, we found that mediator 5 and 5B are highly upregulated in the trichome. And that's what we are thinking, that if we are overexpressing this gene, then also we are not getting higher accumulations and tumor. That's because of this could be mediator complex regulating this transcription factor. So, but to understand the role in this, we need to knock out this gene. And to knock out this gene, firstly, we need to do CRISPR. So here, like you can say 5A, 5B is differentially regulated using qPCR data. So what I did, till that time, there was no report of CRISPR-Cas in HOP. So we need to establish this CRISPR-Cas system in HOP. So we use marker gene phytoin desaturase, and we designed 2 sgRNAs and use multiplexing, golden gate assembly to assemble in the vector, and use plant codon optimized Cas9. And when we do the transformation, you can see that we get the clear-cut albino phenotype. Why? Because if you knock out the PDS gene, it leads to a albino phenotype. This is normal leaf. And when we knock out this PDS5 gene, the leaves are become like this. But you can see we also got plants in which you get white and dot green. This is because there is chimeric. And chimeric plants or mosaic plants are very common when you do tissue culture using explant like leaf or nodal segment. So we also get that chimeric, and we were very happy that we are able to establish CRISPR-Cas system in hop. And till now also this chimeric beautiful leaf plants is in our Czech Republic, in our greenhouse. And when the kids, school kids come, we show them that we established the system. And then finally we done the sequencing, and then data analysis, which shows that there were deletions, such big, so 15 base pair, 30 base pair deletions, and these are the raw chromatograms of the mutant lines. We done some analysis using T7 endonuclease assay, which in assay to determine whether the mutation is homozygous, heterozygous, or chimeric. So this is the plant which I was talking in our glass house and in Czech Republic. And now this was the first report of CRISPR-Cas in hop, and it was in lot of in media. We have an interview in our news channels, and it was also news in extracts. This our work has catch a lot of media attention. So we finally published in Plant Physiology and Biochemistry Journal, and that was the starting motto of our CRISPR-Cas establishing in hop. Now what we need to do, we need to after establishing, we need to knock out 5A and 5B gene. These two we need to knock out. So I prepared 3 constructs. One construct knocking out only 5A gene, one construct knocking out only 5B gene, and one construct will knock out both the gene. So 3 constructs were prepared. We clone the gene, find the sequence, and then we design the sgRNA, make the construct, make the transformation. So this is a just 12-week cultivation. But one interesting things arrived. We were thinking that bitter acid content will increase. But what strange thing we found, that in a 5A mutant, the cone size has been increased as compared to wild type, which was very, very like interesting thing. We were expecting something will happen in the metabolite, but the cone size increase, which indicate that this 5A gene is having a multi-dimensional role. It's not specific only to modulating the metabolite, it also regulating the physiology of the plants. And we done the transcriptome and profiling of this lupulin gland. and metabolite profiling. So we found there was increase in the content of bitter acids and terpenoids, and there was also upregulation of structural gene, a pathway gene of bitter acid and phenylpropanoids, which was corroborating and which was very exciting for us. And these are the mutation which we got in 5A gene. And when we do the double knockout of 5A and 5B gene, those plants cone size were the same, which shows that 5A gene mutation lead to increase in cone size. But if we knock out both 5A and 5B gene, cone size doesn't change. That means like which
Excuse
give
me, sir.
an yeah.
Sorry to interrupt you, sir. Please
Yes.
conclude the session, sir. We are running
Okay.
out of time, sir.
Okay, just I'm in the end. So that's what we got the data, and what conclusion we make that 5A is associated with a negative regulator of phenylpropanoids and 5B with a positive regulator. So for this work, I want to acknowledge Professor Joseph Spack, director of our Institute of Plant Molecular Biology, Dr. Tomasz was our head of department, and Dr. Yaroslav Matusek and Dr. Ajay Kumar Mishra for helping me in transcriptome, and Miss Andrea, our technician, who was taking care of lab things. So, and I also want to give the funding agency Gacha for funding this work, and thank you. That's all from that side.
Thank you, sir.
Yeah.
Thank you, sir, for the wonderful lecture, sir. So question answer session we will discuss after the completing the session.
Okay.
So now I'll call the next speaker. Dr. Anurag Sharma. Currently, Dr. Sharma is Associate Professor, Department of Environmental Health and Toxicology at Nitti University Center of Sciences Education Research, Mangalore, Karnataka. Dr. Anurag has a PhD from CSIR, Indian Institute of Technology Research. His early research honed in the understanding cellular and molecular stress response to environmental toxicants using the Drosophila melanogaster model system. Transitioning to center the biology, the development, Toulouse, France, for his postdoctoral tenure, he delved into the blood cell development using the same Drosophila model system. Currently, Dr. Anurag's focus lies in the unraveling the molecular mechanism behind the environmental toxicant-induced immune injury and exploring host-microbe interaction due to the environmental toxicant exposure. His contribution include over 25 published research papers in SCI Index Journal and a 9-book chapter. I welcome Dr. Anurag Sharma to deliver his lecture. Sir, please.
Thank you, ma'am. I'm audible?
Yes, sir, you are audible.
I'm just sharing the is it in full screen now?
Yes, sir.
Okay, so can I start now?
Yes, sir, now you can start.
Okay, so thank you, ma'am, for nice introduction, and I also thanks to the organizer, Dr. Anurag Sharma, and sorry, Dr. Anurag, and to introduce me on this platform. So basically, as you said, this is a little bit different talk which we are listening right now. So we are using, or we are developing basically a invertebrate model system to understand the environmental toxicant induced hematopoietic injury. This is our basic goal of our lab. So basically, as we go our routine life through the industrial or from the agricultural or from the anthropogenic activity, n number of chemicals are introduced in our environment, and directly or indirectly we are exposed with all these chemicals, and they can pose a negative impact on our physiology. It can hamper a different type of organ system to us. And so here I am talking about the blood cell development. Why I am particularly interested? We are particularly interested in blood cell development because a, a good immune system is always needed for any type of to maintain the homeostasis, and this is known. These environmental chemicals and these immune system is highly prone to any type of such stresses because they are highly metabolic active cells. So here our so the only problem is because we are interested in the blood cell development, and so the process we call is like hematopoiesis here. So because if you compare the invertebrate hematopoiesis, it's really very complex, and the bone marrow, which is like the home of the hematopoietic all these uh, like all the program in what we call vertebrates, it's very complex. So that's why we tried. Why don't we use this invertebrate model to understand the complex hematopoietic problem induced by the all these chemicals? So we tried. So if you see the hematopoiesis in the vertebrates, basically it happens in the bone marrow. And in bone marrow, we have the stem cells, we call hematopoietic stem cells. These hematopoietic stem cells are responsible to make all type of blood cells, erythrocyte or what you call myeloid progenitors, or and these are the differentiated blood cells, and these we call the endothelial niche, and this is the vascular niche. So these are the, you can see, this is the stem cells, and these are the differentiated cells, blood cells, and these are the two niches, endothelial niches and vascular niches. They are basically a non-hematopoietic cells which controls these stem cells progenitors, and they maintain them. So this is a basic structure of the bone marrow of higher vertebrates. But if you compare the fly hematopoiesis organ, so similar, fly also have a hematopoietic organ, and this hematopoietic organ we call lymph gland. So here also you can see they also have this blue part, is they also have the stem cells. They're the blood stem cells here, and these are the like differentiated hemocytes, or what blood cells in the fly, and these are the endothelial niche, and this is the vascular niche. So what I want to focus here, why don't we use this model to understand the hematopoiesis for the higher vertebrates? So this is a basic model which we are which I am using, that's like this lymph gland. So if we talk about the blood cells in the flies, definitely fly doesn't have lymphoid organ, what lineages. They only have the innate immunity, and they have the myeloid progenitors and what are myeloid cells. So fly have three types of blood cells, like macrophages, and second one is platelets, and third one is lamellocytes. So they are just a counterpart of macrophages and platelets in the human beings or higher vertebrates. And if you see the development of all these cells, fly also share the similar type of like signaling pathways. So what I can say, the functionally the blood cells, especially for the innate immune response, or we can myeloid progenitor, they are the same in case of development also, and in case of function also. So these are the two things I just want to introduce here. So here, as I told, I am working, and these are the other important, like why this fly is important for the research, for what biomedical research, because they have this short life cycle, low cost maintenance, well dissected genetics, and like homology with the uh, like humans genes, and high fecundity rate, and easily available transgenic animals and the mutants, and most important, limited ethical concerns, which makes Drosophila as a ideal model for the preclinical or biomedical biomedical research. So here I am talking about one of the important like toxicant benzene. Benzene is basically a monocyclic aromatic hydrocarbon, and this is widely used in the industry as a solvent. And if you see all this toxicity is known, like and if you talk about the hematopoiesis, also hematological malignancy is also known, like benzene cause. So here we took benzene as a reference chemical. So whether the same type of effect whether fly also are having if we expose this benzene. So this is our plan. So what we did, we just mix the benzene in the food, then we expose this larvae, and after some times like 24 or the 48 hours or 96 hours, we dissected this hematopoietic organ, lymph gland, and then we analyze the what different kind of cells or different kind of response. So if here you can see after the benzene exposure, we got a reduced what plasmatocytes, which is basically the macrophages. And second, we also got the less number of hematopoietic stem cells also. And here we also found a aberrant number of a different type of cells, lamellocytes, or other type of cells. And this is also known benzene also produce lot of ROS. So we also check whether benzene is producing ROS or not. So here we found that a lot of ROS is there when we expose with the benzene. And most important thing, we got like a lot of cell death here. Here you can see this is the control, and here you can see the caspase 3 staining, which denote the cell death. And here also cell death in the hemocyte or blood cell progenitor. So all together, and when we prolong the exposure, like for 96 hours, so we got a tissue injury also. So here you can see the gland, but after the 96 hours of the benzene exposure, there is a tissue injury here. So here, if I summarize this thing here, like after 48 hours, we are getting the reduced progenitors, we are getting the aberrant differentiation of the blood cells, and the ROS generation and cell death in the hemocytes also, and in the progenitor also. And after the prolonged exposure, this lymph gland is going to burst or it's going to damage. So if I compare all this phenotype in the human or higher vertebrate, so it looks like this. Yes, aberrant blood cell production also is correct here, and we are also getting the same response, like same reduction in the in like blood cell progenitors. Yes, in mice is also is there, fly is also there. And oxidative stress, yes, we got the ROS. Yes, ROS is also reported in the higher vertebrates. In fly also, we are getting the same thing. And the death in progenitors also reported in the mice, and we also got the this. Then this is our basic baseline. Okay, whatever